in Animal Sciences, University, Ludhiana, Dr. Yashpal Singh Malik, Dean, College of Animal Biotechnology, GADVASU, Ludhiana, other officers of the university, our esteemed dignitaries and resource persons of the day, fellow colleagues, participants, and dear students. I, Dr. Simranpreet Kaur, on the behalf of the organizing committee, wish you all a very good morning and welcome everyone assembled here on this virtual platform on the occasion of International E-Symposium on One Health Approaches to Tackle Zoonoses, which is being organized by the Center for One Health, Guru Veterinary and Animal Sciences University, as a part of the celebrations of the World Zoonoses Day. Well, just briefing you all about the World Zoonoses Day, it is an annual opportunity to raise awareness of those diseases which can spread between animals and human beings and vice versa, and to celebrate the global activities taking place to minimize the risks in the future. This date has a special significance in that it marks the scientific achievement of Louis Pasteur, who successfully administered the first vaccination against rabies on July 6, 1885. This year, the World Zoonosis Day holds significance in the wake of the novel coronavirus disease, that is COVID-19 pandemic, wherein the growing severity of COVID-19 is compelling us to rethink ways to reduce transmission risk of emerging diseases from animals to humans and operationalizing the One Health approach at the human-animal environment interface. Therefore, it is high time now for us to be aware of the zoonotic infections and thinking about changing our relationship with the animals in order to avoid and prevent such future pandemics. So not taking much time, I would now like to invite Dr. Jasbir Singh Bedi, the Director, Center for One Health, to welcome the dignitaries and participants on this platform. Dr. Bedi, please.
Thank you, Dr. Simran. And a very good morning to everyone. Greetings from the Center for One Health. A very happy World Zoonosis Day and a very warm welcome to the International E-Symposium on One Health approach to tackle zoonosis on the World Zoonosis Day 2021. Sir, it is my privilege to welcome today's guest of honor, Shri Inderjeet Singh Ji, Vice Chancellor, Guru Angad Dev Veterinary and Animal Sciences University, Dr. R.K. Singh, former Director, IVRI, Vijayanagar, Dr. S.P.S. Bhuman, Dean, College of Veterinary Sciences and Principal Investigator of ICAR IDP NEHAP project, officers of the universities, heads of the various departments, other distinguished dignitaries, fellow colleagues, and dear participants from various parts of the country. So first, let me give you a brief about the Center for One Health, which has been emerged from the Department of Veterinary Public Health and Epidemiology, which was established in 1984, and it was upgraded to School of Public Health and Zoonosis in 2012, and then to the Center for One Health in 2021 under the dynamic leadership and visionary Dr. Inderjeet Singh, Vice Chancellor, GADVASU. The Center for One Health, along with the prime mandates of teaching, research, and extension activities, has various collaborations with the national and international funding agencies, including DBT, UGC, ICAR, ICMR, Gates Foundation, University of Saskatchewan, Sydney University, Australia, Royal Veterinary College, London, etc. And this center is also associated with the various awareness drives on many important zoonoses, such as brucellosis, rabies, hydatidiosis, cryptosporidiosis, along with these antimicrobial resistance, climate change, etc. And this center has also initiated a one-year diploma program in One Health. As we all know that today is the World Zoonosis Day, and the importance of this zoonosis can be judged from a figure that out of 1,415 species which are known to be pathogenic to humans, 61% are considered to be zoonotic in nature. And 175 pathogenic species which are associated with the disease considered to be emerging. And out of these, 75% are considered as zoonotic in nature. The various factors, they are playing role in the emergence or re-emergence of the zoonotic infection, such as change in the biodiversity of animals, domestic wildlife-human interface, rapid or transportation or trade of the wild animals, lack of the biosecurity system, increasing tourism, etc. So the World Zoonosis Day is a very important to mark all these ongoing activities. And it is also important for everyone to understand more about the diseases which are caused by animals and the ways in which we can protect ourselves from these diseases. This day also promotes good health for both animals and human beings, and also highlights the importance of veterinarians in our life. And the One Health approach, we all know that it is the collaborative efforts of multiple disciplines working locally, nationally, or globally to attain the optimal health for people, domestic animals, wildlife, plants, and our environment. And many past experiences and studies have provided the enough evidence that successful application of efforts of One Health framework has yielded positive outcomes to combat the problems which are emerging at the human, animal, and environment interface. And moreover, to deal with the existing as well as other emerging public health challenges, such as we are encountered with the COVID-19, these problems, they cannot be dealt effectively within a limited professional silos. So One Health framework, by using intersectoral collaboration, capacity building approach, they are the need of the hour. And without going into too much details, I would like to inform you that we have many eminent national and international speakers to take this topic into further finer details. And this is a great opportunity to listen their words of wisdom and clear queries regarding the various zoonotic diseases. So again, I welcome all the participants, all the dignitaries, and all the officials of the GADVASUs, as well as Dr. R.K. Singh, to this event. Thank you so much, sir.
Thank you, Dr. Bedi. So to celebrate this auspicious day, we have with us our guest of honor, Dr. Sarabpreet Singh Guman, who is the Dean, College of Veterinary Sciences, GADVASU campus in Ludhiana. Dr. Guman has immense experience in teaching, research, and extension, and has been the key member behind various state-level prestigious projects. As an administrator, Dr. Guman has always been on forefront on various prestigious assignments and tasks pertaining to animal health solutions, and is always guiding our strategic research and academic endeavors in the direction of zoonosis, One Health, and food safety. So I invite Dr. Guman to please address the audience on this occasion. Sir, please.
First of all, good morning to worthy Vice Chancellor, Mangalde Veterinary and Animal Science University, Dr. Inderjeet Singh, Chief Guest of the event, Dr. R.K. Singh, Director at IVRI Ijatnagar, guest of officers of the university, heads of departments, my colleagues, and students. So as we know that today on July 6, 2021, we have gathered to celebrate the World Zoonosis Day. As Dr. Simran already told that in 1885, about 146 years back, the biologist Louis Pasteur first ever administered successfully the vaccine against rabies. is a well-known zoonotic disease. So that was the first breakthrough in the history of medical science and the rabies research. While now, actually in the recent times, the importance of this day has become much more than as was thought earlier. To educate the students, faculty, and public, and to raise awareness of disease that can be spread between animals and people. So we all know zoonoses are the diseases common both to animals and humans. Thus, the health of animals and humans is interlinked. So in brief, healthy animals, healthy environment, healthy people. So on World Zoonosis Day 2020, actually last year, a new report titled Preventing the Next Pandemic, Zoonotic Diseases and How to Break the Chain of Transmission was launched. And that has some useful recommendations like raising the awareness of zoonotic diseases, strengthening monitoring and regulation practices associated with zoonotic diseases, including food systems, improving biosecurity and control, identifying key drivers of emerging diseases in animal husbandry, and encouraging proven management and zoonotic disease control measures. The last was the operationalization, the One Health approach in land use and sustainable development planning, implementation and monitoring, among other fields. So keeping that, the World Zoonosis Day 2020, even now today, the significance of World Zoonosis Day is perhaps now greater than ever before. That's why this year theme is let's break the chain of zoonotic transmission. So on this day, it's good that efforts have been made by the team Center of One Health, GADVASU, to organize the International E-Symposium under NAHIP IDP umbrella is highly appreciable. So in fact, ICR launched this NAHIP IDP project described as National Agriculture Higher Education Project, which caters to the institutional development plan for improved learning outcomes, skills, and entrepreneurship as the main objectives for the students as well as faculty. So under this project, we are trying to strengthen the academic programs to produce industry and market-ready professionals, as well as faculty and student development programs for enhancing competence and faculty-student diversity. So overall, even under the new education policy, keeping in view the new education policy, objective is to have a holistic model to raise the standards of the current agriculture education system and make system entrepreneurship oriented to provide more jobs to the students. So I believe that this event would be a good platform to discuss the ongoing issues related to zoonosis and how to control the same in order to avoid future endemics or pandemics. And this is a great opportunity for the undergraduates and postgraduates to learn from the eminent scientists of international repute and explore the opportunities in research. So I'm glad that, as told by Dr. Bedi, about 200 participants, including faculties, UG and PGs across the country and internationally are participating in the event. So I extend best wishes to the organizing team for the event, and we hope that we will be able to break the chain of zoonotic transmission, as is the theme of World Zoonosis Day. Thank you.
Thank you, sir, for your wishes and for your encouraging words. So the next dignitary to honor us with his presence at this virtual event is Dr. Raj Kumar Singh, former director cum vice chancellor of the premier veterinary institute of the country, that is IVRI Nagar. Dr. Singh is a noted scientist and academician of high repute in the field of veterinary sciences and with specialization in veterinary microbiology, biotechnology, molecular epidemiology, diagnostics, vaccinology, and equine sciences. He has special interest in climate changes, environmental conservation, animal and microbial biodiversity conservation, and capacity building. Now, I would like to humbly invite Dr. Raj Kumar Singh to address the audience on this event. Sir, please.
Thank you, Dr. Simran. Greetings on World Zoonosis Day. Congratulations, Dr. Inderjeet Singh and team for organizing this program. I also congratulate the university and Dr. Singh in person for establishing the Center of One Health, which is a very timely action taken by the university. And this had sent a signal because there are only a few Center of One Health functioning, and I am very happy because I have been associated with this university for quite some time, and it has a very dynamic staff, and at the moment you also have a very dynamic vice chancellor. So this is an opportunity to move forward in various directions. Colleagues, we had been in the midst of a lot of unprecedented discrepancies in in the last 1.5 years, and there had been a lot of issues going on. We have a different kind of mixed feelings about the disease which had been, but then it also comes to the people mind, especially the managers and microbiologists, that why it is happening time and again. If you see in 2000, from 2001 to 2020, we had four pandemics. 2002, we had SARS coronavirus, wherein it went to 4 continents and 37 countries. Then we had H1N1 PDM9 pandemic 09. There it was a global event. Then Zika came, 76 countries. And now again, novel corona, it is a global event. So there had been a lot of issues during this period, but then, of course, because the impact was not so serious, therefore our preparedness was very little. I don't say that any country can be prepared for meeting such an exigency when it comes to like unprecedented crisis. But then we need to be prepared in terms of general health principles. And also we must have a surge capacity that within 1, 2, 3, 4 months, we should be able to do. Now, the country has demonstrated about the diagnostic capacity. Many of the non-ICMR institutions, universities, including veterinary universities, all of you have been involved. So we have the capacity, it needs to be reorganized. Now, coming to the increased number of public health events, we see, besides pandemics, we did have Ebola during this period. We also had MERS. Ebola and MERS, they are still continuing. And as you are also aware that H1N1 pandemic, it is not really gone. It has become a seasonal, especially seasonal case, especially in US. So why these things they happen? Because many of the epidemics and pandemics, they will go after a particular cycle. Some may go after 1 year, some may go after 2 years, and some may go after 2, 3, 4 years, like Spanish flu took about 3 years. So same thing we expect for corona also, that it will continue at least up to 3 years till it is gone. But then also there is a fear that yes, we may have uh, some endemic cases as well. Now, because this is all happening because majority of people who are today participating in this, they must be having a lot of background because of their degree, that the drivers of emerging diseases, and also they are epidemiologists, they use term determinants. So because there is increase in the drivers or determinants of the emerging infectious diseases, and it is because of the basically developmental activities, human developmental activities. What we are seeing today, as we move towards the path of the development, we are also strengthening the various drivers which are responsible. And land use change is one of the drivers which is responsible for more than 30% of the diseases. And if you see all of our activities, they somehow, somewhere, they involve land use change. And this is one of the major driver. Now here, I would like to emphasize two things because time is pretty short. You have to go a long day. Number one is that we should be prepared for surveillance. We are well aware, but then we need to plan it as a national activity. But till then, as part of the GADVASU, the Punjab state, and you also have a network on Zoonoses. So there we can have such kind of a network where we can get the data collectively. And then later on, there will be mechanism for sharing because now One Health platforms in India, ICMR has already done. DADH is also launching. So many national One Health programs are coming up, and they will all desire data to be shared. So get on collating the data, and as much as possible, it should be digitized so that when someone approaches us, we should be ready. Second part, which is very important, I would like to say here, to colleagues at the GADVASU, because I consider GADVASU as one of the not one, but the best veterinary university. Congratulations to you, because I have been interacting at different levels with scientists, vice chancellors, outsiders, and the activities of GADVASU, that you have really proved to be the best university. Here, what I would urge that while the routine activities related to One Health and Zoonoses you are performing, awareness programs, etc. And also congratulations because you have started a PG diploma course in this, a diploma course in One Health. Now there are many universities outside which have started giving One Health PG degree. I don't say here because then it would be issue that then the students, they are roaming here and there, they won't get any job. So, but then One Health has to be given priority in practice. So I would urge the university colleagues that if we can take a cluster of maybe 2-3 villages, not very large area, but if we can take a cluster of 2-3 villages and demonstrate a One Health application in practical. So get all the animals vaccinated, go for surveillance of all the data as per the epidemiological surveillance planning, and then say that this is one village or 2 village or 3 village cluster wherein GADVASU has demonstrated that how One Health can be practically implemented. I think you would be the first model. And why I am telling this today, because I have confidence in your Honorable Vice Chancellor, I have confidence in the university, that if you take up a small cluster and start working on it, where within an year or two, you can really prove that how One Health is effective. So it will involve all the activities, creating awareness about the veterinarians, about the general public, about the farmers, then vaccinating, diagnosis, surveillance, and you can very well do it. Maybe as a majority of veterinary college, we used to have some adapted villages. We can take them, but then this has to be from the soul of our heart. And how to make a plan here, again, you can see there is One Health codex. They say, because people say that it's very difficult to get people on the platform, all medical, veterinary, environmental sciences. But now that platform provides an opportunity, and it is a flexible platform. They say that if you are, say, in veterinary sector, you are conducting surveillance, go ahead, you conduct your own surveillance program, no issues. But then you can share the data at the time of it. Second point is that you can, while you are continuing your surveillance, so at some specific point, say some anthrax outbreak has taken place, humans also got infected, take medical people along and go for a joint surveillance program for that particular event, for a particular public health event. Otherwise, routine surveillance you continue. Third is that from the beginning, we can chart out an integrated surveillance plan involving medical, veterinary, and environmental people. So that is a very flexible, and I think that is a guideline that following these guidelines we can prepare, because a universal One Health program is not never going to be a reality. So it has to be context specific, it has to be location specific, it has to be region specific, and then we can chart out. So again, Dr. Inderjit Singh, I don't know, just think of it is my wish that university can take lead in by taking a cluster for implementation of One Health. I am happy that large number of students, they are participating in the program, and I think you're doing great. I congratulate once again, and my best wishes to all of you for your future endeavors. Thank you and all the best.
Thank you so much, sir. We are indeed overwhelmed to hear you, and I must appreciate your valuable advice and guidance towards the One Health approach. And we are really thankful to you for taking time out of your busy schedule and gracing this occasion. Thank you so much, sir.
Dr. Inderjit, if you permit, I have 9:30 to 10 another program with Virology Society. I'll be missing your lecture, but then I have to join there somehow.
Welcome, sir. No issue.
Thank you so much. Thank you so much. Thank you.
Thank
Thank
you,
you,
sir.
sir.
Thank you, everyone.
So moving ahead, we have amongst us a great administrator, an astute scientist, and a teacher par excellence. The chief guest of today's function, Dr. Inderjit Singh, Vice Chancellor, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana. Dr. Singh has served in different prestigious capacities and is a renowned figure in the area of buffalo breeds improvement pan-India. Dr. Singh has always emphasized and stressed upon strengthening of One Health framework and intersectoral capacity building approaches. And taking a leap forward in this direction, he established the Center for One Health in GADVASU under his acumenship. With these words, I would now like to invite and request Dr. Inderjeet Singh to address the audience on this occasion. Sir, please.
Thank you, Dr. Simranpreet Kaur, and thank you, Dr. Jaspir Bedi, for organizing this International E-Symposium and lining up eminent speakers from India and abroad, so that our colleagues in the not only in the university but across the country, they get acquainted with their knowledge and their talk on this important aspect of World Zoonosis Day. And as we all know that livestock are an integral part of our system, whether we are in cities, urban areas, or we are in rural areas. In rural areas, it is the livestock which is the means of livelihood for many large number of farmers, and there are pet animals which are companion for our many people in urban areas. So we have been living with them for ages, and several diseases have been transmitting between the livestock and humans. And in the recent years, as Professor Singh, he is very well known in this aspect, who knows better than him, that it is the population pressure, the invading of their territory, the change in land use, that has pushed the animal population, particularly the wild animal population, to the brink. And they find it difficult, and they are venturing out into the populated areas. And that's also posing dangers not only to humans, but also to the ecology of the wildlife as well as the wildlife population themselves. This university established the Center for One Health this year only, and with the objective that we are already providing services for COVID testing. Our COVID testing lab has already done 3.5 lakh tests for human COVID, and we are also dealing with many diseases which are zoonotic in nature, not only bacterial or viral diseases, even parasitic diseases which are zoonotic in nature. And during our interaction with the medical fraternity, we found that many of these diseases are not much into the mind of the clinicians when they are interacting or when they are getting this type of cases reported in their clinics. And just once you give them an idea that it could be something like that, then they definitely look into that, and many times it comes out to be true that they have been dealing with something which is zoonotic in nature, which has come from the animal kingdom. For that matter, I think there is cross-learning between both medical sciences as well as veterinary sciences, not only in the pathogenesis and transmission of the diseases, but also for preventing of these diseases. Maybe vaccination, if we take, in vaccination also, the veterinary scientists, including the CEO of Pfizer, he is a veterinarian, and they have come out with the vaccines. Many other scientists of veterinary background, they have been involved in the development of vaccines against COVID-19 in the last year. So the integration between the two sciences, two streams of health, whether it is animal health or livestock health or human health, it is very important. And that is going to be a win-win situation for both of us, for veterinarians as well as medicos. And with that in mind, we started this One Health Center. And the PG diploma that Professor Singh mentioned about that, we were able to attract scientists or the graduates, not only from the veterinary sciences, but also from dental surgeons and from agriculture also. So that is the beauty of One Health, that we all fight. And just now I was going through a ICR circular that on 100 years of ICR or 75th year of the Indian independence, let all the ICR institutes, health universities, veterinary universities, they should plant trees. And plantation of these trees is going to create or to rectify the damage that the human development has done to the environment. And this is an important activity which the ICR has also taken cognizance in the times of this crisis. When we also saw that when it was at the peak in the month of March and April and May, then there was a lot of hue and cry for the availability or non-availability of oxygen. And just yesterday I was reading in the newspaper that some people in Ludhiana, in the complex where the district admission sits there, some people in Sangrur, they have cut down decades old trees. That I think is something which needs to be checked, because we have to honor the life of not only livestock but also the trees. And we have to learn from many these what I call the revolutionaries like Mr. Bahuguna, who started the Chipko movement to protect the trees, like Guru Jambeshwar from Bishnoi Samaj. He allowed that he rather said that no trees should be cut, because he was in the Rajasthan area where the trees are already very rare. So he protected the trees. And we have many stories of this type of movements that have been taken place within India. But in spite of that, if this thing happens in the cities, and where we consider that we are more educated, we are more civilized as compared to our rural brothers, I think it is we have to think again. And it is only with the health of environment that the health of animals and the health of humans can be ensured. So I would have liked to inform Dr. R.K. Singh that as he suggested that we should have a cluster where all the One Health awareness and activities are taken up. We have aligned up with the Department of Health and Family Welfare of Punjab, and across the state a awareness drive is being started. I think is it from tomorrow or day after, following the colloquium tomorrow. Thereafter, there will be a 15 days drive where the brucellosis awareness and testing will be undertaken, because this is a important disease which not only affects the economics, but it badly affects the economics of livestock farmers as well as health and fertility of the humans as well. So I wish all the best to the symposium as well as the colloquium that will be tomorrow. Thank you very much.
Thank you so much, sir, for your kind words and good wishes. You have always been guiding us in all our scientific endeavors. Thank you, sir, for gracing this occasion. So moving ahead, to mark this special day, the faculty of Center for One Health has compiled a beautiful handbook on zoonoses to create awareness among the masses. So with the consent of our worthy Vice Chancellor, Dr. Inderjeet Singh, we would like to release the booklet on this special occasion. So this is the booklet which has been compiled by the Center for One Health. The booklet is on zoonoses, and the main aim of the booklet is to create awareness among the common masses on the important zoonotic diseases. So thank you, sir. So along with this booklet, Dr. J.S. Bedi has one other important information to share with the audience towards the in the direction of One Health. So Dr. Bedi, you can share it.
Yeah, thank you, Dr. Simmons. As VC sir has already told that the initiative which we are taking is for 15 days And with the visionary guidance of Dr. Inderjeet Singh, Vice Chancellor, GADVASU, and with the kind support from Dr. SPS Guman, Dean College of Veterinary Sciences, we took this initiative to write letters to the Department of Health, Government of Punjab, to observe the World Zoonosis Day on 6th of July. And yesterday we received a notification from the Department of Health and Family Welfare, Government of Punjab, that they have directed all the civil surgeons in Punjab to observe 6th July as a World Zoonosis Day and create awareness on the prevention and control of the endemic zoonotic diseases in Punjab or in the northern region of the India. So this is a great, I think, initiative by the Department of Health and Family Welfare. And sir, I would like to share that the team of the Civil Surgeon Ludhiana, they will visit GADVASU. They will interact with the dog owners. They will talk with the aspects of the zoonotic diseases, and they would also like to visit Center for One Health so that we can go for the future work with the Department of Health and Family Welfare, Government of Punjab. Thank you, sir.
Thank you, Dr. Bedi. So I would now like to invite the organizing secretary of this symposium, Dr. Rajneesh Sharma, from the Center for One Health, to extend a vote of thanks. Dr. Sharma, please.
Thank you very much, Dr. Simran. And once again, good morning to all of you. On behalf of organizing team, I would like to, first of all, thank Dr. Inderjeet Singh, worthy Vice Chancellor GADVASU, as well as chief guest of this morning, who always support us to organize such events, which are not only beneficial to the faculty, to the researcher, to the scientists, but also to the students of undergrad as well as post-grad programs. So thank you very much, sir, for sparing your valuable time and coming today morning and joining us. Thank you very much once again. We are also thankful to Dr. R.K. Singh, who is a guest of honor for today's event. And he has shared his advice with us, with experiences with us. We are very thankful to you, sir, also. Thank you very much. And we are also thankful to Dr. SPS Guman, who is Dean College of Veterinary Science, as well as PI of IDP and HEP, and who always encourages us to do such kinds of events like workshops, trainings, as well as small events like symposium, as well as conferences, and without whom these events, they are not possible. So thank you very much, Dr. Guman, once again. And we are also thankful to all the officers of the university, all our colleagues, students of the university, as well as the participants who have joined us today to make this event a big success. Thank you very much. And I would also like to thank all the speakers, national as well as international, who joined us today and spared their time and accepted our invitation to speak today, because and at some places of the world, it's a night and early in the morning when they are going to speak. And the two of them have already joined us, Dr. Emily Jenkins as well as Dr. YPS Malik. I thank you once again on behalf of our team. At the end, last but not the least, I would also like to thank the 4 pillars of our departments. So firstly, our teachers, our researchers, who are always in helping mode to help us to organize such kind of events. Secondly, our non-teaching staff. And thirdly, our research fellows. And fourth, and above all, our students, our postgrad students, who always help us in these kind of events. So at the end, once again, thank you very much, all of you. And I hope all the participants will enjoy this event and actively participate in this event. So thank you very much, and happy World Zoonosis Day. Thank you.
Thank you.
Thank you, sir.
So Now we will start our first keynote address, which is by Dr. Emily Jenkins. And good morning. What should I say? So could you please unmute your mic?
Good morning.
Good morning. So we are having Dr. Emily Jenkins with us. And I would ask my colleague to please share the introductory slide. So Dr. Emily Jenkins, she is a professor of veterinary microbiology at the Western College of Veterinary Medicine, University of Saskatchewan, Canada. And she is acting head of this department currently. Dr. Emily Jenkins, she has been involved in parasitic zoonoses for more than 2 decades. And she has published a number of papers on zoonoses as well as One Health aspect. We are very fortunate that she has accepted our invitation. However, it is, I think it's about 10 o'clock in Saskatoon right now. We thank you, Dr. Jenkins, once again, and over to you. Thank you very much.
Thank you so much for the invitation. And I want to commend you very much on founding a Center for One Health. That's something our university, my university, still has not done. So it's very visionary and very appropriate to the times. So my talk will focus on wildlife as sentinels for One Health, and forgive the pun a little bit, but "We Stand on Guard for Thee" is a line from the Canadian national anthem. And the idea that maybe wildlife can be sentinels for threats to our health is certainly appealing. And I'd also like to thank very much my 2 PhD candidates, Émilie Bouchard and Adrienne Hernandez Ortiz, whose data I will be drawing on for this presentation. So what makes a good sentinel for One Health? Animal sentinels. And the first criteria is quite simple. They should be highly exposed. They should show some kind of response, be it clinical, overt disease, or perhaps more ideally, a detectable serological response. Samples should be accessible. It should be a population that we can access. The animal population that we use as a sentinel needs to be relevant to our population of interest, which is, of course, very often us. And so they should share our environments or share our risks of transmission. Ideally, wildlife and animal sentinels should not be a source of transmission to people, especially if they're harvested for food. And finally, the wildlife population itself should not be of conservation concern, particularly if the pathogen or disease causes effects on survival or reproduction. And it's always very important to ask sentinel for what? And in this case, I'm going to give you 2 examples, one where we're using wildlife as a sentinel of the risk of human exposure, as well as risks from a changing environment. So the parasite I'm going to talk about, and I noticed there's another talk on this later on from Pika from Finland. So you will be getting a lot of Toxoplasma gondii, I'm afraid, but it's a very cool parasite. And it's the kind of parasite that makes headlines. And in fact, this week it is in the news because Elon Musk tweeted about Toxoplasma. I don't think he's necessarily an expert in the subject matter, but there's a lot of interest in this parasite because it does cause changes in behavior of its hosts, and because it affects well over 100 vertebrate species as intermediate hosts. So the life cycle of Toxoplasma, it's a tissue cyst forming coccidian parasite. It is transmitted through a wide range of routes, including through food, water, the environment, directly from cats, as well as vertically for mammals that are infected for the first time in pregnancy. They can transmit the parasite to the unborn fetus. Cats are the ultimate source. They're the only host in which sexual reproduction occurs, and oocysts are passed out into the environment through their feces. And it's an incredibly successful parasite. We estimate that at least 1/3 of the world population has been exposed, is seropositive for Toxoplasma gondii. Clearly, it doesn't cause severe disease in most people who are immunocompetent, and in And really the main concern is its transmission in utero. So in Canada, we know that the seroprevalence for Toxoplasma is not uniform, particularly in the north. And we think that the North American average for Toxoplasma is around 10 to 15% of the population in the US and southern Canada. But in the north, we know that there are pockets of very high prevalence. So in Nunavik, which is in northern Quebec, over here in the east of Canada, we see communities that have as high prevalence as 88% and overall seroprevalence is 60%. It's about 32% in Nunavut, which is still probably at least double what we would expect. And then it's more what we'd expect in other areas across the Canadian North. So we had a question of whether or not we could use foxes as a sentinel for human exposure to Toxoplasma gondii. Now, we study foxes for other plasma, but they can serve very well, we think, as sentinels for human exposure. And we specifically wanted to know if there was hotspots for seroprevalence in foxes as there were in the peoples in the Arctic, and also about how foxes and therefore people could be exposed. So this is work by my student, Emilie Bouchard. She looked at almost 700 foxes. She mainly looked at serological response, so antibodies in heart fluid, but she also correlated that with detection of DNA of the parasite in brain and heart. So we know that the seroprevalence was slightly higher, as one might expect, because it reflects lifetime exposure. If we could mute. There we go. So her results just coming in because she's just finished her PhD. And she found very interesting overlap between areas where we have high seroprevalence for Toxoplasma in foxes were very similar to the areas where we had high seroprevalence in people. So from the Inuit Health Survey, the indigenous peoples of our Arctic, we know that in the West, it's very low seroprevalence, 10% in foxes and 8% in people. And then as we get into Nunavik, which is that hotspot for Toxoplasma in Canada, we found that particularly in this little area called Hudson's Bay, there was 60% seroprevalence in foxes and people, and a lower seroprevalence in the Ungava Bay region of this province. Elsewhere, the correlation wasn't as tight, wasn't as nice. 30% of foxes were positive in Labrador, 11% in people. And in the James Bay region in northern Quebec, 30% of foxes positive, only 9% of people. And so we hypothesize here that the diet of the local people in those regions probably means they're eating their meat more cooked, and they're probably not being exposed through the similar ways that we think foxes are exposed. So how are foxes exposed? So part of Emilie's work involved taking fox hair and looking at stable isotope signatures, which told us what the foxes were eating. And she found that based on the isotope signatures, that foxes that ate a lot of marine prey had a high nitrogen carbon ratio up in the right-hand corner here. Geese were in the middle, and at the bottom were small rodents, lemmings, voles, etc. And sure enough, when she looked at the likelihood that a fox was seropositive for Toxoplasma, the foxes with the highest seroprevalence were those that ate quite a lot of marine prey and those that ate quite a lot of geese, suggesting that they were primary sources of exposure through carnivory of foxes. When we look at these human seroprevalence and human data from this region, which has just also come out very recently in 2021, we can see that the higher human seroprevalence was correlated with eating fish, with preparing wild birds, and with eating walrus, seal, and shellfish, mostly raw, because that is the cultural practice in that region. And so there does seem to be a nice correlation, not only in hotspots for prevalence in foxes, but also in probable similar routes of exposure through consumption of tissue cysts in raw or undercooked meat, particularly from marine systems and from geese. So the second scenario I wanted to share with you was wildlife as a sentinel for changes in risk due to climate change. And we know that the Canadian Arctic is warming at 3 times the global rate, so much higher than the rest of the world, and indeed much higher than the rest of Canada. And I'm going to take you to a region just west of this big bay called Hudson's Bay, called creatively Western Hudson's Bay, which is one of our hotter spots for climate change. And so we had another simple question, always starts off simple anyway, is are polar bears increasingly being exposed to pathogens? And polar bears are, of course, the poster wildlife species for climate change. They have been listed as directly threatened by climate change. And in this region of Western Hudson Bay, the habitat of the polar bear is supposed to be on the sea ice. And as you can see here from this pictorial representation, in the 1980s, the sea ice was this blue region. In the 1990s, this green region. And in 2015, 2017, limited to this small gray region. So that means with this diminishing sea ice platform, the polar bears are pushed more and more onto land, which is marginal habitat for them. The other thing that's happening is that overall we're seeing warmer temperatures, and overall we're seeing more summer precipitation, i.e. more rain, washing into the region. So we had a unique opportunity to look at archived sera that had been collected as part of wildlife studies by biologists over the last 50, 60 years. We used targeted samples from adult bears because we know adults have a higher risk of lifetime exposure, and we also balanced the gender ratios since we know that there can be differences in male-female exposure to toxoplasma. And we used a commercial indirect ELISA from ID Screen or ID Vet that is mammalian specific. So it targets IgG of all mammalian species. And overall, we found that the polar bears were quite exposed, that they had a fairly high seroprevalence of 56%. And furthermore, there was a significant increase over the 3 study periods from 43% in 1980s up to 70% in 2015-17. We also found that seroprevalence was higher in those summers that had more precipitation. So those wetter summers, we theorize, enabled more toxoplasma transmission because it mobilized oocysts that were produced by wild or domestic cats in the region, spreading them into fresh and marine water where they probably infected prey species of polar bears, and therefore higher seroprevalence in our bears. And this is fairly consistent with what's being seen elsewhere in the world. What the power of our study has is that we used the same methods. We had archived sera over 3 time periods, whereas other studies were more snapshots of a period in time, and then they went back and did another study 10 years later and said, oh, look, the prevalence has doubled. As is true for Svalbard in northern Norway, the prevalence had doubled between these 2 study periods. And in the southern Beaufort Sea in Alaska in the US, the prevalence, although lower than what we're seeing in our bears, still doubled over a period of 7 years or so. There are also correlations or reasons why they think climate change is causing increased transmission of toxoplasma, largely due to higher survival of oocysts in ocean currents, higher concentrations in filter feeders in those marine systems, and exposure through the prey of polar bears. That latter study, the Alaska study, also showed that polar bears using land were 7 times more likely to be seropositive than those that stayed on the sea ice. So it's clearly a terrestrial parasite. So when you think of polar bear habitat, you probably think of the bottom picture, ice, and that's what it really should be. And increasingly what we're seeing is they're living more and more on the terrestrial lowlands for which they're not really designed. Also along the theme of this conference with changing land use, this is increasingly polar bear habitat, and we really don't want bears coming into town. They pose a great risk to people, and it's also a great risk to the bears. So generally what happens is the bears are tranquilized, taken to the bear jail, and then they get a free helicopter ride as far away from town as they can go. But we don't want bears coming into town, eating at the dump, getting exposed to parasites through landfills and human refuse. Well, a cat refuse in this case. So we do know that that polar bears are essentially marine mammals. They really shouldn't be on land, and that the diet that they can get on land is not suitable. And our study also shows that they are more increasingly exposed to terrestrial pathogens. So going back to that framework for what makes a good One Health Sentinel, in this case for toxoplasma in the Canadian Arctic, both foxes and polar bears were highly exposed, so both would be good sentinels on that note. They both showed a detectable serological response. Where things start to differ is the next couple criteria. So for foxes, they're actually routinely trapped for fur, not as much as they used to be, but they still are. So that's an easy way to get samples because a trapper can just give us the carcasses once they have the skin. So it's very easy to get samples from foxes. Polar bears, not so easy. Generally, it requires helicopters and it's not always very safe. We've lost 2 polar bear biologists in the last decade or so, one just this year, due to adverse weather conditions. So it's not very simple to get samples from polar bears, which generally involves wildlife immobilization from a helicopter. Foxes live close to people. They come right into town. And so they are pretty representative of the peoples of the Arctic, whereas polar bears, despite the picture I just showed you, generally aren't coming into town or shouldn't be coming into town. Foxes are not consumed, so they're not a direct source of foodborne transmission to people of the parasite, whereas polar bears are actually consumed by indigenous harvesters. So they are potentially a source of transmission. And then the final one is the big one. In Canada, foxes are doing fine. They have good populations. They're not of conservation concerns. So we can check the box for them being good sentinels of a new threat or changes in Toxoplasma exposure. Polar bears, on the other hand, are of high conservation concern and a high threat from climate change. So really, we shouldn't be using polar bears as a sentinel because it's really not ideal for them. That doesn't mean we shouldn't study Toxoplasma in polar bears because we don't know anything about whether or not it affects survival, if it affects reproduction. We do know it causes abortion in many mammals, and that's the last thing a species under threat needs. So very worth studying in and of their own right, but using them as sentinels for our health and for one health, maybe not. So this paper is coming out any day now in Global Change Biology, and a shout out to Adrienne, who did the polar bear testing. And this was my research group a few years ago now, and you might see a familiar face there. This is what my research group looks like nowadays, unfortunately, due to challenges of COVID. So I'd be happy to answer any questions, and if there's time in the chat or via the audio platform.
Thank you very much, Dr. Jenkins, for such a wonderful talk. We do have a few questions which I have received. So the first one is about how it was related to the wetter summers with the high prevalence of Toxoplasma. So that is the first question for you. And then the second question is about the sentinels. So question is, what is your take on the livestock scenario? How could we pick up a sentinel in that scenario?
Okay, good questions. The first one about the high summer precipitation. We do know that oocysts survive very well in water. They can survive months to years in particularly marine systems. So oocysts produced by cats could, with increased precipitation, increased runoff into marine systems, as well as increased transportation through freshwater, could be one route that the parasite is reaching, if not polar bears directly, certainly the prey that they feed on. So that's one theory. But again, this is all correlation, not causation. So that would require more mechanistic investigation. The second question about livestock sentinels, that's an interesting one. I think livestock are challenging because most of the time domestic livestock are food animals. And so the last thing you want to do is intentionally expose food animals to a foodborne hazard. Certainly, in a way, sheep and small ruminants are a good signal for Toxoplasma because they are sensitive to it and it can cause abortion storms. So that would be obviously an indication that there was transmission occurring. I would say that would be not a great outcome for the producer as well as for public health. So it's tougher to use domestic livestock as deliberate sentinels, but certainly if you think about a good monitoring program, that's exactly what you're doing, is you're testing to make sure there aren't threats to human health. And if there are, that you can find critical control points to intervene.
Thank you very much, Dr. Jenkins. And one last question is about, can we take rodents as the sentinels in the scenario where there is wildlife as well as livestock interface?
Yeah, that's actually a good question. And that would be, in fact, an excellent way is to trap peri-domestic rodents and monitor them for toxoplasma, because not only are they a source of infection for cats, but they would also be generally not a risk for human. They would be a dead end from a human transmission perspective. So yeah, testing rodents is potentially useful. The reason that we test high-level carnivores as sentinels for toxoplasma, and I apologize for not mentioning this, but is that because the parasite does tend to bioaccumulate. And so the higher up the trophic food chain you go, the higher the prevalence. So unfortunately, if you do test herbivores like rodents and ungulates, you tend to get fairly low prevalence, like 10% if you're lucky. Whereas if you test carnivores, you're higher up the food chain, you're much higher to get a louder signal. You get a higher seroprevalence. There's just more to detect, and it's much more fun to test animals that are going to be positive than testing thousands and thousands of rodents to detect small changes in prevalence over time.
Thank you very much, Dr. Jenkins, once again for accepting our invitation and for such a wonderful talk on One Health and zoonosis. Thank you very much, and thanks for your time. Thank you.
It's my pleasure. I hope someday I can come in person again.
Oh, yeah, let's go for the best.
Thank you, Dr. Emily.
So now we are having our next speaker, Dr. Joe Rubin, with us, and Dr. Rubin is a veterinarian, and he is a microbiologist at the University of Saskatchewan. His area of expertise includes antimicrobial resistance, and he has been studying this in bacteria from people, animals, and food. Currently, he is acting as associate professor and graduate chair in the Department of Veterinary Microbiology. We are very thankful to Dr. Joe for accepting this invitation and sparing your time to speak on this event. So over to you, Dr. Rubin. Thank you.
Well, thank you very much, Dr. Sharma. And sorry, I'm just getting my screen shared there. Hopefully that's working.
It's visible, yes. Perfect. Yes.
Okay. So yeah, thank you very much for, again, the invitation to speak. And it's a pleasure to be able to go after Emily. And I have to warn you, I have a bit of a different approach to my talk than she had. Mine is not going to be so much focused on data and my own research, but it's more, I kind of want to just stimulate some new thoughts and get people thinking about foodborne infections from maybe a bit of a different perspective. When I was asked to give this presentation, I really struggled with what I wanted to talk about. I didn't want it to be just another talk about E. coli and salmonella from, chicken meat and eggs and things like that. I think that's something that a lot of us are already familiar with. So I thought about kind of where we are in the pandemic, what some of the trends are and how people are preparing and consuming foods, and whether there might be some yet unidentified hazards associated with that, or something interesting going on. I took a look through what the current food recalls are by the Canadian Food Inspection Agency to see if there was any sort of interesting things going on there as well. And so I'm going to share sort of two, maybe not even stories, just sort of two ideas under this theme of sort of unrecognized or maybe underappreciated types of foodborne infections. So first, I guess I just want to say, when we're talking about foodborne illness, at least from my perspective as a bacteriologist, we're really talking about two things. So one is the presence of bacterial toxins, bacterial toxins in food. So this is sort of the classical Staph aureus food poisoning. The other is the presence of actual bacteria. So you get a foodborne infection. So E. coli's, salmonellas, campylobacters, things like that, that infect the host and cause disease that way. I think it's important to consider what the food chain sort of looks like, how our food gets to us and what we do with it once it's received. So, we have our animals, we have our plant-based food products that are being produced and slaughtered and harvested, all over our respective countries. Those foods are then brought to markets or grocery stores. They can also be sort of processed in a secondary facility. Maybe they're transformed into some sort of prepared, ready to eat food. Maybe in the case of meat, those animals are, the carcasses are broken down and dressed and sold as, particular components. This is where we buy them and bring them into our kitchens, where we have, the responsibility of preparing and handling these foods safely. At the groceries or markets, there can also be some sort of secondary food processing. So it's really common, certainly here, for grocery stores to prepare their own ready-to-eat meals. So they're taking these raw ingredients, making, a dinner that you can just take home and eat, sort of like a restaurant. And what I think is really important to consider is that foodborne illness can occur when we have a breakdown in sort of hygiene or good food practices at any stage of this process. So it can certainly be on farm or at food processors, where I think we have traditionally much more focus on food safety and where there's really a lot of regulatory oversight. This is where we can do something about bad practices. But what we do in our own kitchens is also very, very important. And actually, we're probably responsible for, maybe the majority of our own foodborne infections. This is just some data from Canada. So from the Public Health Agency of Canada. Here what you can see is the annual frequency of human campylobacteriosis. So between 2007 and 2017, there were roughly 10,000 laboratory confirmed cases a year. Now these are laboratory confirmed cases, so it's really just the tip of the iceberg. There's probably 10 to 100 times as many people that are infected. It's just that a relatively small proportion of them go to their doctor, have the sample taken, have that sample shipped to the lab, etc. So many, many, many people are infected. These things are very, very common. Similar story with salmonella. Between, you can see, maybe 2000 and 2020, we have between 6,000 to 8,000 cases a year. So again, really reflecting these breakdowns in food hygiene in our own kitchens. People are cross-contaminating raw meat products or animal products with foods that are perhaps not cooked or are eaten raw, really giving a great opportunity for transmission. When we're talking about food safety in the kitchen, certainly we're concerned with cross-contamination. That's very, very important. But cooking our foods to an adequate temperature is also critically important. So hot foods, we really want to cook them to an internal temperature of at least 74 degrees Celsius and keep those foods hot. We want to keep them above 60 degrees. Once that delicious meal comes out of your oven, you don't want to have it sitting on your kitchen table for hours and hours and hours, cooling off and getting to this really nice, warm, hospitable temperature where microbes can grow and then make you sick when you eat it. On the flip side, cold foods really need to be kept cold. So ideally 0 to 4 degrees Celsius. I'm reiterating all of this and talking about all of this because what we need to consider from a food safety perspective is what happens in between these ranges. And this is really what we can sort of refer to as the danger zone. This is where our bacteria are going to be multiplying. This is where they're going to be producing toxins. And this is where food quality really suffers. And where I think this is really, really interesting and entirely unstudied, like I said, this is just sort of my thoughts on some current food trends that I'm seeing. where I think this is really interesting is in the current really pandemic trend of home fermentation. These are some of my own fermentations. So I like to make my own homemade yogurt, homemade ciders. So this is like apple-based ciders with other fruit juice. Certainly breads, cheeses, all sorts of foods are fermented. And when we ferment products, really what we're doing is deliberately putting them into that, kind of danger zone. We're wanting our microbes to grow, and we're wanting them to transform the ingredients in healthy and delicious and culturally important ways. And that's a really, really good thing. But I mentioned earlier that, people in their kitchens aren't very good at food safety when it comes to, sort of easier to deal with foods like meat and eggs. What are they doing when it comes to fermented foods? Are they handling these things in a safe way and preventing, possibly contamination of their fermented product with some potentially pathogenic bacteria that can then go on to grow and grow and grow? So sourdough bread was a huge trend at the beginning of the pandemic here in Canada. And you can see these are just some screen grabs from YouTube. There are probably thousands of instructional videos out there telling people how to make their sourdough starter. For any of you who aren't familiar with sourdough bread, it's essentially utilizing wild yeasts and wild bacteria for the bread's leavening. You're not using a store-bought packaged yeast. Now, from a food safety perspective, I don't think sourdough, there's really much of a risk there at all. The benefit of bread is it has this protective step of cooking. So it goes into your oven, you kill off everything that's there, and you're left with a delicious loaf, or hopefully delicious loaf. Where I think it gets really interesting is when we talk about the fermentation of products that aren't then cooked. And one that I've sort of been thinking about for a long time, and I'm kind of curious to do maybe some studies on, is something called kombucha. I don't know if this is popular in Punjab, but it's certainly something that's become very, very popular in Canada and in many countries as sort of this fermented health drink. And what kombucha is it's like a sweetened tea product that's fermented with a bacterial and yeast community. You can see here, I hope my pointer is showing up, but in this top left image, somebody has this large sort of grayish brown slimy blob. This is the microbial community that does the fermentation. And you can see in these, again, screen grabs from YouTube videos, people are fermenting their kombucha in these large glass jars. They have their microbial community. It's called a SCOBY or a mother or a mushroom, depending on sort of your favorite lingo related to kombucha. And that's reused again and again and again to produce new batches of kombucha. So, like I said, it's a fermented tea beverage. Classically, this is a product used in Japan. That's where the name actually comes from. It's made using a huge variety of products. There is no standard ingredient list for kombucha. So many different types of teas are used, juices, the liquid before the fermentation starts is oftentimes sweetened to 5 to 10% sugar as a starter. So you have this really nutritive, rich environment for the microbes to grow and replicate and do all of the things that we want them to do. It's a complex and probably quite ill-defined microbial community. Certainly you can purchase starter cultures online, although who really knows what you're getting there, but people oftentimes also start their own. The name, so the SCOBY, it's an acronym referring to the symbiotic culture of bacteria and yeast. So there's really quite a number of things growing in there. And the fermentation period for kombucha is 5 to 10 days. So there's a long period of time where we're allowing these microbes to grow and replicate. And, you know, there's certainly an opportunity for unwanted things to also be growing in there. The final product is ideally something that's acidic. So we have lactic acid and acetic acid producing bacteria that are lowering the pH. hopefully in a way that can inhibit the growth of pathogenic organisms and other microbes that we don't want. We also get a little bit of alcohol production. We do have yeast there. And so this is something that you don't want to drink, you know, maybe right after exercising or while you're driving. We don't want people to be getting drunk while using this. The final composition varies incredibly widely. So depending on what your inputs are, depending on what the microbial community is, you know, the chemical composition of the kombucha is going to vary quite widely. What I think is really interesting about this from a food safety perspective is that there can be other microbes that become incorporated into the SCOBY and can sort of become a part of that microbial community, including potential pathogens. And because kombucha is oftentimes made at home, this is a food safety hazard or potential hazard that's really not regulated. You know, people can cook and do what they want in their kitchens, and they may not be aware of some of the risks that they're exposing themselves to. So this is a paper that's really hot off the press. And what these researchers did is they investigated, you know, how well salmonella and Shiga toxin producing E. coli could grow and replicate in different types of kombucha, so produced using different commercial kits. And they found that experimentally, if you inoculated that kombucha with salmonellas and S-Tex, some of them are going to survive quite nicely. And it really depends on what the composition of the kombucha is. So there's no there's no sort of reliable or there's no guarantee that the product that somebody's making in their home is going to be acidic enough or going to be inhospitable enough that it's going to inhibit the growth of potential pathogens. Now, there hasn't been a lot of research looking into this, which is why I think it's interesting and I thought it was sort of fun to talk about. But one of the really sort of horrifying case reports I found was actually a case of cutaneous anthrax associated with the kombucha. In this case, they called it a mushroom. This is the SCOBY. This is the microbial blob that does the fermentation. And in this particular case, it really highlighted the need for using good food hygiene practices. So the people making kombucha in this case report were doing so in a really unhygienic, filthy environment. It was done in a plastic container on the ground in close proximity to cattle. And so the authors speculated that that may have been how transmission of anthraces occurred. Now, in this particular case, I should also say that Bacillus anthracis was not actually isolated from the SCOBY, although when they experimentally inoculated it, they found that it actually was a suitable medium, and so it could be growing. Now, after having said all of this, I'm certainly not trying to scare anybody away from drinking kombucha. I think it you know, probably has a lot of health benefits, and it's something that a lot of people see value in, but I think it's an interesting area for food safety research. And I think that the sort of do-it-yourself home fermentation presents some really interesting food safety hazards that we really don't understand. And this is an area that I'm certainly interested in doing more research in, and I'm sure many of the people attending today might be interested as well. The next one that I wanted to just briefly talk about is Listeria monocytogenes. And I wanted to mention it because when I was looking through the food recalls from the Canadian Food Inspection Agency, there were a number of products that I don't generally think of as being associated with Listeria, where there had been recent food recalls. So Listeria is a not common, but certainly not unheard of cause of foodborne infections in North America. Roughly 1,600 cases a year in the US, resulting in 260 deaths. So the case fatality rate, at least among identified cases, is relatively high. Pregnant women and immunocompromised individuals and older adults are much more likely to have severe outcomes. Most commonly, people who have a Listeria infection have no signs at all. Transient asymptomatic carriage in the GI tract is the #1 thing thing that happens to you if you're infected with listeria. Acute symptomatic illness can certainly also occur. Could be a simple flu-like illness. It could be a gastroenteritis. You could have a much more serious disease. It is associated with abortions and very severe illness in newborns. Also sepsis and meningoencephalitis in non-pregnant adults. So definitely something we want to avoid. In Canada, we have somewhere between 1 and 200 cases a year. So it's not common, but it is something that does occur at sort of a low level on an annual basis. If we look at the foods that we most often sort of consider when we're thinking about listeria, things like uncooked meat, raw dairy products, soft cheeses, and processed foods, so deli meats, smoked fish, things like that are sort of our most obvious culprits. These are the things that we tell people who are at increased risk, pregnant women, elderly people, to avoid eating. What was interesting and what I was sort of alluding to earlier is when I looked at some of the recent outbreaks, they were in food products that maybe we don't always think of as being associated with listeria. So this particular set of vegetables, this veggie power blend, was recalled in Canada back in late 2019. Here we have some peanut spread, sweet kale blend, so leafy greens that, you know, certainly may not be cooked, something that people might eat raw. And, you know, there's certainly an increased risk of transmission when we don't have that protective cooking step. And then very recently, there have been a number of recalls associated with enoki mushrooms. So kind of unusual products. And really what I wanted to get across is when you're thinking about foodborne illness, think more broadly than E. coli, Salmonella, and Campylobacter. Don't just think about poultry. Think about different types of foods. Think about how those foods are handled. Think about how they're processed, what we're doing with them in the kitchen, and, you know, try and get a more holistic perspective on foodborne illness, because I think there's a lot that we're probably missing, and there's certainly, you know, ample fascinating avenues of research that we can pursue. So with that, I'm happy to try and take any questions or have some discussion about food safety and foodborne infections.
Thank you very much, Dr. Joe, for your really an encouraging and informative talk. So one of the question is about one of the graph you have showed that the cases of listeria, they were less in 2015, and then they rose up in 2016, and then they again came down in 2017. So what could be the reason for that?
Yeah, that, you know, I have to admit, I don't actually know what the reason for that is. So this is compiled data from the Public Health Agency of Canada, and I don't believe that they, you know, report anything more granular than numbers. There could have been an outbreak that year. It's still within sort of this narrow range of kind of 1 to 200 cases, which when you consider that Canada has 38 million people, 1 to 200 is a pretty tight range. So I'm not sure what happened that year. There may have been something interesting, but I don't know.
Okay, so one more question is, what could be the major source of listeria? So I assume it is what could be the major foodborne source of listeria, right?
Yeah, I mean, you know, recently I'm not aware of there being any, you know, singularly large outbreaks. In years past, we've had, I think about 8 years ago, there was recognition of contamination at a meat processing facility that resulted in a large cluster of listeria outbreaks or listeria infections. I haven't heard anything about that recently. And so what I'm assuming is that we have, you know, a ubiquitous organism. It's present in soil, it's present in animal feces, and we probably have sort of a large number of either small clusters of infections or individual infections. And the types of food are most likely to be those things like soft cheeses, deli meats, smoked fish, smoked meats. Those are the most common ones, although like I showed, we can certainly have it with vegetables, it can be in mushrooms, it can be in prepared food products as well.
One question is from Dr. Sani.
Yes, hello, Joe. My question is sort of comparing practices being followed in developed nations and then developing countries, maybe, you can say India. When we also work on food-related microorganisms, and but then whenever we look for information, we always see information published by developed nations like US and Canada, where we quote reference of all those cases that are recorded there. So what are the reasons you think that we have so many cases reported or recorded in North American countries or developed nation as compared to the country like us?
Yeah, wow. That's a huge question. I have to admit, I don't have a ton of insight into that, but it's kind of the tragedy of the scientific literature that there is such underrepresentation of, countries that aren't Europe and North America and Australia and some parts of Asia. And we have big gaps in our knowledge. So whether that actually reflects differences in the incidence of disease, or whether it's just that the disease isn't reported to the same extent as it would be here, I'm really not sure. Certainly in some areas, I know I had a visiting student in my lab who was in Nigeria, and one of the big problems that he faced, and would certainly be a reason for a gap in knowledge from the state in Nigeria where he comes from, is that they just simply lacked the infrastructure to do the research, and they lacked, really the ability to publish their findings, because they couldn't do the microbiology work to sort of the standard of the literature. Certainly having visited Louisiana a number of times, I know that's certainly not the case where you guys are. You have the facilities to do, these sort of epidemiological investigations in food microbiology. And so I don't know. I guess I would sort of pass that question back to you. And as the Western person, I'll ask for sort of your expert opinion on why you think that might be.
Do you think cooking could be the reason? A difference in the way food is being cooked in developed nations, and how the liking of the people, the way food is cooked, and the way we cook our food here, and then the reporting system, surveillance on foodborne diseases being more rigorously followed in Western nations as compared to a country like us?
Yeah, that's certainly possible. In general, people in Canada have very high expectations of food safety, and the public does not tolerate food of low quality. If somebody goes to a restaurant and they get sick, that's a big problem for that restaurant, and they're gonna have difficulties getting customers. When it comes to reporting and surveillance, we do have a really good network. So when I talked about all those salmonellas that had been cultured from people in those 6,000 to 8,000 cases a year, those all go to our national reference lab, and they're fingerprinted. Their DNA fingerprinting techniques are used to compare them to each other so that outbreaks can be identified. And when we look at what food recalls are sometimes based on from our national regulators, we might have 5 or 6 cases across the entire country But because the surveillance system is so strong, those outbreaks can be identified even when they're, a couple people in British Columbia and maybe a couple in Ontario. Those outbreaks can be recognized and then dealt with. So I think it's probably a combination of all of those things. And certainly there are, big differences in preferences for how food is prepared and how people like to like to consume different things, whether it's raw or cooked or whatever.
All right, thank you, Joe.
So thank you very much, Dr. Joe, for your wonderful talk. Thank you very much, and looking forward for your talks in the future. Thank you very much. Thanks again, and
Thank
good
you.
night.
All right, good night.
Okay, so now we are having with us our third keynote speaker, Dr. Eleftherios Melidis. He holds a degree in veterinary medicine, and he is doing research on development and application of Bayesian methods and deep learning algorithms for the evaluation of diagnostic tests. Currently, he is participating in 3 European research projects, conducting 2 scientific missions for EU COVS, action sound control, standardizing output-based surveillance to control non-regulated diseases of cattle in the EU. He is core member of the EU COVS Action Harmony, novel tools for test evaluation and disease prevalence estimation. So we welcome you, Dr. Melidis, and thank you for accepting our invitation and giving a talk as a keynote address in this event. Thank you very much, and over to you.
Good morning. Thank you very much for the introduction and the invitation to join your sermons. So basically, it's a very nice opportunity for me to present the work that we did as part of the national program on epidemiology burden and control of zoonotic diseases in India. This study, as you can see, is about assessing the diagnostic accuracy of 3 diagnostic methods to detect Coxiella burnetii infection in the domestic bovine population in Punjab, India, using Bayesian class models. So basically, in this brief presentation, what I'm trying to do is describe the main objectives of this study. That is where the diagnostic evaluation, as expressed by the sensitivity and specificity of an indirect ELISA method that was applied in serum samples and a trans-PCR method that was applied in genital swabs and milk samples. As you know, both the sensitivity and specificity are probabilities. They are defined as the true positive rate and the true negative rate accordingly. Since that in the literature, no gold standard, meaning a test with perfect sensitivity and a perfect specificity is described, this study was implemented in the Bayesian framework using the Bayesian class models. But before we go into the part of the model and the analysis that we do, I'm just going to briefly describe some basic characteristics of Coxiella burnetii and why this microorganism is considered as a microorganism of great animal and public health concern. So Coxiella burnetii basically is an obligate intracellular gram-negative Coccobacillary bacterium. I think that the genus Coxiella is morphologically very similar to the Rickettsia species and genus, and I think that's why the original term that was suggested for this microorganism was Rickettsia diaporica, because it represents the ability of the bacterium to go through to pass through filter pores. Back in 1938, Coxiella burnetii was identified as the causative agent of Q fever, that is a zoonotic disease that was first described back in 1920s in abattoir workers. And because at that point the infection agent, the cause of the symptom was unknown, it was given the name Q fever, where Q stands for query. Generally, the pathology, cattle and buffaloes that are the species under investigation in the study. However, both sheep and goats and humans are commonly affected by the bacterium, even though it has been isolated by many domestic and wild animals, birds and arthropods. Also, into the epidemiology of the bacterium, it has been addressed in the literature that several ticks have an important role because they may serve as reservoirs of the bacterium. Okay, now limited information is available about the pathogenesis and the transmission of the bacterium, and several studies have been conducted on animal models. And basically, the result is that the transmission, the dispersion of the bacterium can happen through the air and via direct contact with animal secretions and excretions, basically the vaginal discharge, the placenta, the milk, the feces, the urine, and the amniotic fluid. However, in a study published back in 2007 by Wang, he described as the main route of infection the inhalation of aerosols or dust contaminated with birth fluids. Also, it's very important to see that there is no predominant shedding route of the bacterium, because a study that was done back in 2006 by Gutao, he studied 3 different shedding routes of the bacterium. He studied milk, vaginal mucus, and feces. And his study results indicated no predominant Coxiella burnetii shedding route, and also most of the majority of the shedder cows had only one Coxiella burnetii shedding route. It was not a combination, there was only one. Moving on to the clinical signs in livestock, the form that is most commonly seen is the subclinical one. However, the clinical is rare, and basically it's associated with reproductive complications such as abortions, stillbirth, weak calves, or repeating breeding syndrome. So it's very important that we may have a bacterium dispersion via feces or vaginal mucus and or milk. It has been reported, and that could be independently of an abortion history or any clinical sign presentation. Okay, since that as we said, that the Coxiella burnetii, the causative agent of acute fever, is a zoonotic disease in the humans, we have the acute form that is described as having basically flu-like symptoms, such as atypical pneumonia, hepatitis or cardiac involvement. And the chronic form that is being described is considered more severe than the acute one and could be fatal without the appropriate therapy. Therefore, again, to summarize, that's why Coxiella burnetii is considered a microorganism of great animal and public health concern. So moving on to our case in our study, in India, according to the literature that I have searched, the first Coxiella burnetii infection in a cattle herd was back in 1952, while the first human case was in 1953. Up to that point and until today, there has been a lot of studies conducted for that disease, and this case has been an increasing trend in prevalence, outlining the potential threat of Coxiella burnetii to public health. However, the thing is that most of these studies were performed in a frequentist framework, in a frequentist approach, meaning that they assumed that in the test they were making that either a perfect test was applied or an imperfect test with known sensitivity and specificity was applied. However, in our case, what we try to do is to do the Bayesian part of this analysis. So the Panjam setting, as I called, it's 22 villages, 1 per district of the Panjam state, the northern state of India, were selected for the study. 179 households, both cattle or buffalo herds, participated, and a total of 610 animals were sampled. The sampling unit, as we call, is the speech study investigation in this study are the cattle and the buffaloes. We refer to this as the domestic bovine population, because we think that this is logical in a way that both species are members of the Bovidae family, and that the course of infection is similar in both species. Regarding the analysis and the study design, more information is available on the study that I cite here from Kesavamurthy, published 2 years ago. So the tests that were applied, we had a trans-PCR method that was applied to genital swabs and milk samples, and an IgG ELISA method applied to serum samples. As it makes sense, the population consisted of female lactating and non-lactating animals and male animals. Therefore, PCR milk could not be applied to the second category. Therefore, we split the population into 2 subpopulations, where one had included 361 lactating female animals where all 3 tests were applied, while the second subpopulation consisted of male and non-lactating female animals, a total of 249 animals where only PCR genital and IgG ELISA were applied. So moving on to the model, let me get this down. Oops, sorry. Okay, the Bayesian latent class models are the type of models that have been applied in the diagnostic accuracy studies that assume the absence of the gold standard and have been applied for over 20 years in that type of field. Basically, it's a method that we can able to acquire unbiased estimates, and the Bayesian framework can be considered as a product of the objective is to acquire the posterior estimates that can be considered as a product of the likelihood that is considered the information derived from the data, and the prior that is the model allows in comparison of prior information, that's expert information or information we know. Towards building an identifiable model, meaning a model that can estimate its own parameters without posing any, all unknown parameters without posing any constraints, we have to set and check some key assumptions. The first assumption is a definition of the infection status, meaning what do we define by infection in our study. Basically, in this case, we have the PCR method that detects the bacterial DNA, meaning the presence or the absence of the bacterium, and we have the ELISA method that basically measures the antibody titers, meaning the immune host response. So in our case, we have defined 2 distinct biological principles, meaning in a way that we may have presence of the bacterium without the presence of the immune response of the host. Secondly, we have to check the conditional independence to check if of the applied test, to check if there are correlations between the 2 applied methods, the 3 applied methods. As it makes sense between PCR and ELISA, because the methods are based on different biological principles, conditional dependence can be assumed. The main thing is to assume conditional independence between PCR genital and PCR milk. In this case, given that the 2 methods are applied in different organs, we can assume conditional independence in the thinking that presence or absence of the bacterium in one organ does not imply presence or absence of the bacterium in the other organ. And the third key assumption is whether or not we are going to incorporate prior information. Again, this can be considered as an optional step. However, in our case, we run the model both with informative prior information that was provided by an epidemiologist and an expert on zoonoses and a member of the project, of the national project in India. And we run the model again without informative priors. And as will be demonstrated later, we chose the one with informative priors. So to move, okay, here, as we said, the Bayesian framework is about estimating the posterior distribution to acquire the posterior estimates that can be considered as a product of the likelihood and the prior. The likelihood, as we said, is calculated by the data, and on the upper left table, we can see the cross-classified results, the test results on the 2 subpopulations. We can see that the main thing with that is that we had 0 cell observations. And on the right table, we can see the prior estimates as expressed by the mean and the 95 percentile value. In a way, we know where we think that the true answers should lie around, in which space. So with that in mind, we run the model using the OpenBox software. And in this table, we can see the posterior medians and the 95% probability intervals for the sensitivity and specificity of each applied diagnostic test. We can see that all 3 tests are highly specific, with PCR genital having the largest specificity. And IgG ELISA was the method with the highest sensitivity. That makes sense since serological techniques are considered more sensitive. Again, as part of the sensitivity analysis of the model to check the robustness, we ran the model assuming not informative priors, and to check that the cause of the infection is similar between the 2 species, we ran between cattle and buffaloes, we ran the model using only the test results from the cattle. In the first case, without informative priors, The specificities were similar. However, the sensitivities were slightly different regarding, I think, most of all, the PCR milk. In the second scenario that we had to check, the second assumption we had to check, there was no difference in the estimates using all the data from the cattle or the data from cattle and buffalo. So with that in mind, and to summarize, it is important that in order towards controlling Coxiella burnetii and getting accurate and unbiased estimates, a combination of diagnostic tests that are based on different biological principles may provide better information on the Coxiella burnetii infection. And to close, I would like to thank Professor Nadine Dant, who suggested me and introduced me to be a part of this symposium. I would like to thank Professor Barbiri Sain for helping me with the analysis, and I would like to thank Professor Polchons Kostoulas, who had helped me undergo the analysis and present you the results today. I don't know if there are any questions.
Thank you very much, Dr. Meritis, for your wonderful talk. So one of the question is about how do you make these assumptions when you are saying, so is there some guidelines for those assumptions, or how do we do that?
Yes, there are some guidelines assumptions in a way, because those are the three assumptions that you need to provide a definition of infectious status. What do you define? To calculate what numbers of parameters you need to estimate. Basically, here we have to estimate 6 parameters, 3 sensitivities and 3 specificities. And you have to assume for the contextual dependence or independence, if it is there, to adjust for the potential correlation. So basically, those are the, let's say, the guidelines of Bayes and latent class analysis. Yes, those are 3 main assumptions that you need to take into consideration while constructing a BLCM.
Thank you once again. One more question is about, so one of the participants' question is about, so when we don't have any gold standard, so at that point we usually use this Bayesian model?
No, you can use Bayesian model at all points. However, when we don't have any gold standards, that's where they are the only solution in a way. You can use Bayesian latent class models even when you have gold standard information, because as Bayesian stated, there is no perfect test. All tests are missing something. So with a BLCM, you can get the unbiased estimates for the gold standard. You will know the sensitivity and specificity of the gold standard, which is not definitely 100%.
Okay. One last question quickly.
Yes.
So how is it different from normal statistical method? Like,
the main difference, to be honest, is that you allow the model to include prior information, and this allows you to build more complex modeling. Prior information is not any way that you will, with priors, you can get the answer you want to have, but it will help build more complex models in that way.
thank you very much.
Thank you very much. Thank you.
Thank you very much once again for your talk, and looking forward for such collaboration in future too. Thank you very much, Dr. Meritis.
Thank you very much.
So now we are going to invite our next keynote speaker, Dr. Hannah Holt. Dr. Holt, she is a research fellow in One Health at the Communicable Diseases Policy Research Group in London School of Hygiene and Tropical Medicine, and Veterinary Epidemiology, Economics and Public Health Group at Royal Veterinary College. So her main research focuses on the transmission of zoonotic pathogens at human-livestock interface, and she has completed her PhD from RVC on the development of evidence-based control programs for bovine brucellosis in Punjab state of India, and was based at GADVASU during this time. So we welcome you, Dr. Hannah. Thank you very much for joining us today, and you can share your screen, and over to you now. Thank you.
Hi, good morning, everyone. I'll just say a quick hello and then work out how to share my screen.
Yes, you can share. Yeah.
Okay.
Just to let the participants know that it's early in the morning, if I'm correct right now.
Yeah, it's
Yeah.
very early in the morning, like 6:30, but it's very grey and raining. So hope the weather is nicer for you.
Thanks for your time, Hannah. Thanks.
No problem. Okay, so I share. Okay, can you see this? Wait, let me check. Okay, sharing this one. So it's working. You can see the presentation.
Yes, ma'am. Yes.
Okay.
Yes, perfect. Yeah.
Okay, sure. I'll turn my camera off. Just my internet's not amazing, and then yeah, put it back at the end. Okay, perfect.
And you can make it again on slideshow. Yeah.
Yes. Okay. Yeah, so good morning, everyone. Today I'm going to talk to you a little bit about our project. So it's between Royal Veterinary College and GADVASU, also working with PGI in Chandigarh and London School of Hygiene and Tropical Medicine. So this was mostly, it was part of a big project, but then it was also my PhD. So I'll just talk you through this. Yeah, so I don't need to tell you that India is the world's leading milk producer with the largest bovine population, and Punjab state produces the most milk per capita. But as you know, the majority of this is still produced in small rural households. So this project was looking at brucellosis, which is an important zoonotic disease, imposing significant impacts on livestock production through reduced milk yields and abortions, and also is a huge public health issue worldwide. So the disease is transmitted to humans via consumption of unpasteurized or not heat-treated dairy products, or direct contact with livestock, particularly around calving. So the WHO has estimates that there's around 800,000 cases of brucellosis per year, but you can see there's a wide uncertainty interval. And when a systematic review was conducted of the frequency of human brucellosis, no studies in Asia were retained in this systematic review that passed the criteria to describe human frequency. And obviously, given the size of the human population in this area, and also bovine population, it's important that we have estimates of how prevalent the disease is here. So the aim of this work was to investigate the epidemiology of bovine brucellosis to provide evidence for its control in Punjab. So my PhD was kind of split into two main parts. So the first was to look, these first parts I won't talk about today. So this was a situation analysis of the dairy sector. And then we did some risk ranking of milk-borne hazards to look at the possible importance of Brucella via the foodborne route. But because of the heat treatment of dairy products in India, this wasn't considered a high-risk route of exposure. And then today I'll talk to you about the cross-sectional surveys we conducted of brucellosis. And then we then looked at scoping for improved control. So we had a stakeholder workshop. We looked at comparison of serodiagnostics. Again, I won't talk about this today, but can another time. And then we did some disease transmission modeling and combined that to make recommendations for a progressive control pathway for brucellosis in this setting. This is quite a key time in brucellosis control, because at the same time they were launching a Brucella Free Village campaign, which some of you may know about. So they were proposing a pilot program to try and eliminate brucellosis from 10 pilot villages in 10 states, and Punjab was included in this. Yeah, so the first thing I'll talk to you about is our cross-sectional survey of brucellosis in dairy farms. So the aim of this has just been published in PLOS NTDs, was to estimate the seroprevalence of brucella in cattle and buffalo in dairy farms in rural Ludhiana, and to estimate the seroprevalence in persons in direct contact with this livestock, and then to identify risk factors associated for both species. So this is an overview of our survey. So first we selected 4 out of 7 tehsils, and then selected 60 villages, probability proportional to human size. So the larger the village populations, the higher the probability of being selected. And then within these villages, we conducted 2 parallel surveys. So this part was led by PGI. They conducted a survey in the general population. So regardless of whether or not they were keeping cattle, they did a random selection of households, and then they sampled 1 male and 1 female household member. And then if the household was keeping cattle and buffalo, they requested a bulk milk sample. So this was one study, and then this is the study that I'll mainly talk to you about. We also conducted a random sample of up to 8 dairy farms, which we classified as having 4 or more dairy animals, so adult female cows or buffaloes. And then we conducted a random sample within these farms of up to 9 individual animals. So we collected individual milk samples from up to 9 animals that were giving milk. And then we offered all persons on the farm testing for brucella. And this is quite an important study because a lot of the previous work has throughout the world has looked at seroprevalence in animals or looked at the epidemiology in human populations, but here we were sampling people and animals within the same farm and within the same household, so we could really see the link between human and animal brucellosis. So a few results from this. So in persons in direct contact with livestock in the dairy farms, the seroprevalence was estimated to be around 10%. So this means 10% had antibodies to brucella, so were exposed to the disease at some point, which is obviously high. Around a third of farms had at least 1 animal test seropositive, and the individual animal seroprevalence was around 15%. So this was really high. Just as a comparison, in the general population survey, around 2% of people, so around 2% of people had evidence of exposure to brucella. So it's still significant in the general population as well, because obviously contact with bovines in this area is common. The household seroprevalence, the seroprevalence of household herds was around 10%. So this was a lot lower, mainly because in household herds, they're not trading as much, it seems. So they're not buying new livestock as often, and the herd sizes are obviously smaller. So we looked at risk factors for seropositivity. So in animals, main risk factors were cows had higher risk of buffalo, and herd size increased the risk of dairy farms being positive. So we then did multivariate logistic regression. So this is the results in the people in direct contact with livestock. And we found that people that reported assisting with calving or abortion on a farm where at least 1 animal tested positive had 5 times the odds of testing positive compared to people that did not assist with calving and abortion. So there was a lot of people in our study that would maybe assist with milking. This wasn't a high risk practice. So this was the main risk factor. And again, in the general population, the main risk factor was assisting with calving and abortion. Also, interestingly, consumption of goat's milk was a risk factor in the general population survey. Yeah, so it seems that in this area, there's a high risk of exposure to brucella in people that are assisting with calving and abortion. So this is an important to create awareness around this. So next we looked at, we conducted a stakeholder workshop. So this first one was conducted at PGI. And the idea was, okay, we now know what is the current situation, like how much brucellosis there is, the different risk factors. So let's discuss with stakeholders these findings and inform them. So there was a lot of public health participants, so from PGI Hospital or Department of Health. So we wanted to make them more aware, especially because primary practitioners in the villages we were sampling weren't aware of the risk of brucella. And then we also wanted to know, so what is currently being done to control the disease in Punjab? What's working? What do stakeholders think could be done better? And then we could use this to inform our next component, so to look at control strategies for the disease. Yeah, so we had like stakeholders were split into groups, and we asked them to say what current resources are directed towards the control of brucellosis, what are the barriers to controlling the disease, and what are possible future solutions. So some of the barriers mentioned were things like vaccination, so lack of access to S19 vaccine or other brucellosis vaccines, and then concerns over safety. So some veterinary officers and farmers being reluctant to use the vaccine because it can cause abortions in obviously older pregnant animals, and there's a risk of human infection from vaccine strain as well. Yeah, and then a main issue in humans is the lack of awareness of the disease as a zoonosis and guidelines for diagnosis and treatment. And then we also, yeah, this was the Brucella Free Village campaign, which was originally proposing to select villages for inclusion, screen animals in the village for brucellosis, and then segregate positive animals, so probably slaughtering buffalo but moving cows to a area outside the village to isolate them, which obviously in our epidemiological survey, around 1 in 6 animals were seropositive for brucella. So this would be, if we did this in Punjab state, this would be a huge number of animals that you need to isolate. So we used all this information, and then we developed a disease transmission model for brucellosis to investigate control strategies for the disease in this setting. So yeah, we developed this model, and so I'll just take you through like a representation of the model. So the model was conducted at village level because this was the level proposed by the Brucella Free Village. And within villages, we had lots of farms. So we used the data from our cross-sectional survey to develop this. So how many farms in a village on average. And then within, so this was all represented by equations, but it's used to describe a system. So within a farm, we told the model that you can have susceptible or uninfected cows or infected. And in our model, susceptible cows give birth to infected calves, and that's how you get replacements into the farm. And then we also had infected animals giving birth to infected calves, or they can also give birth to susceptible calves, and that has an associated probability. So each event has a probability of occurrence. When infected cows gave birth, they become infectious, so they secrete lots of brucella into the environment, and then at which point they can infect susceptible cattle, so they can then become infected. Once they've been infectious for a couple of months, once they've stopped shedding the bacteria, they go back to becoming infected. Calves, become heifers, and then heifers become adults. And then, heifers can become infected as well. Animals can also be introduced into the farm through purchases. So you can have new births or purchases. Then we built in vaccination. So we could look at the impact of vaccinating just calves, would then become immune to the disease, or vaccinating all ages. So doing an initial vaccination of all animals within the herd. And then, these animals can be sold, can die and leave the model. So we simulated lots of farms, and infection could be spread between farms through purchase of infected animals. So this is just a more complicated representation of that. So the model was run over a period of time, and it describes both herd demographics, so births, deaths of cattle, and also the disease dynamics, so spread of brucella. And it was fitted to our observed data from our seroprevalence survey using approximate Bayesian computation, which sounds really complicated, but basically means that we set up our model, and then you have this beta transmission parameter, which is the rate at which susceptible cattle become infected when they contact an infectious animal. And we fitted this by sampling lots of different values of this parameter and then running our model and seeing which best matched our distribution of within herd prevalences from our large epidemiological survey. So we'd used it to simulate farm level controls, so private control by farmers like vaccination or screening replacement. And then we also used it to simulate population level control programs. So things that are proposed by like the Brucella Free Village campaign. So different vaccinations, testing, vaccinating calves only versus adults. And also we looked at this test and isolation, so removal of positives. So this is just another diagram of the different states so this is the different states the animals can be in. This would be an infected calf. This is an event, so this is an infected calf becoming a heifer. This is a susceptible adult becoming infected. And then these equations represent the rates at which animals move between each compartment. And then we have different parameters. So delta is the rate of leaving the farm. So the model was used to investigate vaccination. So we looked at vaccinating different proportions of herds. So A is vaccinating all herds within a village. B, scenario B was 50% of herds randomly selected. And then C was targeting large herds, so only vaccinating herds with above 9 animals. And then we also looked at vaccinating different proportions of livestock at the inception of the control program. So whether we did mass vaccination of all animals in year 1, 50% of the herd. And then we also looked at, as calves are born, vaccinating them to see which is the best strategy. Then we also with some of our vaccination strategies, the ones that look best, we also looked at the screening, so with and without screening and removal of positive animals, to see how this impacted it. So yeah, these are the results. So I'll just point out a few. So this was scenario A, where we vaccinated all herds. And then this is the different proportions. So, so this is not vaccinating any animals at the start. This is vaccinating 50% of the herd at the start. This is vaccinating all of the herd at the start. And then each line is a different proportion of replacement calves vaccinated after. So in general, we can see that scenario A is obviously the best. Vaccinating all of the herd at the beginning versus not brings the incidence down quicker, but you can see if you're vaccinating a high proportion of calves, the incidence is brought very low in both scenarios after sort of over 10 years. Then scenario B, which was randomly selecting 50% of the herds, wasn't as good as scenario C, which is where we only targeted large herds. And this was quite a useful finding because large herds meant only vaccinating around a third of herds in the village. But because these are the herds that are purchasing the most animals and involved in the most trade, this actually, in some of our scenarios, brought brought incidence like very low to similar levels as the scenario A. Which could be useful, especially at the minute there's not enough vaccine doses available to vaccinate everything. So it allows you to target high risk herds. And yeah, so so similar results with prevalence. This is when we did the screening. So obviously removing positive animals at the start of the control program drops the cumulative incidence straight away. But sort of after you've ran the control program for a while, it brings the incidence down to similar levels as vaccinating a large proportion of replacement calves. And the reason, so this is important because in this scenario, even though the incidence was brought down quickly, it meant that you were isolating more than 100 animals per village, which is perhaps not a realistic control strategy. Also, because the test specificity isn't perfect, then a lot of susceptible animals would also give false positive results. So you were also isolating a large number of animals that were actually uninfected. And then because the incidence, you can get down to similar levels of incidence without doing the test and screening, which is a very, very, very expensive thing to do to isolate positive animals. We're kind of promoting that maybe targeted vaccination might be more appropriate at the current levels. And yeah, so this is just looks at the number of infections saved. So you're saving in the high, where you're vaccinating lots of calves, you are saving like over 1,000 infections per village per year, which if you accumulate that to district or state level, that's a lot of infections. Obviously, this is only looking at infections in cows, but then if you think about all the infections that it would prevent in terms of people assisting with the abortion of calves, that's very significant saving in public health as well. So these are the scenarios that achieved elimination, which we defined as prevalence below 2%. So a lot of scenario A and C scenarios where 100% of calves were vaccinated managed to eliminate the disease, which is exciting. And then this looked at the time the scenarios took to achieve elimination. And you can see that even in our best performing scenario, it took about 10 years to achieve elimination. So this, and now when you're talking about control programs, sometimes they're only launched for like 5 years or only sanctioned for a short time. So with Brucella, it's important to note that control does take a long time because of the chronic nature of the disease and all the older cattle present. So you have to commit to long-term commitment, long-term vaccination. Yeah, so I think the modeling said that, predicted that you could achieve significant reductions in Brucella through strategic vaccination, and targeting large herds and replacement calves may be a more efficient use of resources when vaccine campaigns are low. And then in the scenario where we looked at test and removal, you had around 100 animals, susceptible animals, so these are uninfected animals, and 140 infected animals removed from the population. So this is only over 200 per village. So these would have to be isolated outside the village if there were cows. And after running the campaign for a longer period, it achieved similar reductions in the number of new infections in the long term. So yeah, I'll just skip this quickly because we're running out of time, but the last exercise was to try and, we held a conference at GADVASU, a stakeholder workshop with Department of Animal Husbandry and Public Health workers, and presented these results and looked at how we could use them and discussed this with stakeholders. And then we also use this to propose, so according to FAO, you can do progressive control pathway for brucellosis, and that Punjab is probably somewhere between stage 1 and 2. So they know exactly, we know the situation, we know Brucella is high risk, and hopefully we can look at beginning to implement strategic vaccination and awareness campaigns. And obviously GADVASU play a very important role in increasing awareness. So the extension services disseminate these messages to farmers, and they're offering testing. And excitingly, India announced the launch of a national control program, I think last year, but obviously this is paused due to the current situation, but hopefully this is something that can be achieved in the future. And this is proposed to target vaccination of calves, which is in agreement with our results. So according to the results of the modeling, this looks like it's a very sensible strategy to target calves. So this is exciting. Yeah, so the work, brucellosis was widespread in the area. Assisting with calving and abortion was the main factor. And although we all talk about a lack of management of positive livestock as one of the main barriers to brucellosis control in India, the results of the modeling suggest that you can achieve a sustained reduction in incidence if you do vaccination. And then prioritizing replacement calves and large farms for vaccination would be sensible if you don't have enough vaccine doses for so that's my talk. So yeah, thank you for listening, and yeah, I can turn my camera back on maybe if I work out how to do it.
Thank you very much, Dr. Hannah. So
Thanks.
it was a wonderful talk. So there is a question for you. So the question is, you have mentioned that about the segregation of infected herd.
Mm-hmm.
So the question is, in the developing nations, so what would be done for these segregated infected herds? How?
So I think in the Indian setting, I think they proposed that infected animals could be segregated in gaushalas. But obviously, because the infection levels are so high at the minute, we wouldn't suggest to do that now. So usually in the UK and in other settings, you can bring the incidence down with vaccination first. And then once the seroprevalence is very low, which, I mean, in the UK, I think we took like 40 years to get to this stage. Then you can talk about, okay, now we have such low level infection. What should we do? And maybe at that point in India, you could talk about the isolating the remaining few seropositives. I mean, in other countries, they would usually cull, but obviously that's not possible in this setting. But yeah, I think when the seroprevalence is so high, which it is at the minute, you should look at bringing it down with vaccination first, and then talk about removal of positives or segregation later.
Okay, thank you very much, Dr. Hannah, once again, and I would like to thank you on behalf of our committee members for sparing your time. Thank you very much once again.
Thank you so much. Enjoy the rest of your conference.
Thank you. So now we will start our next session, which is the oral presentations by participants. So we are having our first presenter, Arun Prince Milton. So could you please share your screen?
Yes, sir.
So the time allotted is about 5 minutes. So please finish within time.
Okay. Can you hear me, sir?
Yes, so you can have it on your slideshow mode.
Is my screen visible, sir? Can I have confirmation?
Okay, perfect.
It is okay?
Yes. Time. Okay, you can start now.
Okay. So a very good morning, everyone. Thank you for giving me a start to present here. The title of my paper is High Prevalence of Multidrug Resistant Staphylococcus aureus, Salmonella, and Virulent Clostridium perfringens from fresh and processed pork, which reveals putative infection risk to humans. To give a brief introduction, as we all know that Northeast India is the largest pork consuming region. As there is increased demand for pork than the local supply, the region relies on supply of live pigs from mainland India, such as UP, Bihar, Rajasthan, and many more. And the pig production system here is of a very small holder system with backyard system of rearing without much husbandry practices. And other concerns are, absence of professional abattoir and unhygienic slaughtering of pigs in villages and retail points of a substandard nature. All these are the reasons of concern that pork may become an important carrier of foodborne pathogens. Continued required support of safety and control of diarrheal diseases, and according to WHO, foodborne diseases contribute to 600 million illnesses and 420,000 premature deaths. And as foodborne bacteria are the top-rated food safety priorities, it will be more time to study them in Northeast India, which is an important meat-consuming region. And according to CDC's 2020 report, there are top 5 pathogens contributing to foodborne illnesses in US. That is, in which 3 we have addressed in this study: the Salmonella and Clostridium perfringens. This is about our study location. We have covered 2 districts of Meghalaya, that is Ribai and East Khasi Hills. As we know, Meghalaya is a state of very low density population, and the villages are at least 10 kilometers away from each other. Here you can observe that most of our sampling points are following the road route, as the villages have their meat shops besides the road. In this study, we have covered 19 retail points in 2 districts, and 82 samples were collected. These are some photographs of retail pork shops of Meghalaya. And quickly moving on to the results of the study, these are the isolation methodology we have followed, and these are the of the criteria. And following some biochemical investigations, the isolators confirmed the deploying endpoint PCR. And this is about the prevalence of the study. It was 9.8% for Salmonella, and 24% for Staphylococcus aureus, and 46% for Clostridium perfringens. Regarding the virulence gene profiling for Salmonella, the EuroB gene was highest, 87.5% isolate. And as far as AMR is concerned, 100% isolates were resistant to penicillin, erythromycin, and chloramphenicol, and 37.5% isolates were MDR, as they were resistant to more than 3 classes of antibiotics. And this is regarding the virulent gene profiling of Staphylococcus aureus, and you can see that 80% of the isolates harbored MEK gene, and 10% of the isolates carried this Pantone valentine leukocidin gene, and most interestingly, 15% of the isolates carried this ETB gene. Which is an exfoliative toxin gene, which is important. And the isolates did harbor one or more enterotoxin genes. And with regard to AMR, 100% isolates were resistant to ampicillin, penicillin, and methicillin, and the MDR was 80%. And regarding gene in Clostridium perfringens, most of the isolates carried CPB2 gene, and the most importantly, 2.6% isolates were having the enterotoxin gene. And this is about the toxinotyping, as we have toxinotyped, and here you can see the presence of type F is important in the outbreaks. And here to visualize the overall distribution pattern of a heat map supported by dendrogram was generated using the R program. It revealed grouping of isolates from 2 different localities into mainly 3 clusters, that is cluster 1, 2, and 3, and cluster 1 contained majority isolates belonging to type A, and cluster B type A and type C. Cluster 1 only type A, and cluster 3 it is type A, type C with CPB2 gene, and type F strains also. And in this heat map of we have correlated the virulence potential and antimicrobial resistant pattern of the salmonella isolates. Again, the 2 clusters. In the first cluster, mostly it was resistant to fluoroquinolones and extended spectrum penicillin, chloramphenicol, and potentiated sulfone-kanamycin. In contrast, the majority of the isolates in the cluster 2 was only of virulent nature, not of resistance. And this is the heat map of the correlation between the virulence and the antimicrobial resistance of Staphylococcus aureus. Again, the 2 clusters were displayed here. One is of high level resistance to gentamicin and ciprofloxacin, and the other cluster was of having high virulence potential. This is about the conclusion. So 80% of the Staphylococcus aureus isolates were of MDR, and the most importantly, exfoliative toxin gene were present, which is very important for the scalded skin syndrome in infants and young children. And 95% of the isolates were having some enterotoxin genes, and SEB, that is Staphylococcus E and B, that is an important biological weapon and classified as an important select agent. And Clostridium perfringens type F strains were present that was having the enterotoxin gene, which is important because it is an important outbreak strain. Thank you so much. Thank you.
Thank you. Questions by the evaluators? Questions by evaluator?
Hello.
Yes, you can ask.
Please tell about the reason. What are the reasons of this fluoroquinolone resistance in your isolates?
Yes, sir. Okay, sir, as our isolates are from the food origin, because it can be the contribution may be from the human side, and also because most of our human handlers, they are also facing the antibiotic pressure, and it is animal source meat, and it might be due to isolate from natural origin also. So both the sides, the antibiotic pressure is there, and the levofloxacin, ciprofloxacin are regularly used in treating cases in human and veterinary side. So that might be the reason that most of our isolates are resistant to the fluoroquinolone, and also extended spectrum penicillin.
Okay, thank you.
Yes, Milton, one question from my side.
Yes, sir.
How do you think these organisms, multi-drug resistant and virulent organisms that you are talking about, are risky to humans?
Sir?
Are risky to humans.
Sir?
How these organisms, drug resistant, multi-drug resistant organisms and virulent organisms that you are talking about are risky to human?
Yes, sir. Yes, definitely. It is very obvious that it has already entered the food value chain from our study. So it will definitely, contaminate any consumer's kitchen, and due to because although the meat foods mostly they are being cooked and consumed but sometimes the consumers following all the cooking practices, if they mess up in the kitchen, definitely it will lead to cross contamination in the kitchen, and it will definitely affect the elders and infants in the houses. It will be a risky because it will be too difficult to treat infections, and it may prolong the hospitalization in case of infection.
Thank you, Dr. Milton. First, could you please stop sharing your screen, and I ask the next speaker, Dr. Deepa Jolie, to share screen.
Yeah, hello.
Slide,
Morning.
yes,
Yeah.
on the
visible?
slideshow mode.
Yeah. Okay.
And please restrict to the time, and yes, perfect. And your time starts now.
Okay, sir. Thank you, sir. A very good morning to all the eminent members of the panel and delegates. With the permission of the panel members, I'll be presenting the paper on antibiotic resistance pattern of Campylobacter jejuni isolates from marine fishes, crustaceans, mollusks, and associated areas in Kerala. So this is part of my PhD work, and as we all know, the number of foodborne organisms are like globally rapid. And of these, of the leading 4 organisms, Campylobacter species is one which is very important. And this organism has been isolated mainly from poultry and other foodstuffs also, including water. And the presence of this organism in seafoods have been rarely done. And also, there is instance of antibiotic use in aquaculture, especially in the freshwater farms that grow these fishes and shellfishes. Now, the sanitary shortcomings in aquaculture farming has resulted in augmented use of these large amounts of antibiotics for a prolonged effect and for a better growth of these fishes and shellfishes. And there's a use of non-biodegradable antibiotics which are used in the human medicine, and these may find its way into these water bodies also. And because of this, there is a chance of the resistance, the transfer of resistance determinants between the microbes of both humans as well as animals. Now, and this has resulted in antimicrobial spread in the environment. Taking all these facts into consideration, the present study was undertaken to study the antibiotic resistance pattern of marine isolates of Campylobacter jejuni. And these marine isolates were taken from the fishes, crustaceans, mollusks, and the points of contamination in fish catchment areas of Kerala. Now, talking about these, the number of the samples that were collected, around 245 samples were totally collected from these fish catchment areas. Of this, around 12% were showing the presence of the organism. Now, and of these 21 isolates, the marine isolates were subjected to in vitro antimicrobial potency of the most commonly administered antibiotics. That's around 36 different antibiotics were tested here using the DIS diffusion method by the procedure Saheli and co-workers in 2007. Then in this, the Mueller-Hinton agar plates with 5% sheep blood was used, and the organism was, the culture of the organism was inoculated, and it was incubated microaerobically at 42 degrees centigrade for 24 hours, because this organism is it's coming under the thermophilic group, the Campylobacter jejuni. Now, the clinical breakpoints of Campylobacter species susceptibility testing was defined according to the UCAS 2020, and the isolates depending on the zone of inhibition were grouped as sensitive, intermediary, sensitive, and resistant. Now, this is a antibiotic resistance pattern, the procedure by which this was done by use of Macfarlane standard 0.5 Macfarlane standard, and the preparation of the inoculum. Inoculum was then plated onto the medium. This were applied, and zone of inhibition was made. This is just the list of the antibiotics and the concentrations that were used here. And going over to the results, all the 12 of the 21 isolates, 12 were from fish and shellfishes, and 10% showed resistance to cefepime, and more than 90% showed resistance to, of the isolates showed resistance to cefotaxime, ampicillin, and vancomycin, and whereas 10% was sensitive to doripinum. Now, the 9 isolates from the points of contamination that is points of contamination really meant collecting samples from the boats, the surface of the boats, the boat decks, the flow onto which the fish was unloaded, the baskets, the containers that were used, the ice that was used, the water that was used for cleaning. So these were the points of contamination. And 10% of those isolates, that is the 9 isolates from these points of contamination, showed resistance to cefotaxime, cefepime, and ampicillin, whereas 10% showed sensitivity towards ceftriaxone, azithromycin, gatafloxin, carbonicillin, and nitrofurantoin. Now, this is a pattern of the antibiotic resistance pattern that was obtained, and around 13 different classes of antibiotics were done here. So, of this, this shows the resistance pattern of both these slides of Campylobacter jejuni in seafoods and the environment, related environment. Now, this is a diagram showing the resistance pattern graphically, and so as we can be seen here, cefepime showed 100% resistance, followed by the cefotaxime and ampicillin and vancomycin. Now, this is a dendrogram showing there are 4 different clades which are formed here, showing similarity between that the action of that the resistance pattern of that particular antibiotics of these isolates. Now, 10% of these isolates from seafood and related environment showed multi-drug resistance, because more number of classes were applied here. So we got 10% with him, because 10% showed the MDR, and the multiple antimicrobial resistance, which is a number of antibiotics applied, that is showing resistance to the total number, and this was ranging from 0.33 to 0.72. Above 0.2 indicates that these are possible risk contaminant points of risk. Now, measures to regulate, there should be creation of awareness. You should monitor the use in aquaculture farming, then control of unregulated antibiotic availability, enhance the biosecurity measures that are followed, capacity building, improving the veterinary supervision, water quality management, pond preparation with liming practices, adhering to the withdrawal periods can somehow reduce this. Now, to conclude, I would like to say that the fish and water are acting as potential source of the human infection. Antibiotic resistance is on the rise, as seen in the study, and antibiotic use in aquaculture has to be controlled. A concerted multi-stakeholder action is required to reduce the emergence as well as the spread of antibiotic resistance in aquaculture. Thank you.
Thank you very much. So, question for evaluator 1. Any questions for you?
Deepa, you have used, I think, 30 or 36 antibiotics, and
yes.
From each class you have used several antibiotics.
Yes,
Do you
sir.
think you could have avoided that? That many
Actually,
antibiotics?
Actually, we just wanted to see how the resistance pattern was going. Like usually we go for very few numbers, around 10 or 12 usually. So we just wanted to see the commonly used antibiotics, what are being used, especially in the aquaculture. In aquaculture, actually, chloramphenicol, ciprofloxacin, and furazolidone are the most commonly ones that are used in Kerala. So I just wanted to see how it would, how the antibiotic resistance pattern would project in this antibiotics. That's why I took this many number of antibiotics. So.
See, you have also used vancomycin.
Yes,
is it recommended? It is recommended.
No, it is not recommended, sir, but it is present in the environment, because that is why this isolates, they have like acquired that resistance towards that antibiotics in some of the isolates that I have obtained here. So that is why we have to be aware of the environmental contamination by these antibiotics that is being used, especially in the water bodies. I was getting so many isolates that were found to be resistant to many of these antibiotics, and the birds that are usually frequenting all these fish areas, these were also found to, the droppings I had collected, these were also found to have this organism, and which were also found to be having that resistance pattern, sir.
Thank you. Any other question from evaluator 2? Any question?
Okay. Ma'am, please tell me the Campylobacter whether require any treatment or not, whether it is self-limiting in human being or this require treatment.
Actually, sir, Campylobacter infections are actually self-limiting, especially in the Asian population. It just tends to give, it's in some cases, they usually show some very mild symptoms. But in the case of non-Asian population, the symptoms are much, they're much more prone to fatal cases. So there is a chance, that is why we have to be aware of this organism existing here. Actually, the Campylobacter is not usually organism which is very easy to like work with. So that's why we went for this organism.
So what was the status of this aminoglycoside in your this multi-FDR pattern? What was that?
Compared to the other antibiotics, this was better, like it was showing better results. It was sensitive to some of the antibiotics that that group, so the isolates that I obtained. Compared to the other, because this organism, it has a natural inherent resistance towards the quinolones and the macrolide group. So but compared to that, this was showing very much sensitivity, sir.
Okay, thank you.
Okay, sir.
Thank you very much, Dr. Deepa. So could you please stop sharing your screen? And I now invite the next speaker, who is Dr. G.K. Sivaraman. So please share your screen. And you have 5 minutes to speak, and then 1 minute discussion. All set, Dr. Sivaraman?
Yeah, yes.
Okay, sure. Yeah, it's could you just scroll down a bit? Not able to see the title.
Okay, one. Thank you. Just.
That's perfect.
Yeah.
Deepa, could you please? Okay.
Yeah.
Okay, so yes, perfect. Dr. Sivaraman, now you can start.
Yeah, good morning to all. I'm Dr. Sivaraman, working as a principal scientist at ICRC-IFT Cochin. And we are working as an Indo-UK project. And these all the genome sequencing were carried out at Department of Veterinary Medicine, University of Cambridge. My work is a prevalence of virulent and biofilm forming ST88, 4 and spot type 2526 MRSA clone circulating in local fish market in Assam. Yeah. As all we know, Staph aureus is a ubiquitous organisms, and also it is considered to be one of the important opportunistic pathogens, and it causes a wide range of diseases and toxinosis, and often sometimes life-threatening due to bacteremia as well as sepsis, and also it will lead to pneumonia and cause severe fatality. In addition to that, if it is carrying an extensive amount of virulence factors and causing enterotoxin, exfoliative toxin, TSST, PVL, Staphylococcus hemolysin, and superantigens. And also, if it is carrying this antibiotic resistant and it's become multi-drug resistant and become a MRSA, then it is considered to be one of the important public health zoonotic. As far as India is concerned, these cases are escalated alarmingly, and also it is still on the rise. As the recent report says that this incidence of MRSA in India is increasing at least 18% over the last 10 years. And as we know, this MRSA carry this MECA gene and the Staphylococcus cassette chromosomes. So it's having that higher transmissibilities. Thereby, it have the greater potential to disseminate AMR genes to other hosts as and when come in contact. And as we know, this MRSA in 1960, it was reported after the introduction of penicillin in 2 years, and then we could found that it was the incidence of MRSA was noticed in the hospital setting as well as nursing home. And then within the 2 years of incidence of MRSA, and also it was noted in community settings where they are not exposed to. And then now recently, MRSA is found in animals, food origin, and environmental sample also. So fish I have collected because our fish is considered to be one of the important nutritious items, as well as it will contribute significant economic returns. So a lot of reports recently there, the incidence of MRSA in food origin of animals as well as aquaculture settings. So the present investigation was carried out to monitor the incidence of MRSA and its virulomes and resistance and whole genome sequencing study in order to understand the molecular epidemiology and zoonotic potential of this MRSA from fisheries environment. In this study, we have selected 3 sites based upon that drivers of AMR. that is hospital setting, industries, animals, environment, everything. And we selected 3 sites based on these AMR drivers, that is low, medium, and high. And we went to the site and we developed a stakeholder map, that is including mapping of all the household, animals, hospital, industries, and fishery sectors. Site 1, this is site 2, where the poultries are kept and hospitals are there. And this is the site 3, where the animals are there. And this is about, from last 2 years, we have collected around from these 3 sites 173 samples, and we isolated selectively an MRSA by using PD Phoenix system, where it will identify this Staphylococcus species, as well as it will give the antibiotic resistant pattern at the level of MIC, and then it will give the alert whether it is resistant to methicillin or vancomycins. Once we identify it is a MRSA, then we have developed PCR, triplex PCR, that is, it's containing McKay, NUC, and Staphylococcus genus specific primer, and we can see on the left side, so it will clearly shows that MRSA. Once we identify its MRSA, then we have genotypically characters for its resistome, virulence, and then SEC MAC type and whole genome sequencing. And these are the different set of primers we use for complete characterization of this MRSA genotypically. Around 8 set we used it. The first one is genus identification, then McKay locus, then these are the toxin genes we used it, these are the biofilm associated gene we used, these are the MAC typing primers we have used, these are the spot typing, then accessory gene regulator, then MLST we did for this MRSA isolates. So for the result we considered that for the site 2 is considered, it is expected that more number of Staphylococcus as well as MRSA accordingly it was there. So the overall incidence of Staphylococcus aureus in these 3 sites was found to be around 55%, and the site 2 it was all the site it was almost equally Staphylococcus aureus was there. As far as MRSA is considered, site 2 was the highest, that is 87% was there, followed by site 1 from 13%, whereas in site 3 we couldn't found. So it is not presence of MRSA in food, in fish. So definitely it is a ubiquitous organisms, and it was present, and it is clearly indicated that there is a possibility of potential contamination, because fish is the one of the commodity where it is most number of time it is handled, as well as that we know that hygienic condition of the vendors in the retail market. And this is shows that antimicrobial resistant pattern of these MRSA isolates. The most commonest MRSA profile shows that it is resistant to ampicillin, cephalosporin, foxtin, then followed by norfloxacin, oxacillin, and penicillin, with the MAR index of 0.35 in 74% of the isolates. And the second antimicrobial resistant profile go with that same antibiotic with erythromycin and with the MAR index of same 0.35. So it clearly indicated that it is a possible risk of these organisms. And as we know, MRSA, once we declare it is a MRSA, considered to be a multi-drug resistant. And definitely because of this multi-drug resistant, in our study also it is showing more than 3 classes of antimicrobial resistant to antibiotic in addition to penicillins. So this dissemination of this multi-drug resistant MRSA mainly due to its genome plasticity and presence of IS elements. And this property shows that the presence of virulent genes, and all the MRSA isolate in site 2 is potentially harbor that all kind of virulent genes like PVL and enterotoxin genes like B, G, and I, and these all others are biofilm associated genes. Similarly, in case of site 1, there is the one of the important gene we used to consider is a SEB. The we knows this is one of the potential enterotoxin causing genes, and we also this isolate we extracted the crude and we injected it into mice, and we found that it is causing severe food toxins. This is the overall picture of the genotypic characterization of these MRSA isolates. And we found that these SEC MAC typing by using the recent classification around 13 sets of primer, and we would able to type the first 4 isolates that mainly belongs to type 4. So it is clearly indicate that the origin of humans, and rest are we could not able to type. As far as these AGR types are concerned, this type 1 and type 3 majority of them are. This also it is clearly belongs to the human isolates. As far as the spot typing is considered, it is T22526 as well as this ST88. And this is the unique clone we have found in fishery environment. It is the nowhere it was reported, and it is considered to be one of the novel clone that is circulating in the fisheries environment. And then we taken this isolate and we went for this whole genome sequencing. We annotated this one and we mapped all these antimicrobial and resistant genes. These all are as per the genotypic characterization. All the genes also we could found based on the whole genome sequencing studies like resistome as well as virulomes in this one. As far as the consideration of this pathogenicity by using the pathogen.
Dr. Sivaraman, could you please wrap up quickly?
Yes. Just 1 second. That's it. So it is clearly from this pathogenic finder. It is based on the whole genome sequencing. It shows that potential human pathogen with 100% minimum identity threshold in respect to more than 800 pathogenic families. All these most commonly present in the plasmids are INC18, Rep3, and RepL. And based on the whole genome sequencing study, it typically belongs to CCR type 2 and SEC MEC type 4A and 2B element. In our study, the uniqueness of the whole genome sequencing, we could found that an emerging Indian clone, that is ST672 and spot type T1309. And another one is ST5 and T015. These are the first time we reported in India, we reported from the fisheries environment. And this is based on the genotypic characterization. It is recently published in food control journals. And based on this whole genome sequencing, we found that ST672, it is submitted to environmental microbiology report. So with this, I would like to conclude the prevalence of this clone ST88 with a higher level of virulence and resistomes. It is clearly suggested that it is very nicely, it will adjust to that fisheries environment and it is circulating in this environment. And based on this whole genome sequencing study, it's clearly indicated that these are originating from the hospital settings. And quite nice, very high important genetic potential as and when these consumers are the stakeholder, when we are likely to eat the fish, definitely they may contract these clones. So these studies strictly warrant that active surveillance studies of MRSA in fisheries environment and also emphasize the paramount importance of food handlers and consumers to follow the strict hygienic practices. And also we should adopt the proper ETP, that is effluent treatment plant in the nearby hospital setting in order to prevent the spread of these notorious MRSA from this setting. So I want to acknowledge this DBT as well as ESRC UK jointly funded this project to carry out. Thank you.
Thank you very much, Dr. Sivaraman. So now the questions from evaluators. Quick questions.
Dr. Siva, in your study, I see all your isolates, they were sequence type 88. How this?
Yeah.
You didn't find any other sequence type?
Yes, sir. That in case of MSSA, it is methicillin sensitive Staphylococcus aureus. Sir, ST5, sir. MRSA all are belongs to ST672. And we used for whole genome sequencing MSSA. So we want to compare this. What are the different kind of virulum as well as resistome it is carries. So we could found ST5 and spot type 105.
Thank you very much.
This is the first report from fisheries environment. Okay. So far it was not reported.
Thank you once again, Dr. Sivaraman, for your wonderful talk.
Yes, thank you.
Thank you very much.
Thank you a lot for giving this
now
opportunity.
Thank you. So now we will move to our next keynote speaker. We will stop here about this session, bacterial zoonosis. We will start having those oral presentation after these two talks. First by Dr. Pika and then by Dr. YPS Malik. And thereafter we will start with Dr. Siva Chandrian, Dr. Senthil Kumar, and Dr. Vijay Sharma for their oral presentations. So we are having our another keynote speaker here, Dr. Pika. And
good morning.
Good morning, Pika. How are you?
All good, thank you.
Oh, good. Okay.
So Dr. Pika, she has been working on toxoplasma for more than 2 decades, and she has been associated with One Health aspect of this parasite. We are very lucky and that you accepted our invitation and spared your time. You know that we do have another meeting after this talk. So without wasting time, I once again thank you, and then over to you, Pika. Thank you very much, Pika. Thank you.
Thank you. Thank you so much for inviting me. I'm most happy to start this World Zoonoses Day with all of you at this seminar. So really, really timely topic. So indeed, my name is Pika Jokelainen. I am based in Denmark, Copenhagen, at State and Serum Institute, which is a public health institute that also has the veterinary preparedness functions. So really a One Health institute. And my work is indeed a lot about One Health with focus on zoonoses, especially zoonotic parasites. And Toxoplasma gondii has indeed been my favorite parasite and my key professional focus for quite a long time. And it is a great example of a One Health parasite. It is zoonotic, it can infect, and it can cause disease in humans and many, many animal species. And it has an environmental stage. So the humans, animals, environment, the classical aspects of One Health, they are all really strongly there. And with my talk now today, I hope to encourage you all to look at zoonotic pathogens, including Toxoplasma gondii, with a One Health point of view. So, I mean, simply that is needed. We simply cannot address zoonoses efficiently enough if we do not look at the whole picture. And it is a complex picture. Actually, it's a major simplification to say humans, animals, environments. There are many different animals, many different connected environments, food, feed, water, soil, and all of these things are connected in many ways. And sometimes it's about the words, actually. So for Toxoplasma gondii, it's often mentioned as a foodborne parasite, which is an interesting expression, actually, because food, that is animals or plants that humans eat. And before we start to call them food, they lived in the environment we all share. And in that setting, they could be infected or contaminated with Toxoplasma gondii. And then they may be sources of infection to us humans. So everything is really closely linked. With parasites, we always look at life cycle illustrations, and I really love them because they tell these linkages so nicely. Toxoplasma gondii has 3 infective stages, and the infection can be acquired in many ways. Most of the infections are acquired after birth during the lifetime of the host. Here it's illustrated, for example, by those arrows going to the 2 adult humans. And the congenital infection that is also shown there, actually that is also practically just a further arrow from an acquired infection of that mother. So it's also linked that way. Certainly one of the key reasons why we really need to prevent toxoplasma infections better is the congenital toxoplasmosis. It is a devastating disease. It's heartbreaking. We must do better in this. In humans, the parasite can also cause many other manifestations, most severe ones usually in the immune compromised, but also in just anybody. One disease manifestation that is causing substantial disease burden is ocular toxoplasmosis, so the eye disease caused by the parasite. Well, to simplify these many arrows and ways here, so if you look at those that go to the humans, we can simplify and group them to be a meat-borne pathway. So ingesting the parasites that are present in meat or other tissues or milk of infected animals. And then there's environmental pathway that is ingesting oocysts of the parasite that can be contaminating food, water, or environment. And key thing is actually that this environmental pathway is a fecal oral transmission, because the oocysts are the forms that are shed by infected felids, cats, in their feces. If they get the infection, they can do that. Compared to many other pathogens, with a transmission by ingesting something contaminated with fecal matter, Toxoplasma gondii oocysts are really an environmental issue. This is because the sporulated oocysts, they are highly resistant to environmental factors. This is impressive, I would say. Here there's one arrow on top of this table that is highlighting one result that is really showing this resistance in the environment. So sporulated oocysts were kept in water in +4 degrees for more than 4 years, and they were still infective. So this shows that environmental contamination with Toxoplasma gondii oocysts, it is really a long-term contamination. And it's been also shown to spread further. For example, I'm sure you have all heard about the sea otters in the Californian coastline, where oocysts from the terrestrial felids are contaminating the sea environments where these hosts then are living. And one thing about these oocysts, they are indeed one of the 3 forms that can cause also human infections, also outbreaks, including waterborne outbreaks. Moreover, if you think about the meat-borne pathway, those tissue cysts, the forms in the tissues of the animals, they don't just appear there in the animals. So they exist only in infected animals. And as those animals that humans usually eat are herbivores or omnivores, typically those infections actually are from the oocysts. So I would say, I mean, oocysts should not end up to be eaten by the animals that are raised for human consumption. It's kind of surprising that quite substantial proportion of domestic animals raised for human consumption worldwide, they have been eating or drinking something contaminated with feline feces and this zoonotic pathogen. So either their feed or water, the environment is contaminated. So there's a lot to do to do this better. Going further upstream the arrows, very important also is that the oocysts, they don't simply appear from the cats. Felids, the cats, they can only shed if they get the infection. And actually in the original version of this illustration, there was an important arrow lacking. I have added it there. So I have adapted and added an additional arrow to the upper part with tissue cyst going to the cat. This illustrates a result from several studies showing that for domestic cats, like pet cats, if their owner gives them raw meat as part of their diets, that's a very common risk factor for the infection. Anyway, the arrows, they lead to arrows, and humans, we are usually like dead-end hosts because we are typically not eaten by any other possible hosts. If you focus on the arrows going to humans, even though we know so much about this parasite, we actually don't know much about these arrows. The relative contributions, the relative importance of these arrows is largely unknown. And it's very likely different in different settings. So this is a studies that we should be doing all over the world. Not knowing more about the source attribution has been partly due to lack of appropriate methods. And this is both laboratory methods and also modeling approaches. Different source attribution methods, they have been used for zoonotic foodborne bacterial infections and diseases extensively and really to prioritize food safety interventions. But many of those methods, they are not directly applicable to parasites and parasitic infections. This is still quite recent document from European Food Safety Agency. They had a scientific opinion that listed different approaches there are for doing source attribution for Toxoplasma gondii, all with limitations and all also with potential. Interestingly, outbreaks seem to be going unnoticed in Europe. Some, especially waterborne outbreaks, have been reported from other areas of the world. There are a few of these quantitative microbiological risk assessment studies that have provided evidence on relative contribution of different meat types, but not what is the relative contribution of the meat-borne versus the environmental pathway. It's also been mentioned that studies that would be using serology that is able to differentiate whether infection is from oocysts, that could have a lot of potential. Typing challenge is that the current genotyping and typing methods, they often don't allow to discriminate enough between the strains to really trace the sources. So the most frequently applied approach for attribution, like just doing the attribute, saying where the Toxoplasma gondii infection could be coming from, so attributing those to the major transmission routes, has been expert elicitations. And this is really a gap, especially because Toxoplasma is highly ranked among foodborne parasites, both globally and also in Europe. So it's really in the top in all these ranking studies. And luckily, this gap in Europe was recently turned into a funding opportunity. And I have just a brief mention of our project called Zoonoses, which is a joint research project within a larger One Health project called One Health European Joint Program, One Health EJP. We have more than 20 partner institutes across Europe now working on Toxoplasma. This is a 3-year project. And this is our research question, which we answer using different approaches, including both modeling and new methods. And we do several multi-center studies. Major outcomes of this project will be quantitative estimates of those relative contributions of the main sources and transmission pathways and routes to humans. Focus in this study is in Europe. Major outcome already, I would say, is the consortium. So we are now 1.5 years into this project, and I'm a little bit biased, of course, but I think I can convincingly say that based on this experience, a cross-sectoral international consortium is really a strength to address a zoonotic parasite that is present widely in different areas. So I would encourage you to follow our work and hope to widen the collaborations in the future also outside of Europe, because this is a global parasite. So let's hope that this is an example for future collaborations. Back to this picture. It's not all about human infections, of course. Toxoplasma gondii is indeed also infecting animals and causing disease in animals. In this illustration, there's no wildlife really, but wildlife is certainly relevant. Wildlife can be indicators of how widely the environment is contaminated with Toxoplasma oocyst. Wildlife can be sources of infection when they are eaten by other hosts. And they can get clinical, even fatal toxoplasmosis as well. Many species have been described. And domestic animals, they're not just the source of the infection to humans, they can get sick from this parasite infection and have clinical toxoplasmosis. Classical example, I would say, is abortions in sheep, which is also a major economical challenge. But also the cats, the definitive hosts, I mean, we usually mainly talk about them as the source of the oocyst, but they can also get sick and even die from Toxoplasma infection. These are examples of how the histopathology looks in cases of fatal feline toxoplasmosis. So when a domestic cat has died from generalized toxoplasmosis. Liver, lungs, and the central nervous system are typically affected. And here is also immunohistochemical staining that is showing the parasites at the site of the tissue damage. And the infection is very common in cats, in felids, in domestic cats. They are really a host that is getting the infection easily. These are just a selection of studies of many of my students from my region, corner of the world, showing that it is common in domestic cats. A large proportion of domestic cats have been infected. So seroprevalence here, this is IgG antibodies that were measured. Having those shows that the cat has encountered the parasite and made antibodies against it. And what is interesting is that here we can see the proportion of antibody positive cats by age in years. It is very important to notice how the seroprevalence increases by age. This means that the infection is typically acquired during the lifetime of the cats, as in humans. Very similar picture actually for humans. Of the young cats, it is clearly a smaller proportion infected. and only those who are infected are shedding and contributing to the oocyst contamination. Risk factor analysis for cat infections have indicated that major source for them is eating tissues of other animals that are infected. They are carnivores, cats are carnivores. Makes sense. These are again studies from my region, and here it's clear that eating raw meats that the owner gives to the pet cats, or if a cat is allowed to go out and hunt small prey animals, those are the risk factors for the infection. So indeed, those arrows that I added to the illustration. And once more back to this picture. Indeed, those risk factors are those arrows up there. If a cat gets raw meats of infected animals or is allowed to hunt small animals that could be infected, then it has a risk to get infected. Turning that the opposite way is that if the cat does not get infected, it will not contribute to this picture. So we should not blame the cats and the felids for this. They are just part of it. They are a key part, but it's more like, are they given the opportunity to contribute to this picture? And yeah, depending on how cats are kept in different areas of the world, I think we could do in many cases more to prevent the infections of the cats, and also to think that we keep the cats so that their feces, where there could be oocysts if they got the infection, that these feces don't contaminate what the other hosts are eating and drinking. So we should really think of the whole stream of these arrows. If we stop the transmission or limit the transmission better upstream at the earlier arrows, then there would be less toxoplasma downstream these arrows. So less contamination of our common environment, smaller risk for contamination of vegetables, salads, water, also feed of the animals, and fewer infections in animals and in humans. And well, we do know that it's challenging to stop a zoonotic parasite that has so many ways of infecting new hosts. But we should, so taking that into account, certainly we also need to repeat and emphasize the really last arrows too before the humans. So washing the salads and vegetables, wearing gloves or washing hands when doing work with contact to soil, and cooking the meats well, pasteurizing milk. These really work against this parasite and disease too. So really my take-home message from this talk, always when you consider a zoonotic disease or zoonotic infection, I would encourage to think how the humans, the animals, and very often also can change something. I think we should think like alongside the transmission chains, a bit further upstream. And also, I would encourage to think about the reasoning, the motivation to do something. It is to protect human health very often with zoonotic infections. Sometimes the focus is in the animal health, and it's also the environmental health, or how contaminated or not it is. So it is really to protect one health. And with that, thank you very much for your attention.
Thank you very much, Dr. Pika. For giving detailed insight on Toxoplasma gondii as one health parasite. And I think it is a very relevant topic in concern of the zoonosis aspect, as toxoplasmosis is becoming an emerging zoonotic disease in many parts of the world. So there is one query from the participant. Pika, could you like to share your views on the toxoplasmosis or Toxoplasma gondii in developing countries like in India or other countries where the stray cat population is a very high in number, and there is also probably contact of stray cats with the human beings when they are living in the nearby houses of the humans. So what do you want to say about this?
Really excellent question, because the challenge is, I mean, this is so complex picture, and the parasites is Toxoplasma gondii is awesome in adapting to different environments and which hosts are available and how it can be there. So really all this, it needs to be, even in Europe, there's differences by area. And I would say the main challenge in more like developing countries is that this is an endemic parasite. Somehow it's easy to forget, it's neglected. It's really causing a disease burden. It's somehow not so high on the focus as it maybe should be if you look at those ranking things. And then another thing is that very often it's more like where it's possible to start making some changes along the line. So if it's difficult there where the cat is, maybe it's somewhere later. I mean, there is always, luckily, I would say, I more like see these arrows as opportunities to do something. But really, this is a challenge when there's a lot of cats roaming free. So then it's more like, how can we stop that the feces of the cats would not go directly to our drinking water, for example, and how we are having salads and so on, vegetables like that area. So maybe it's possible to go that way. But a really important question. This is there's no like one fits all way to address it.
Thank you, Pika. And there is one more question that do you have any idea about how long the infected cat they discharge inside?
This is also a really good question. So actually, the shedding of the oocyst is short term. It's usually like it starts after the infection and it lasts few weeks. Previously, it was very strongly thought that it's only once in their lifetime, but it's been shown that they can shed repeatedly. So one shedding period is short by limited time, but it's highly efficient. There can come millions of those oocysts, and then yeah, it can be repeated. And usually the cat has no clinical signs. It looks totally healthy. So we cannot know from the cat that this is now shedding. A further thing is that the oocysts, when they come out, they are not immediately infectious. So it takes 1 to 5 days for them to sporulate and become infections. But so that can be sometimes relevant too. Yeah.
We don't have any further questions. So we are grateful to Pika for giving this wonderful lecture on Toxoplasma gondii and also very good or informative reply to both of the questions asked by the participants. Thank you so much.
Thank you. Bye-bye.
Thank you. Thank you very much, Pika. Thanks a lot.
Thank
you.
Anish.
So now we will start some of the other oral presentations. Am I audible, Dr. Bedi?
Yes, yes.
Okay, so now we will start in few minutes the next oral presentation by the our participants. So please wait for another 2 minutes and then we will start the fourth oral presentation. Please wait.
Yeah. चालू कर रहे हैं। हाँ? पता नहीं क्या शुरू कर रहे हैं। बच्चों की वहाँ ओवरसीज वाले एडवाइजरी। हाँ तुम भी भरो और पीजी वाले ही भरेंगे। नहीं कंपल्सरी नहीं है, जाना चाहो मतलब वैसे कंपल्सरी नहीं है।
Okay, we are going ahead with our presentation, oral presentation, before our next speaker get ready. So we have in line our next speaker, Shivachandran. Shivachandran, are you there?
Sir, am I audible, sir? Yes, sir. Yes, sir.
You are audible. Shiva, we will give you 5 minutes for
sir.
presentation. Please stick to the time limit, and then it will be followed by a few questions from evaluators. Okay.
Sir, am I audible, sir?
Yeah, you are audible.
Okay, sir. Thank you,
Are
sir.
you ready?
I am ready, sir.
Okay, your time starts now.
Good morning to all. I am Dr. R. Sivachandran, an MBSc scholar from Department of EPH, Puducherry. And I'm going to talk on detection of Listeria monocytogenes and enterotoxin producing bacillus series group isolates in milk and milk products sold in Pondicherry. These are the objectives of my study: is isolation and screening of virulence gene, assessment of biofilm formation and determination of antibiogram of Listeria monocytogenes and bacillus series from raw milk, pasteurized milk and ice creams which are being sold in Puducherry. And Palcom agar was used as a selective agar for Listeria monocytogenes, and the suspected colonies were subjected for a biochemical test for further confirmation, and DNA was isolated and was screened for the presence of virulent gene HLY-A by PCR, and this is a primer sequence used for the target gene, followed by the PCR mixer, and this is a PCR condition which was used. And then coming to the isolation of bacillus series, the selective agar, Pemba agar was used as a selective agar, and the suspected colonies were subjected for biochemical test for further confirmation, and DNA was separated and examined for GYRB gene, which is specific for bacillus series, and enterotoxigenicity was tested by the presence of other genes. And these are the target genes and the corresponding primer which is being used. This is the reaction mixture for bacillus series, followed by the PCR conditions. Then coming to the biofilm production assay was done using 0.25% safranin, and the results were classified as strong, moderate, weak, and negative. Then coming to the antibiogram, 16 antibiotic discs was used to find the drug susceptibility of Listeria and bacillus series as per CLSI standards. Then coming to the results and discussion, 4 raw milk sample and 1 ice cream sample was positive for Listeria monocytogenes by culture and biochemical test, which is a matter of public health concern and warrants regulatory mechanism to be put in place to reduce the contamination, which was confirmed by PCR by HLY-A gene at 4456 base pair. And this is isolation of Listeria on Palcom agar. Then coming to the bacillus series, 32 raw milk samples, 9 pasteurized milk, and 23 ice cream samples were positive for bacillus series. Although the minimal infective dose for producing diarrhea ranges between 10 to the power 5 to 10 to the power 7 for bacillus series, their level in our present study was completely safe, but there may be they can multiply even when there is any storage temperature fluctuations. And this is the bacillus series group isolates on Pemba agar. Peacock blue colored colonies were observed, which was confirmed by PCR by using GYRB gene at 475 base pair, and 4 different enterotoxigenic genes at 4 different base pairs were seen in multiplex PCR. This is the percentage of various samples containing the enterotoxigenic genes of bacillus series. Then in vitro pathogenicity test was conducted, and 64 bacillus series isolates produced beta hemolysis on 5% sheep blood agar, followed by the biofilm production assay by tube method. Safranin stain was used, and the results were classified as negative, weak, moderate, and strong. And Listeria monocytogenes isolates were from moderate to weak biofilm producers, which is of public health concern, since the biofilms can be more resistant to disinfectants, whereas bacillus series isolates in the present study were a majority of the baseless series isolates were biofilm formers, which may be responsible for the recurrent contamination and spoilage of dairy products, and they form adhesive capacity along on various substrates, including stainless steel, which is being used in the food processing lines. Then coming to the antibiogram of Listeria monocytogenes, the isolates in the present study showed an increased resistance to Genta, vancomycin, and penicillin G, which are commonly used in the treatment of Listeria infection, which is a matter of public health importance. Whereas the Bacillus series isolates, it was found that they were sensitive to ciprofloxacin, gentamicin, vancomycin, streptomycin, and norfloxacin. And many of the Bacillus series isolates in the present study were multi-drug resistance, which is of public health concern. And these are the conclusions of the study, which it is evident that there is a need for the proper hygienic measures before and after collection of milk, proper processing of milk and dairy products, and proper storage to reduce the food pathogens. And there is a severe need for the surveillance and action to reduce the contamination of Listeria monocytogenes and Bacillus series group in milk and milk products in Puducherry. Thank you.
All right, thank you, Shivachandra.
Yes, sir.
Shivachandran, did you do the enterotoxin detection also?
Yes, sir.
And what were the serotypes you found for Listeria monocytogenes from samples? Predominant serotypes.
Sir, we didn't go for serotyping, sir.
No? Okay. Any other question, please?
Hello.
Yes, sir.
Yes, Shivachandran.
Yes, sir.
Have you performed genotypic method of biofilm production? Genotypic confirmation.
Genotypic.
Right?
Yes, sir. We went for actually we species specific only, sir. We just went for
no, for biofilm production. I want to ask for biofilm production. Have you confirmed by genotypic method?
No, sir. Only by phenotypic, sir. Okay. Phenotypic.
And second question is, what was the reason of presence of Bacillus cereus in pasteurized milk? Because pasteurized milk is it is supposed to be, it will not contain any pathogenic organism. So what was the reason?
Sir, it may be due to that storage temperature fluctuations, sir. Some post pasteurization contamination may be there.
Okay. Okay.
All right. Thank you, Shivachandran.
Okay. Thank you, sir.
Let us move ahead with our next presentation, and it is by, I think, Senthil Kumar. Do we have? Sir. All right. Senthil Kumar, you can start sharing your presentation, and you are allotted 5 minutes for your presentation, and followed by 1 minute of discussion. 1 minute of discussion. Please restrict your time.
Is it visible, sir?
Yes, we can see that.
So my presentation is on rabbit and visual diagnosis of canine leptospirosis by recombinant polymerase amplification assay. It is like leptospirosis is a re-emerging zoonotic disease. Mainly the dogs are the reservoir source of the infection to the human being. Leptospirosis immediately serological test, microscopic application test is in our mind, but it will antibody will be appears only after 5 to 7 days of period. But the early diagnosis of the disease is required for prevent the disease in animal and also transmission of the disease from to other animals. So molecular methods are nowadays used for confirmatory diagnosis. Molecular methods PCR assay is commonly used, but it's an alternative method. We have tried this recombinant polymerase amplification assay. It is an enzymatic amplification of the DNA target. There is no requirement of the thermal cycler, denaturation of the target sample, and also it is a very short duration of time. It is mostly less than 30 minutes only it requires. So we have developed this assay for diagnosis of the leptospirosis in canine infection. So first we have targeted the LIPL32 gene. It is one of the pathogenic gene. It is highly expressed during the infection. So we have developed the primers. With that primers, we have first screened all the serovars. It is having efficiency to amplify all the serovars. Then the same primer was used for the recombinant polymerase assay. The twist DX, we have obtained the reagents. With the reagents, we have added the primers and the buffers, and we mixed the reagent solution. Then we have added the magnesium acetate and the lead. Then we are spinning that one, allow for reaction mixer. After that one, we have incubated at the water bath at 39 degrees centigrade for 25 minutes. Then we have examined the amplified product with the across gel electrophoresis. But here we have seen the non-amplification methods. So then we have optimized the critical parameters. The magnesium acetate concentration is very, very important because it is starting the enzyme activation, and that time period we have optimized. So with this too, we have optimized the clinical condition. So in the optimized clinical condition, magnesium acetate with 2.8 millimole
Senthil, just stop for a moment. We have some problem here. Can you start your presentation again from where you were there on some slide, and yeah, we can begin from there itself. Senthil, can you hear us? Senthil, can you start sharing your slide again? कर रखा है इसमें. I think Senthil, you are muted. Senthil. Senthil, please unmute yourself.
Are you able to speak, sir? Sir?
Senthil, we were unable to hear because you are muted. So you have to, yeah, you have to start your presentation from, slide where you were showing. Can you show your slide again? Then we can start your time.
So this analytical assay, sir?
Start sharing your slide. Start sharing your slide, please.
Go back.
Yes, sir. Is slide okay, sir?
Yeah, you can.
So to analyze the sensitivity of the RP assay, we have compared this RP assay with the PCR assay and real-time PCR. The PCR assay it is able to detect up to 100 organisms. The RP assay is able to detect up to 100 organisms. RP able to detect. The specificity of the assay, when we have found out, we are not able to find any of the other genomic DNA. That for the making the diagnosis of the efficiency of the RP assay, we have taken 98 samples and we have compared with the PCR assay as a standard one. This RP assay shows the sensitivity of 93.1% and the specificity of 90.1%. But every time we are going for the RP assay and we have find the finalized product with the gel electrophoresis assay. To overcome this one, to find out the amplified product with the naked eye, we have tried this method. The amplified product we have added the cyber green dye. This is a new method we have find out that one already some pathogens they have tried. So we have adapted this one also here. The amplified product we have added the 2 microliter of cyber green dye. The cyber green dye at a different concentration we have prepared 1000x to 62.5. When we are adding this dye, it is able to produce a green color appearance. The normal negative one, it is appears as a orange color one. So here, then we have to find out the concentration of the DNA, how much concentration in the initial one. Then we have find out a different concentration of DNA used as a template, 100 nanogram, 10 nanogram, 1 nanogram. When we are using up to 10 nanogram, we are able to find out the fluorescence color when it is the naked eye picture. So small difference is observable on the naked eye between the NTC and the sample amplified product. But the same thing when we are seeing under the blue LED light, we can clearly visible the color changes. And also we have tried the different processing method. One process method we have used the unheated product, the amplified product we have heated and add the dye. And another thing without heating we have added. And another one we have purified the product and we have tried the dye. So, but all the three things we are having the same type of picture only. So this method can be useful for reduction of the amplified product in the sample. The conclusion, this method can be used for detection of the canine leptospiral DNA in a clinical sample, simply amplifying the DNA with the recombinase polymerase enzyme without any thermal cycler within a short period of time. It can be applied for the diagnosis of the canine leptospirosis at the field level where the sources are limited conditions. So it can be applied as a point of care test in the field condition. This project is funded by the DPT Government of India under the DPT GADVASU Canine Research Center Network project. Thank you for the DPT for funding this project. Thank you, sir.
Thank you, Senthil. What was the detection limit of the test? What was the detection limit?
Sir, it is up to 100 organisms we are able to detect, sir. We are compared with the PCR.
What about PCR? How much PCR? How much PCR?
PCR also we are able to detect up to. This RT-PCR it is up to 10 organisms it is able to detect, sir. But PCR it is 100 organisms we can detect. But this is compared to RT-PCR, this less sensitivity only, sir.
Has it got any sort of similarity with LAMP?
But compared to the cost and other things, it can be reagent cost. So we have not compared with this LAMP, sir. Human number of primers are required, but here only one primary is required. So we have adopted this one.
Okay, thank you. Any other question, please? Evaluator 2, do you have any question, please?
Okay. Yes, Dr. Senthil?
Yes, sir, please, sir.
can we amplify the RNA from this method, RP method, RNA?
Yes, sir. Yes, yes, sir. Yes, sir. RNA also we can do, sir. PPR, they have done this reduction of the PPR in the samples. They have done the RPM method. They adopted, sir. Published papers are there, sir.
So in laboratory, what we can use in routine or we can. So there are some special things are required, or how much cost is required?
Sir, no special training is required, sir. Only problem we have to get the enzymes and other things because the Twitch DX, they are having the reagents. Only we have to purchase with them. They are the monopoly in the market. This is the limitation in this condition.
Okay.
All right. Thank you, Dr. Senthil. You can exit your screen share.
Okay, thank you, sir. Thank
Okay,
you,
Dr. Rajesh, you can just take over.
Dr. Sir, just give me a second. I want to share. नहीं Dr. Sir. ये वो उन्होंने बंद कर दिया।
Vijay, you have to wait for some.
Okay, sir. Yes, sir. Okay, sure.
Rajneesh.
So now we are having our next speaker, Dr. Yashpal Singh Malik, Dean, College of Animal Biotechnology, GADVASU, Ludhiana. Dr. Malik, he has served as ICAR National Fellow at the premier veterinary institute of the country, IVRI. He had his postdoctorate in molecular virology at University of Minnesota, USA. He is member of several international committees. One of those is International Committee on Taxonomy of Viruses on Birna viridi and Pico Birna viridi Study Group, and a managing committee member of World Society for Virology, USA. He is a recipient of several prestigious awards, including ICAR Jawaharlal Nehru Award. He has developed several technologies and diagnostic kits and has 2 national patents to his credit. He authored 7 books, published around more than 200 scientific research articles, and he is a Secretary General and Secretary in the World Society for Virology. We are very fortunate that Dr. Malik has accepted our invitation to speak on this event. We are very fortunate, and we welcome you, sir. And now over to Dr. Malik. Thank you very much, sir.
Thank you very much, Dr. Rajneesh, for such good words. And at the outset, I first greet everyone who is attending this event on World Zoonoses Day. As told in the inaugural session, that we are celebrating this for the awareness on the zoonotic diseases, and it started from 6th July 1885 with honoring the Louis Pasteur first vaccine for combating breaking the zoonotic diseases. And the theme of this year 2021 exactly reflects that let's break the chain of transmission of zoonoses. That is what is required. And if we go with the number of news, if you, in the last 1 decade, if we scroll down the dates, we could see that number of diseases are coming in the animals, in the humans, and some of them are linked together, and we call them as zoonoses. We are wrapped up. This whole world is facing some of the big infectious problems, including the food security, food safety, antimicrobial resistance, which is being discussed at several platforms, the emerging infectious diseases. And for me, what I can see that all are interlinked. If there is some problem at one corner, it goes to the another one very quickly. So infectious diseases are harming. And if we can go with that left side picture that shows that nearly 100 years back, we suffered with one pandemic of the flu. And again, a century later, we got COVID pandemic. So viruses were 100 years back also, and currently we are facing that. So this is a big crisis world over about the emergence of number of viral infections. The diseases are common in animals, and some of them what have occurred in last just 1 year. There are many which are affecting the Indian livestock, and they have been reported by a number of workers from different parts of the country. And some of them are even transboundary nature. They are crossing the boundaries, and particularly what we are having the porous borders towards the northeastern states. Number of countries are surrounding that part, and many of the diseases, including some of the porcine diseases, just like African swine fever that appeared last year, and more than 5,000 pigs died in number of outbreaks that occurred in that period. And Congo hemorrhagic fever, that is again, that was associated with the sheep, and it is also zoonotic. So several zoonotic, emerging, re-emerging, and transboundary viral infections are spreading, and not only in India, our surrounding countries. So we must be prepared for tackling all of these conditions. There are several factors which have been identified which could be the cause of that fast spread, and that one is globalization. Everything is changing. The way of our lifestyle, that is changing. The population of vectors, that is uncontrolled in many of the conditions leading to the spread of infections. Then bat is one common problem that has been considered in past and leading to emergence of several infectious diseases, and particularly harboring the viruses. I will discuss some of them, including the COVID, that is the current problem throughout the world. Then there is a movement of the animals, and including the migratory birds. Several studies have shown that movement with the birds crossing different continents, they are carrying some of the important viruses. And the areas, zones which were free from that infection, they are suffering badly with them. And there is also movement of the livestock products because of the food security. The people are having trade of the several meat livestock products. And that could be one of the reasons. If the checks are not proper, certification is not proper, then there could be chances of transfer of some of the infectious diseases through that. So we are not certain any time that which infection is going to be the next one, and how much harm it will lead to the country or the continent or the whole world. The problem which arisen in late 2019, that COVID, within 3-4 months, it became the pandemic. So we cannot say that which disease will behave in which manner and how fast it will spread to different parts of the world. And what the studies are confirming, that majority of the infections which are being identified by the researcher, different laboratories, majority of them are finding the classification in the zoonotic category. And animals are playing very important role in that as a reservoir, and some of them are becoming the transboundary infections. The forests are burning, and some of the times we are hearing that we are not bothered because we are safe, but we are exactly not safe. If there is no forest, there is movement of the animals living there, and there could be conflicts of the space, sharing of the space, and there will be more chances of spread of the infection. So that is one common. So this is 3 strata where intermixing of the infections occurs. One is the wildlife, then second is the domestic animals, then humans are not safe if these 2 strata are not safe. Anytime spillover is possible. So that we covered in one of the book that was published by Springer on emerging and transboundary animal viruses, and it got a very good citations in the Scopus and other indexing services. Then it is well said by one of the Nobel laureate that from British biologist Peter Medawer, that viruses are not good. They are bad news every time, and they are simply wrapped in a protein. So that is true every time. They are very tiny, and normally we call them as nanobombs. They spread too quickly and destroy the whole community or the villages, and that has been seen in the case of smallpox also. Rinderpest that affected the animals called the cattle plague, goat that affected by the pestis virus, that is the PPR, that also known as the goat plague. So viruses spreads very fast and becomes the global threat. There are numerous viruses which have been identified in very late, even in last decade, if we say, and still many are on the side that we are with the help of the technology that is growing on metagenomics, transcriptomics, we are identifying numerous new viruses. And that's why we call that viruses are going viral and they are spreading too fast. And this is one timeline that shows that how quickly the viruses are emerging. Within a short period of every 1 decade, we are getting a new virus. If we go with the past Hanta virus, Ebola virus, then several influenza viruses like bird flu that appeared in Hong Kong, later the swine flu, avian flu, every time it is hitting the number of countries in Asian parts. Nipah that appeared in 1998 destroyed the pig industry badly, habitats, reservoirs are the bats, and affects the humans also. Then the series of coronaviruses came, including the first SARS, followed by the MERS, and then the one we are all suffering with, that is popularly we call the COVID. So these zoonotic infections like Nipah, we all know that fruit bats are playing important role going to the pigs as amplifier, and then we are having the infection in the humans. Hendra is another that destroyed number of animals and affected the humans in Australia with a very high mortality. Nipah in the Malaysia and Singapore has led to the nearly 40% mortality, and it has affected other countries, including India also. This is about Zika. We cannot forget, few years back, this was a major problem in the world. It started from the western part of Africa, and then it spread to a number of the countries very quickly, but we were fortunate that we had never came across any such problem here, but we must be prepared that it may be the future. In any way, it can affect all of us. So there are number of emerging respiratory outbreaks which have occurred in the past, including the enterovirus, adenovirus, SARS-CoV, MERS, then the SARS-CoV, and thousands and thousands of the people affected in the past, and now the census has crossed the millions. We cannot even not in a good situation to conclude that it has affected this much number of the population of the human beings. And the important thing to say here about the corona, it also affects the animals also. It has gone from the humans to animals, and now it has been established in the mink farms in the Netherlands that it is going from the animals to animals, and then from animals back to the humans. So this is a major concern, respiratory infections, and maybe because of number of viruses. And presently we all are just facing the crisis of corona. This is one bad picture I must consider, that whole world is experiencing the emerging and re-emerging viruses. And the dots are indicative of the new, the red dots, these are the new viruses which are affecting the world, including the Marburg, Ebola, Zika, plague. So many infections have come and almost affected the whole world. This is one part that normally we miss. That is the predicting the zoonosis from where it is occurring, though now emphasis is also on going with the wildlife, and there is shrinking linkages. The borderline is shrinking because of the deforestation, urbanization, and that is one major cause behind the emergence of these zoonotic infections. People are in trade of the wild animals, and even the fruit bats, the sap that is affected by the fruit bats, that also lead to the infection. This is one story that started almost 1.5 years back at the Hunan seafood market, from where we got that the COVID, first pneumonic, unidentified pneumonia cases. And when it was traced that epidemiologically, some of the cases were traced to the Hunan seafood market, where there is a live animal market, and a number of animals are sold in legal or illegal ways there. And that was the one point of origin of the COVID. And several other animals are also considered to be the cause of this COVID emergence. This I am sharing one slide. That is one good work that has been done by the research team. I am also part of that. This is with my students, my research fellows, my senior colleagues, Dr. Kuldeep Dama from IVRI, Dr. R.K. Singh, and several other known personalities in virology. And we could publish, we could analyze the data which started in February. We started publishing on coronavirus, compiling this all about the corona and making people aware on the corona, going with the number of analysis of the genome sequences. And some of the things I will discuss with you, including that whether canine and feline are susceptible for this and how close they are really with the coronaviruses of the humans. This is the simple classification of the coronaviruses. They are classified in 4 genus, but as a human being, we are more concerned with the alpha and beta coronaviruses. And nowadays we're discussing only beta coronaviruses, that is having the SARS, sarbecovirus, marbecovirus, and ambecovirus, which are the subgenus of the beta coronavirus. But there are other genus also which cannot be ignored. We have to go with more molecular epidemiological studies, going with the screening of number of species about the coronaviruses and other important infections. Bat is a common culprit in all of these. This is just one slide that shows that how many persons were affected with a heavy mortality in the past that was noticed with the MERS, but the total number of cases were very limited. Number of the countries affected were only 21. But if we go with the latest one, COVID-19, we can see that the almost whole world has suffered with this. There is almost no country which is left with any impact of it. It may be the direct impact of the loss of human being, or it may be because of the trade or associated factors. So these are some of the coronaviruses which affected the whole world. Whenever we analyze the genomic data, when we go to that, it is a very plastic genome of the coronavirus. It goes on mutating. And I will go with some of the slides which will clearly mention that how viruses are evolving too fast. This is a RNA virus. No doubt RNA viruses are more prone for the mutations. That's why we are getting so many mutants in very short period of time. So this genome is very plastic and it goes on mutating. We analyze the whole genome of complete coronaviruses which are circulating in animals and humans. We found that bat coronavirus is very close with the SARS coronavirus. This was first to be published in early February 2021, when there were very limited number of the sequences which were available. Now the database is too big. Thousands of the whole genome of coronaviruses are available, but still the clarity is not there that it is very close to pangolin. It is very close to the bat coronaviruses. A number of bats are there. The coronavirus of bovine, that also has the linkages with the human one. Human coronavirus, that one strain comes in that category also. Marbeco, that was the MERS coronavirus that came through camels. It also affects the humans. So there are beta coronaviruses which are very diverse, but they are going to infect the humans. And two, Hebeco and Nobeco coronaviruses, they are affecting only the birds, but we are not having any incidents that whether they can go to the humans, and that definitely requires the exhaustive surveillance studies. This is how it spread from the bats through civet, intermediate host, dormitory camels, then snakes. So many animals were speculated that they could lead to the infection in the humans. So almost 10 years is the phase when every time the coronavirus is mutating from SARS to MERS and MERS to SARS-CoV-2. We don't know when we can expect the next one. So that's why we need a preparedness, we must be planned that what could be the next coronavirus. So we are learning some of the things from animal coronaviruses, what has been studied in past, because animal coronaviruses, if we go with the published literature, the first coronavirus in animals was identified in 1930. Nearly almost 80 to 90 years earlier than the coronavirus what is affecting us. So many of the things we can learn from the animal coronaviruses about the pathogenesis, diagnostics, vaccine. Some of the vaccines are available for the animal coronaviruses. So there are some advances which have been done in animal coronaviruses, including the animal models that have been used for study of the SARS-CoV-2 virus also. This is one epidemiological data that shows that almost animal coronaviruses are prevailing almost in all parts of the world. There are different animal coronaviruses, which are alpha corona and beta corona, just like beta corona is affecting the humans. And we have the feline, which affects the cats. Then canine respiratory coronavirus, which is almost resembling in the symptoms in the what we are observing in the SARS-CoV-2. And it is said that canine coronavirus is also very close, and it is OC43 strain of the human coronavirus. So there is a common ancestor of the canine respiratory coronaviruses, and we are delineating that how closeness is between these two viruses. So canine corona, canine enteric corona, they have some resemblance with other animal coronaviruses, and they emerged from other source, other host in the past. This is one study that we performed analyzing the complete genome from different animals and the cats and canine, and some of the viruses which were identified in the mink, lion, and the pangolin. And it clearly showed that the canine coronavirus is and the feline, that is cat coronavirus, they are not very close to the SARS-CoV-2. They are far away and less than 50% they are resembling. But some of the viruses which were isolated, which were identified from the mink, lion, and pangolin, they were almost 99% resembling with the SARS-CoV-2. So we can say that virus which is staying in the humans can stay easily in the animals. And this is the time that we all must come together to work, because we cannot work separately in separate institutes. Ultimately, we have to work for the human community. This is one example that canine coronavirus that was just a mild infection in 2006. And now it became a very virulent pantropic coronavirus because of the mutations in certain spike gene and other related genes of this virus. Just like that, one virus is transmissible gastroenteritis virus and porcine respiratory coronavirus. Both are same viruses, but because of the deletion of 600 nucleotide, a small area in the S gene, a new virus was formed, and that changed the predilection site. Earlier was affecting the enteric, and now it is the respiratory. So viruses in the coronavirus, they are changing very fast, leading to the emergence of new and new viruses. This is one that slide that shows that bovine coronaviruses also shows some genotypic importance, and they are close to the human coronavirus OC43 strain. And similar way, there are some coronaviruses from alpaca. They are also having genotypic implications. Spillover is a big phenomena that is occurring throughout the world. Viruses are jumping the species from one animal to the humans or one animal to the other one. And this is one statement that has been conveyed by Zhengli Xi. She is known as the Bat Woman of China. And she is working on bat coronaviruses. And she stated that this is not the end. This is just one start. And many more viruses are there which are coming from the bats. So what we must be prepared and for working on this concept of bats ecology, behavior of that, and transmission of the diseases from this species. some of the work that we have carried out, this is not only the coronaviruses which are important, there are several other viruses which are playing very important role, including one that leads to diarrhea. That is in the animals that leads to more than 50% of the cases in diarrhea or the mortality are because of this enteric infection. So we studied some of the infections which are zoonotic and extended our work on the particular one interesting pathogen that is very popular in the humans, but not least I can consider for the animals. It is as important as in case of the humans. We have vaccine for the humans and we are working that for the animals also for the coronavirus, for the rotavirus. So that is also that affect the humans and animals, and from animals it is going back to the humans. This is number of, again, this virus is very mutating virus. Number of types of rotaviruses are circulating, but one which is very common in human and animals is the rotavirus A. And one study that we conducted on the rotavirus C that affects the porcine, and we conducted it in different populated parts of the pigs in the country, and we found that the viruses which we isolated, this is the rotavirus C, and majority of them were matching with the genome of the humans. So there was indication that this porcine rotavirus C is also having a relationship with the human rotavirus C. So that we extended the study, and we first time reported that this unexpected detection is carrying the human origin BP6 gene of the rotavirus C. Similarly, in the bovine also we studied number of the viruses. In one of the project that I completed on the under National Fellowship of the ICR, we screened thousands of the viruses of the bovine origin, and we found that some of the bovine viruses were resembling with the exactly which is in the humans. So that also we concluded that both species hosts are exchanging the virus between these two among the rotaviruses also. So we cannot omit the rotaviruses animals, and we must focus in both species to eliminate this problem. And to eliminate this problem, the only one answer that appears and the whole world is focusing, and a number of agencies are coming forward. All the countries throughout the world, they are working on developing the One Health concept. We have all branches of the medical, veterinary, and the environment. We must come together to fight the emerging problems. Some of the times it is said that zoonosis is a problem of only animals. Animals are the threat to the humans, but reverse is very much possible. So it must be always in our mind whenever we are planning, whenever we are working in the animal One Health, that both sides we have to see of the coin. So this is the only approach that can save the whole world. If we, and this is one leads which have been taken by the SAARC countries, that we almost all SAARC countries have established their policies in One Health, including Bangladesh, Nepal, Bhutan, Sri Lanka, Afghanistan, and India is also taking lead. We also have the National Institute of One Health. Nagpur is leading in that. Veterinary colleges are coming forward, including the one that we are having at GADVASU. We have the One Health Center, and leading lead in the country and guiding and giving all the advice to the government. Some of the leads what we have taken in the past, we are advising the policymakers, nodal points we are establishing, going to the grassroots level, advising the people, and we need to integrate the One Health approach with the national planning, and that is the best thing what looks after. If we have to think about the human health, we have to think about the animal health alternatively. We have to work in toto. So this is one lead that has been taken as a One Health concept in the country. GADVASU is having one SARS-CoV-2 testing laboratory, and we have tested more than 3.5 lakh samples from the humans. And we have been recognized by UNICEF and ICMR as the high priority lab and interested with this establishment of more sophisticated laboratory. We have received more instrument under this laboratory. So this is one lead. Being the organization from veterinary side, we are working for the human health as well. So some of the things what we need for preventing the future pandemics, to avoid that, to convert epidemics to pandemics, we have to work together. And there are many species which must be targeted for screening. The whole virome, whole microbiome is the need of the hour. We have to study that as a comprehensive so that we can identify the emerging viruses. Several studies are showing that viruses are emerging. Continuously, they are evolving, and that is the lead by the metagenomic analysis. We have to go with the extending the surveillance programs in difficult areas, going with the large exhaustive surveillance in the animal population, bats, and unidentified natural reservoirs. So we have to go with a ban on the illegal trade of the wet markets. Animal trade must be restricted and regulated. And this is one that we can sleep very well only when we come under the umbrella of One Health. So this, I will end this talk with that one mantra of the COVID, that we have to stay safe, stay healthy, take care of everyone. Thank you very much. Thanks to Dr. Bedi and the Center for One Health giving me opportunity to be with you all, and all the learned speakers who have given their speech in the past and forthcoming day event. Thank you very much, each and all.
We have already started receiving messages from the audience. Thank you very much. You have presented huge information and tried to pack that in a single presentation about zoonotic viruses. We are very thankful to you, sir, for accepting our invitation and speaking on this topic. And there is a few questions. One of those is, you have already explained much of that, is about the role of, possible role of animals in ongoing COVID pandemic. And then another question is, how bad is this COVID virus for pet animals? So,
very good to have some questions, and almost both are interlinked. It is important for animals also, and it has been realized, if you go with the news and some of the publications also, that animals are being found as susceptible, having the receptors which are having good efficiency to bind with the virus ligands. But this is still, I consider, is a point of investigation, research, and people are working throughout the world. Even in India also, it has been taken as one of the priority area by ICAR, by ICMR, launching some of the platforms to go with the surveillance of animals, so that we can conclude that how they can harbor this virus or how susceptible these animal species, particularly the companion animals, are to the public or the human population. Whatever the information has been received as of now, that is still limited. We must not conclude based on this limited information. More information is needed. But mink, the study that has been published in the Science Journal, that clearly indicates that it perpetuated in the mink forms, and from there it has gone back to the humans also. So we must be cautious until we are not revealing or not coming to the conclusion that how close the animals are for this coronavirus too. We must be safe, and we must follow all the guidelines issued by the government time to time. And that is the one best way to keep yourself and your neighbors safe with this ongoing problem of the COVID.
I would like to thank sharing his valuable time and speaking on viral diseases. Thank you very much, sir. We are fortunate to have you in this meeting and hope for further
Thank you so much. Thank you so much, each and everyone. Wish you all again a very happy greetings on this World Zoonoses Day. Keep yourself safe. Thank you very much.
Thank you, sir, for sparing the valuable time out of your very, very busy schedule. And we feel honored that you are today with us and you have given a very detailed insight about the viral zoonotic infections. Thank you so much, sir.
Okay, Vijay.
Yes, sir.
Yes. Okay, we are ready for your presentation.
Yes, sir.
So you know that we have time limit of 5 minutes and 1 minute for discussion.
Yes, sir.
Please restrict to that. If you're ready, you can start sharing your slide.
Okay, sir.
ये होल्ड करना अभी रुको.
Okay, sir.
Vijay, we are ready. You can start.
Is the screen visible, sir?
Yes, yes. Yes.
Okay, sir. Thank you, sir. Good morning, good afternoon to all of you. Myself Vijay Sharma, PhD scholar, Center for One Health, GADVASU, Ludhiana, going to deliver my oral presentation regarding the topic prevalence of brucellosis among goats and associated humans in and around border areas of the Jammu, J&K. Actually, the study was carried out during my MBSc degree program in the period 2016-17 in the division of BPS COVS Jammu. The significance of the study is that there is so much movable goat population in the border areas of the Jammu region that moves from the lowland to the highland during the summer and high to low during the winter. And as we know that caprine brucellosis that is caused by the Brucella melitensis is the most zoonotic among the genus Brucella, may spread from the one area to the another and constitutes a serious public health hazard among the J&K population. The risk group for the caprine brucellosis includes the dairy farmers, veterinarians, abattoir workers, lab workers, and if someone is infected, may show the symptoms such as the weakness, fever, joint pain, orchitis, neurological symptoms also. The disease has been managed successfully in the developed countries by means of the efficient diagnosis, vaccination, test and slaughter policy, but it still remains a problem in the India, including Jammu region of the J&K. Therefore, a prevalence study was conducted among the goats and associated humans in and around border areas of the Jammu region by means of the various tests such as the RBPT, STAT, and indirect ELISA. Now, regarding the material methods, a total of 425 samples, including the 350 goats and 75 humans, were randomly collected from the different border areas of the Jammu region and tested by the RBPT, STAT, and indirect ELISA. Now, this is the distribution of the samples collected from the different border areas of the Jammu region. Now, regarding the results, First of all, overall seroprevalence of the brucellosis in the goats and the humans. The overall seroprevalence was 1.14% in case of goats and 1.33% in case of humans. And regarding the test-wise seroprevalence, maximum number of samples were found positive by the indirect ELISA, then by the STAT, and then by the RBPT in case of goats, and maximum in case of humans by the ELISA, then RBPT, and then STAT. This is the presence of anti-brucella antibodies in the different serological test combinations in the goats and the humans. And those samples found positive in all the tests, that is RBPT, STAT, and indirect ELISA, are considered for the overall seroprevalence, both in goats and humans. And actually, this is a type of serial testing, a type of the multiple testing. This is the images showing the negative and the positive results for the brucellosis. No, this is the seroprevalence of the brucellosis among the goats in the different border areas of the Jammu region, and we found that Vijaypur Samba region of the Jammu show the maximum positivity as compared to the other regions. And this is the seroprevalence of the brucellosis among the humans in different places of the Jammu. And we found that Kanachak sector of the Akhnur region of the Jammu found the maximum samples positive for the brucellosis. No, this is the age-wise seroprevalence of the brucellosis in the goats, and we found that 3 to 5 year age group of the goats were found more positive for brucellosis as compared to the other age groups. And this is the age-wise seroprevalence of the brucellosis in case of humans. And we found that middle age group, that is 36 to 50 years, were found more positive for brucellosis as compared to the other age groups. Now, this is a sex-wise seroprevalence in the goats and the humans, and we found that female goats were found more positive for brucellosis, and in case of humans, only male shows the positivity for brucellosis. No, this is the seroprevalence of the brucellosis in the goats in a different rearing system in case of Jammu region, and we found that unorganized sector shows the higher prevalence as compared to the organized sector. And in the end conclusion, the overall seroprevalence of brucellosis was found 1.14% in case of goats and 1.33% in case of humans. The seropositivity orders were in the following order, that is indirect ELISA, STAT, and in the end RBPT for goats, indirect ELISA, RBPT, and STAT in case of humans. The seroprevalence of the brucellosis in goats was higher in the age group of 3 to 5 years. And the reason for is that that age group is the sexually active age group, generally used for the breeding or the mating purpose. And the males of the humans of 36 to 50 years were found more vulnerable for the brucellosis as compared to the other age group. The female of the goats were found more vulnerable to the brucellosis, and the reason for that susceptibility to the brucellosis increases after the sexual maturity, especially with the pregnancy. Then unorganized sector of the goats show the higher prevalence as compared to the organized sector, and this is due to the better managemental practice in case of the organized sector, such as the regular monitoring, screening, segregation, and vaccination of the animals in the organized farms. Among the serological tests, indirect ELISA was found to be the maximum, most sensitive, and detects the maximum number of the positive samples. Then in the end, the disease seems to be the less prevalent among the goats and the humans in the and around the border area of the Jammu region. And further proper vaccination of the animals, optimal control strategies, and surveillance system can reduce the disease in the future. Actually, the study was carried out in the border areas. Acknowledgement. This research was supported by the Dr. D.K. Singh, Principal Scientist, IVRI, Izzatnagar. Thank you.
Thank you, Vijay.
Yes, sir.
What is your recommendation for testing of diagnosis of brucellosis for humans based on your study?
Sir, we found maximum number samples positive by the indirect ELISA, then we RBPT, and then in the end STAT.
So what do you recommend?
Sir,
do you recommend all three tests, or do you recommend a combination test, or do you recommend one?
No,
test?
sir, I recommend all tests, sir. All tests, because according to OIE, not a single test give the perfect diagnosis. We go for the battery of the test. So first of all, we go for the RBPT, then STAT, and then indirect ELISA, and then we get the confirmatory diagnosis.
Any more question, please?
Vijay Sharma?
Yes, sir. Yes, sir.
Can you tell me which vaccine is recommended in India?
Brucellosis in goats.
Sir, Brucella melitensis Rev 1 vaccine that is recommended for the control of brucellosis in India.
Okay, is it used for endemic areas or for all areas?
So actually, it is used for endemic area.
Okay.
Yes, sir.
Okay.
All right, Vijay.
Thank you, sir.
Thank you. You can exit your screen share and.
Thank you, sir.
Thank you very much to all the speakers as well as to the evaluators. Now we are going to start our next session, which is on viral and parasitic zoonosis, and we do have oral presentation in it. And for that, I invite the first speaker, who is Muhammad Mir Hussain, to please share his screen. Sir. Okay, please do share your screen and then we will start.
Yes, sir. This is visible, sir.
Yes.
Good afternoon, everyone.
Just hold on. Muhammad Mir Hussain, just hold on, please.
Yes, ma'am.
Okay, so Muhammad Mir Hussain, you may start. And I think you know it very well that there is a time limit. So we have 5 minutes for each oral presentation, followed by a minute of question answer session. So if you are ready, you can start, please.
Yes, ma'am. Myself, Dr. Mir Hussain. I'm going to present my topic on risk factor analysis for occurrence of Japanese encephalitis in Assam. So Japanese encephalitis is a mosquito-borne viral neurological disease. We all know that 68,000 people are globally died due to in the cycle of transmission. Cycle P acts as the amplification host, and mosquito acts as the transmitter of these viruses. In Assam, maximum number of ZE cases has been reported from Assam in the last decade. In India, about 46.95 of human cases is reported from Assam in 2019. Swine acts as amplification host, and Assam possesses largest number of swine population in the country, and the maximum cases of Assam were reported from the families which engaged with paddy cultivation and swine rearing. So there is no such specific study is conducted to find out the associations of JE occurrence with respect to the swine husbandry practices. So we take an objective by above mentioned gate. We have taken one objective to assess the risk factor of JE with respect to swine husbandry practices in targeted districts of Assam. We have taken 2 districts. This is Borpeta district, this is Lower Assam, and this is Sonitpur, this is Middle Assam. And according to the Ministry of Health and Family Welfare, Borpeta and Sonitpur is the endemic districts of endemic for JE. Now we have taken we have designed one questionnaire, and we have taken 120 number of collected 120 number of questionnaires from the peak farmers of Borpeta and Sonitpur districts. We have collected different risk factor associated with the JE, and also collected the serum and blood sample from those respected swine sample and tested by RT-PCR, real-time PCR, and antibodies by the IgG and IgM ELISA. This is the question which is divided into 4. First is details of the owner. Second is general information, which contains 16 number of explanatory variable, and this is other party's question. And after getting this factor, we enter the data in the Excel sheet, and this statistical analysis is done by our statistical program. There is 16 explanatory variable and outcome variable. This we have collected 120 number of serum and blood sample. We will take positive that sample which come positive by IgG, IgM, or real-time PCR. So univariable and multivariable analysis is done for the all 16 explanatory variable. In the descriptive variable, we have find that from Borpeta 25th number of pea gunners and in Sanipur 33 number of pea gunners is unaware of the JE infection. Even the in past, JE vaccination is not carried out in the Assam, and pig farms are present in the vicinity of 71.67 household, revealing the pig farmers is one of the main occupations of targeted districts. And we have surveyed 120 out of 120 pig barn. Out of that, 78.33 were poor hygienic condition, while waterlogging was found in 56.67 in the farms. And univariable analysis, we have found that month hygienic status of pig and waterlogging are strongly associated with the JE positive, and pig stay with the straw and thatched pig shelter have the larger odds of getting infected with the JE than in the brick sheds, but it is not statistically significant. The distance more than 15 meter, the pig shelter to the owner house is protective effect in the JE infection, but the association is not found significant. In multivariable analysis, we have found that month and waterlogging area is highly odds to have monsoon season is associated with the larger odds have this pig being infected with the JE as compared to the post monsoon season, and waterlogging also have the higher odds to getting infection than the dry area. Now, univariable analysis, you have found month hygienic status of and waterlogging condition is strongly associated with the outcome variable, that is JE infected animal, and it was found that there is no chance of acquiring infections during the monsoon as compared to the post monsoon season. The variation of peak season might be due to the spatio-temporal distribution, variation of density of Culex mosquito, and there is a presence of floody paddy fields in the monsoon season and the breeding place for the Culex mosquito. This is one picture who is showing this is the close associated of the paddy fields and the pig rearing. And this is the wall of a home, and this is the pig shed is very closely near, and they are feeding the pigs in a like scavenging system, and they are it's feeding with one hand. And this is one pig farm I have visited. This very dirty condition. This pig farm situated in a home itself. Its owner house is far 10 meter less than 10 meter. So in multivariable analysis, we have found that monsoon season was associated with the larger odds of pigs being infected with the JE than post-monsoon season, and waterlogging in the area near the pig shelter is associated with the larger odds of being JE infected than compared to the dry area. In epidemiological study, during the several outbreaks of Assam, they have found that the larger water bodies near the Huon valleys were the constant feature of cases, and the most of the JE reported from the families engaged with the paddy cultivation, that is 78.6, and the pig rearing, this is 55.1, the reported cases. In conclusion, I find that in the descriptive study, we have know that this lack of knowledge of regarding JE amongst the pig owners and the vaccination, even the vaccination of this two district is not done. And in univariable analysis, we have found that monsoon season poor hygienic status of pig farmers and waterlogging area is strongly associated with the occurrence of JE infection. And multivariable model, we have found that monsoon season and waterlogging area near the pig shelter is associated with the larger odds of pig being infected with the JE, and the urgent of conduct of awareness program amongst the pig owners along with the vaccination campaign. Thank you.
Okay, thank you, Dr. Hussain. It was a nice presentation.
Thank you, ma'am.
Okay, so my question for you is, what were the results of real-time PCR, and how many pigs were seropositive by ELISA?
Ma'am, actually, by real-time PCR, 37% of animal came positive by real-time PCR, ma'am.
Okay. And what about ELISA? IgM, ELISA, and IgG, both of them.
Yes, ma'am. And IgG by IgM, ELISA, around 12% sample came positive, ma'am.
By which ELISA, IgG or IgM?
Both, ma'am, IgG and IgM.
Same, how could it be? Like,
actually, ma'am, by IgG, ELISA, ma'am, it came around, ma'am, ma'am, 12%, 12.96% came positive by IgM, ma'am.
Okay, and what about IgG?
Ma'am, it's around 11%, ma'am.
And by real-time PCR?
37%, ma'am.
So what were the reasons for a high prevalence by real-time PCR rather than by ELISA?
Ma'am, Ma'am, real-time PCR, it can detect the minimum concentration, minimum viral concentration, ma'am, in a sample. Ma'am, even it is sensitive to, ma'am, viral concentrations around 10 to the, ma'am, 10 copies number, it can sensitive, ma'am.
Okay, which method you used for extraction of RNA from the samples?
Ma'am, conventional trial method, ma'am, RNA extraction method, trial.
So were there some samples which were positive by 2 or more tests?
Yes, ma'am.
And can you explain me the vaccination regime for JE virus in human beings?
Ma'am, vaccination is like SA strain 1442 strain is used for vaccination in case of human.
Which strain?
Ma'am, SA 1442.
Okay. And what is the vaccination regime? I mean, at what age it is done for human beings? For you had a survey, so you might be knowing that for which age group the vaccination is recommended.
Ma'am, actually 12, at least 12 months old, ma'am. 12 months.
And what were the main findings of your survey, the questionnaire part which you conducted in your study? What were some main findings in that?
Ma'am, in questionnaire, by analyzing, I have found that they're rearing the pig in a close proximity with the owner house, and the vaccination schedule is not done, and ma'am, hygienic condition is very bad, ma'am, in the farm and ma'am, nearby area.
And which is the main vector responsible for transmission of this virus in Assam? It's mosquito, but which species?
Ma'am, Culex tritrancus, ma'am. Culex tritrancus.
Okay, thank you. Okay, now over to the second evaluator. Dr. Saab, you may please go ahead with the question.
Basically, evaluator one, I think she has covered most of the things. But Mir, can you tell me that any recommendations you have come up after this US study for the Assam government? आपने इसकी कोई रिकमेंडेशंस इसको स्टडी के बाद कुछ भेजी है असम गवर्नमेंट को, डिपार्टमेंट ऑफ एनिमल हस्बेंड्री असम गवर्नमेंट को, स्पेशली रिगार्डिंग द योर दिस outcome of the...
Sir, one recommendation is that vaccination to children, ma'am.
Can you tell me the cost of vaccination? Do you have any idea about the cost of vaccination? You are not audible, Mir Hussain. Mir Hussain, you are not audible.
Yes. Mir Hussain, can you hear us?
Mir was not audible, ma'am. I was asking him any idea or he knows about the cost of vaccine which is used.
Yeah, Dr. Saab, even we can't hear him. I think he has lost the connection because his slides are also not visible as of now.
Okay, Dr. Saab, I think you have exhaustively asked him. I think it's okay.
So we can move ahead.
Yeah.
That's fine, Dr. Okay. So I invite the next presenter, Dr. Anamika Sahu. So Anamika, are you here?
Yes, ma'am.
Okay. So please share your slides with us.
Yes, ma'am.
So Anamika Sahu will present her talk on development of indigenous IgM ELISA for serodiagnosis of Japanese encephalitis in equines. So we have another JE here, talk on JE. So Anamika, please share. Okay.
Good afternoon to everyone. Myself Anamika Sahu, and my presentation is on development of indigenous IgM ELISA for serodiagnosis of Japanese encephalitis in equines. So as we know that Japanese encephalitis, these are arthropod-borne diseases, and that affects the horses and pigs and humans. Equines are the most commonly affected animal, and mostly the symptoms are in subclinical form. Until now, case fertility has been reported near about 5 to 15% in endemic area, even sometime more than that. So main reason of having high infection of Japanese encephalitis in equines, because these are not vaccinated, and that leads to continuous decrease in equine population. So research gap is non-availability of indigenous IgM ELISA kit for the sero-diagnosis of Japanese encephalitis in equine, and there is limited sero-surveillance of Japanese encephalitis in equines. So by considering the all gap, my first objective is to standardize IgM ELISA for the sero-diagnosis of Japanese encephalitis in equines, to assess seropositivity of Japanese encephalitis virus in equine population of Uttar Pradesh using in-house IgG and IgM ELISA. So material method, the sample size was calculated by using the EP info software, and that was 319 samples. And the sample was calculated, was screened from various states of India like Uttar Pradesh, Maharashtra, so on. Total 621 samples were collected. These are the samples. And the processes are the screening of clones, the bulk production and purification of recombinant protein, and then further development of recombinant protein based on indirect IgM ELISA. And then commercial kit, commercial IgM capture ELISA kit was used to compare with indirect IgM ELISA. Then standardization of IgM capture ELISA, and further indirect IgG ELISA for sero-diagnosis of Japanese encephalitis in equine. Then result and discussion. Screening of clones was done, and expression was best observed on SDS base at 59 kilodalton. And then different elutes of imidazole was used, and dialysis of those elutes were done, and the protein concentration was estimated by Bradford reagent. And the development of indirect IgM ELISA for sero-diagnosis of Japanese encephalitis in equine, the different conjugate dilution and different serum dilution were tried, like 1:5,000, 1:200 dilution, and the highest P/N ratio was obtained, that was a 5.5. And then overall seropositivity was calculated by using indirect IgM ELISA, that was 30.75. And the maximum seropositivity was reported in Himachal Pradesh, that was 41.3%, and Uttar Pradesh it was 29.1%. These are the sample. And the final conclusion of that result is that overall seropositivity of IgM antibody was 30.75, and it was the first study in India where IgM antibodies against Japanese encephalitis virus have been demonstrated in equine serum, and that indicates the active or acute cases of infection. So validation of IgM ELISA was done, where kappa value was calculated. Then commercial IgM ELISA was used to compare the in-house IgM ELISA, where sensitivity and specificity was calculated, that was 100% and 88.5%. Then standardization of IgM capture ELISA, an attempt was made to standardize IgM capture ELISA by using a polyclonal and monoclonal antibody. So these are the different combination of reagent and concentration we tried by using a polyclonal antibody. These are the different combinations. So final conclusion we got, there was no significant difference between the positive and negative sample was observed. That was already confirmed by indirect ELISA. So to check the cross-reactivity, different combination we used. These are the different combinations. So final conclusion was there was a cross-reactivity of anti-equine IgM antibody with a hyperimmune serum and conjugate. So there were other researchers have also observed the similar cross-reactivity by using the polyclonal antibody. So they suggested to use the monoclonal antibody instead of polyclonal antibody. So different combination of reagent were used for developing the IgM capture ELISA by using a monoclonal antibody. These are the different combinations. And so there was a significant difference was observed when we used these combination and the monoclonal antibody. By using the monoclonal antibody, so IgM capture ELISA was standardized. But the difference of OD was near about 2, and the sensitivity of this test was not so good. So further sample were not processed. And screening of samples using recombinant NS1 protein based indirect IgG ELISA. So overall seropositivity was observed 21.41%. These are the sample. Maximum seropositivity was observed in Himachal Pradesh, and in Uttar Pradesh it was 18.94%. So final conclusion was that in 2006 to 2009, overall seropositivity was 10%, but it was increased. According to my study, it was 21.41%. And that indicates it indicates that Japanese encephalitis virus is it increasing its a domain to temperate area like Himachal Pradesh. Seropositivity study in Uttar Pradesh, initially in 2010, the seropositivity was 11.16%, but according to my study, it came 18.94%. That indicates the reason of having high seropositivity may be because of high rice production system or expanding the migratory birds. And a final conclusion is that indirect IgG ELISA is reliable, safer, efficient for the detection of active or acute cases of infection, and it is a first indigenous IgM ELISA kit for the diagnosis of Japanese encephalitis in equines. Overall seropositivity was observed 30.75%, and for serosurveillance study, the positivity was 21.4%. And increase in the seropositivity of Japanese encephalitis virus in Uttar Pradesh, that indicates it can spread in future also, and that leads to high morbidity and mortality in equine population. Thank you.
Thank you, Dr. Anamika. I appreciate your work. You did a good research work. So, but there are some, few questions in my mind. The first one is,
yes, ma'am.
Like, how many sera samples you collected from equines?
Ma'am, 621.
Okay, from different states. Is that so?
Yes, ma'am. Yes, ma'am.
So, do you have? Did you have the CPSCA permission for that? The ethical permission?
No, ma'am.
So, do you think it was required?
No, ma'am. Actually, sample. Actually, sample collection was, I don't think we need permission for.
Did you use some previously collected serum samples, or
no,
you collected?
Sir, no, sir, no, ma'am. It was just when I started my work, then only collected.
So, is this work published somewhere?
Yeah, no, ma'am. No, till now it's not published.
So, any good journal will not accept your work, my dear, without the ethical permission. So, it is mandatory. How could you say it is not mandatory? Even if you want to collect blood sample from a small mouse, then also the ethical permission is compulsory. So whenever you present such talks, it came to my mind, since I could not see anywhere in your material and methods a slide on ethical permission.
Yes, ma'am.
So your research, okay, please keep in mind that. So any good journal will ask for the ethical permission. Secondly, what could be the reasons for more seropositivity in Himachal Pradesh as compared to Uttar Pradesh?
So, ma'am, the reason can be because of high population of equines, and another reason is that ma'am, this one is
the first study, where I could find such comparison, where Uttar Pradesh was lagging behind JE cases than any other state.
Ma'am, in case of equine, we can say.
Okay. So, and which reference kit you used for the comparison of your results? You standardized both IgG and IgM ELISA, is that so?
Yes, ma'am. Yes, ma'am.
Okay, so which IgG and which IgM kit you used as a standard?
No, ma'am, IgM. I did IgM, not IgG. IgG was previously standardized in our lab.
So which kit you used for your?
Ma'am, I used commercial IgM ELISA kit.
Against which species?
What, ma'am?
Against which species? The commercial IG, G or M, whatever. Was it multi-species kit?
No, ma'am. It was only for single species.
Which species?
Ma'am, only for equine. It was only for equines.
So is there any commercial kit on equine available for ELISA against JE?
Yes, ma'am. We can use this one only, no? This one.
Which one?
Ma'am, IG capture
My
ELISA.
question is, since you have listed a gap in your study that there is no commercial kit available for diagnosis of Japanese encephalitis in equines, so which commercial kit you use to compare your in-house kit? That is my simple question.
Ma'am, I said like, ma'am, it was imported. Actually, I don't know exactly the name of.
Did you do this research yourself?
Yes, ma'am.
So didn't you see the label on it? Okay, leave it. Another question is, did you have any known positive sera samples?
Yes, ma'am. There was
Validate your kits, and where were they procured from? And how could you say they were positive?
Actually, ma'am, I was having it near about 24 samples. They were positive. And ma'am, we have confirmed it by the commercial ELISA kit. That is the capture ELISA. It was based on a capture ELISA. And we confirmed it by indirect IgM ELISA.
Okay. Okay. The last question is, what would be the effective cost per sample for your kit?
Ma'am, per sample. Ma'am,
per sample, what will be the cost of testing one sample? If I'm an equine owner, how much you will charge from me for testing of JE by your in-house kit?
Ma'am, for near about for 70 to 80 sample, we can process it by our kit, and the cost will be within it 10,000. It will be. And if we will take the imported kit, the cost will be near about more than 50,000.
Thank you, Dr. Namika. It was a good work. Keep up the good work, but include the suggestions,
Yes, ma'am.
Which I recommended. So over to the second evaluator. Doctor, please.
Anamika, what technique you have applied for the collection of samples from different parts of India?
Sir, it was a convenient sampling plan.
Convenient sampling plan?
Yes, sir.
Okay. Okay. Thank you very much. I think most of the questions were asked by the evaluator. Okay. Thank you.
Okay. Thank you, Dr. Sahu.
Thank you, ma'am.
We move on to the next presenter, Dr. Gotapu Chaitanya.
Good afternoon, ma'am.
Very good afternoon. So, Dr. Sahu, please stop sharing your screen so that Dr. Chaitanya can share his screen with us. Okay. Dr. Chaitanya, you may please start sharing, and Dr. Chaitanya will present his talk on surveillance of H1N1 cases reported in the state of Andhra Pradesh during 2020.
Yes,
Over
ma'am.
to you now.
Good afternoon, one and all. I'm Gotapu Chaitanya, working as State Veterinary Consultant for Zoonoses under the Integrated Surveillance Program on the Government of Andhra Pradesh. Today, I would like to explain about the surveillance of H1N1 swine flu cases reported in the state during the 2020. I'm the part of integrated disease surveillance project. So we use it to go to field trips, and we will actually identify in the clustering of the cases of the public health importance like influenza and the infectious diseases and zoonotic diseases, and we will further prevent the transmission of these diseases and limit the morbidity and mortality and assess the public health importance. We are the team of 5 members actually. So swine flu, we are all well known about that swine flu, H1N1, and then a recent pandemic 2009 happened with a lot of mortality. And then now the swine influenza, there are the 4 types: H1N1, H1N2, H3N2, H3N1. However, the most of the recent infections are with H1N1. Usually the pig is infected with the avian influenza, human influenza, along with the swine influenza viruses. So there is a reassortment of the process happened in the pig, and then result of the emergence of the new influenza viruses. So it similar to the COVID-19 and similar to the flu infections, the swine influenza is spread in the similar manner, and the symptoms also similar to that of the normal flu. And then some complications may lead to the lethal paralysis and then convulsions. The risk factors like COPD and compromised states and diabetes mellitus, pregnancy, and then cardiac related comorbid conditions. But we have to be careful with the children, because the children, sometimes we need the intensive medical interventions. The list of conditions, they may lead to the intensive medical treatment. And the difficulties mean generally the swine flu case identification itself is the biggest difficult task. SARI and ILA cases and the chikungunya and dengue, confirmation of the cases itself is a very biggest task, and the protection from swine flu we can be used, and the vaccines also now available. And the prevention measures, mostly the using of the face masks and gloves and the covering of the mouth, and then so, and recommendation for the people who are engaged with the pigs, like using of the masks and sanitizers and alcohol.
Dr. Chaitanya?
No, his audibility is very poor.
Yeah, audibility is very poor, and I think he lost the connection midway only.
Yeah, we can wait for some. Let me.
So, Dr. Chaitanya, can you hear us?
Give him 2-3 minutes, ma'am.
So we'll wait for 2 minutes and then we'll proceed
Yeah.
for our next talk.
Ma'am, I'm Gotap Chaitanya. Ma'am, I will connect again, ma'am. Actually, some server problem is there.
Yeah. You hurriedly, you please conclude now.
Okay.
Share your screen. Suppose he's again gone, ma'am.
Yes, Chaitanya, please share your screen.
Give me a second.
Are you sharing?
Ma'am, give me a second. I'm gone. This is the first case I actually reported during the January 2nd, 2020. and then along with our team and then district team, we visited the same place, and then we investigated the surroundings. There the pig rearing is the common, and then we formed the teams and we visited house to house. We done the house to house surveillance, and then we screened the contacts around the 38 persons who we identified as the primary and secondary contacts. We screened all of them. All are got the negative results only. This is a picture showing the screening of primary and secondary contacts screening, and then house to house surveillance activity for the prophylactic measures and for the education propaganda. And total 35 among the 35 cases, the more number of cases are reported in the rural areas, 54.28%, and followed by the urban and tribal areas. And then among the age group population, occurrence is it's equally distributed mostly, and but the elder population, elder people with above 80 years old, there less chance of occurrence. And 60% of the male population who showed the clinically significant symptoms when compared to that of the females. And the month-wise distribution, now the swine flu is a year-like phenomena, but the peak number of cases are reported during January to March, and then post-monsoon season until the November. This period is vary from the place to place, similar in the, but in my results, we observe the similar pattern like the national scenario. We are similar to that of national scenario. And the district-wise and sex-wise, most of the cases we reported in the Chittoor, and total 11 cases out of 35 cases. And then most affected population are the male. And diagnosis, the routine blood test generally used to differentiate from the flu symptoms of the bacteria. If we want to differentiate from the bacterial infection, so routine blood tests. And then chest X-ray, like complications like bilateral patchy pneumonia we can observe. The other some X-rays I got from the KGH hospital Visakhapatnam. And then nose throat swab for the RT-PCR. Generally, the preferred to collect the samples 1 to 4 days of the severe infection period. In case of the children, the virus may shed till 10 days and maybe longer also. And there is an application plots received from the SWIMS Tirupati. And in comparison, we will compare the clinical profile. All the cases were shown, 100%, all the cases were showed the cough and throat pain in 96% of the cases, and common cold fever in 93% of the cases, and breathlessness in 83% of the cases. And the temperature presentation is 99.96 Fahrenheit, and respiratory rate is 25.89 per minute, 25 to 26 per minute, and SpO2 at the room temperature is 82%. And we observe the sputum production, chest pain, and respiratory rate and SpO2 in the room temperature. There is a significant difference. We observed all these 4 parameters. And then this one is a complicated case we observed in the Anantapur district. She is affected with cerebral malaria and then COVID-19 and H1N1, but she recovered. Now she is normal.
Dr. Chaitanya, please conclude.
The findings investigation in 2020, 35 cases all are clinically and laboratory diagnosed with 0% case fatality rate. And then some cases are associated with the COVID-19, and no clustering of the cases are reported. And then we are not find any epidemiological link between any case with another case. But among the 35, 4 cases are the complicated cases. They observed the respiratory signs, and then they required the non-invasive ventilator facility. 2 patients are asthmatic, and 1 patient is chronic smoker, and 1 patient is affected with the COVID-19 and malaria too. The effective surveillance, case detection, and treatment as per the WHO guidelines we followed. So the last
Dr. Chaitanya, are you there? We can't hear you.
And early case detection, and then voluntary and reporting.
Dr. Chaitanya?
Ma'am, thank you for giving this opportunity. Actually, this is like an outbreak investigation compared to that of research, ma'am.
Okay. So in which lab was this investigation conducted?
Ma'am, pardon, ma'am.
Which lab was used to conduct this investigation?
Ma'am, actually, we are the 5 members of team. Here, the 35 cases involved in the 4 labs, ma'am. SWIMS Tirupati and KGH Visakhapatnam and NIV Pune, ma'am.
Okay. So, any particular season is associated with the cases in which it was done in Andhra Pradesh, isn't it?
Yes, ma'am. Ma'am,
So,
previously, ma'am.
Yes, please proceed.
Ma'am, previously we observed the year-round phenomena, but now the peak, not only the peak cases, normally the cases we are getting January, February and March, ma'am. And then ending of the rainy season till November, we are getting few more cases, ma'am.
Okay, thank you. Dr. Evaluator 2.
I think it's okay, Chaitanya. It's a good study, but it needs more exhaustive this approach.
Yes, sir. Yes, sir.
In the future studies. Okay.
Yes, sir. Yes, sir.
Okay, thank you so much.
Thank you, ma'am.
We have the next presenter, Dr. G.S. Gill. So are you there? Gulal Singh?
Yes, ma'am.
Okay.
Sharing my screen.
So you can share your screen with us. And Gulal Singh Gill will present on knowledge, attitude and practices of school students related to rabies in Punjab, India.
Is my screen is?
Yeah, it's visible to us.
Yes, visible to us. Good afternoon to all of you. I'm going to present my PPT on knowledge, attitude, and practices of school students related to rabies in Punjab, India. As we know very well that rabies is a highly fatal disease. Nearly 17 million dog bite cases are reported to occur annually in India, according to Manji's 2008. 20% of the 19 million human bitten by the dogs receive 1 or more doses of rabies vaccine in Southeast Asia, according to Google and Wright 2011, which could be improved by using health education activities. To keep this in mind, we plan this study to assess the knowledge, perception, and attitudes in the students of Punjab. One thing more I want to add in this study, that this study was first conducted in Punjab. Before that, there is no reporting of that type of studies. This study was conducted during 2016 to 17. First, in methodology, we randomly select 3 towns and 2 large cities, out of which from each town and each city, 2 wards are selected on convenient basis. That portion cover the urban area, and for the rural area, from each district we select 1 tehsil on random basis, and from each tehsil we select 1 village on convenient basis. For this study, first we designed a questionnaire to assess the gap in the school students from 6th to 10th class, and then take the ethical permission from the Anand Medical College and Hospital. In the questionnaire, the first part is the demography, second is the knowledge, third is the attitude and practice related questions. In the statistical analysis, the knowledge score was prepared by giving 1 score for each correct answer. The range is from 1 to 20. Univariable linear regression analysis was conducted in R commanded by considering the demography as an explanatory and knowledge score as outcome. Those variables in the demography show influence on the knowledge score were used for the multivariable analysis. This is the frequency tables for the demography portion. In this study, 313 respondents participate, and out of which 62% were the male and 72% were the belong to rural area. And the 65% of the students had the rabies knowledge score more than 50% in this study. And this is the frequency table for the knowledge related questionnaire. And it's find out that more than the students having very well knowledge about the rabies, that the rabies is transferred from animal to humans, and it is transmitted through bite. But they are lacking behind in the knowledge regarding the other routes of the rabies transmission. This study was correlation and with the very very correlated to the other studies like Nielsen 2014 and Sancheti Magolkar 2006. In the another part, it's find out that the students having very well knowledge about that the rabies can be prevented in the humans. More than 85% of the students give answer yes in this. And but the 34% of the students say that the rabies can be prevented by the traditional methods, like by using turmeric powder and chili. And 57% of the students were not aware about that the dog bite wound should be washed with the soap and water. But positive point is that more than 90% of the students have the knowledge that rabies can be prevented through post prophylaxis. This study was supported by Singh et al. 2018. And in the attitude portion, the most of the students say that they does not go for the treatment if they are attached by the stray dog or bitten by the own cat. And more than 85% of the students say that they play with the stray dogs even against their parents' advice. And in the one of the study conducted by the Spingel 2000 say that the children's having the emotional attachment with the dogs, so the children's are more vulnerable to the rabies. So we recommend that the schools need to be educate the children's to change the attitude and practices of the students and children's towards the free-roaming dogs. And in the univariable analysis, the age, presence of animal in the house and education level shows that they having the impact on the knowledge score. In the multivariable analysis, more than only 9% of the variation in the knowledge score is explained by this model. So it needs some refinement. In the end, the nutshells, we say that the analysis revealed that the knowledge score to be significantly associated with the level of education and presence of animal in the house. So there is a need to strengthen up the school education on prevention and control measure on important zoonotic diseases such as rabies in India. Thank you.
Thank you, Dr. Gulal. It was a nice presentation and really nice work at the ground level. So since you were, questioning the students and imparting some awareness also to them, so you could have targeted some other species also apart from dogs. So were the kids aware that rabies is transmitted from any other species apart from dog?
Yes, in the questioning, we also put some questions that which type of the animals transmit the disease. But most of the students are not aware about rather than the dogs. They know, even some of the students answer that the snakes can transmit that. So very less knowledge in the students there rather than the other animals also can transmit the disease other than the dog.
Okay. So thank you so much. It was a good work. What is the association of rabies with today's special occasion? Do you know that?
Mainly in the Punjab level, mainly this disease is maintained by the dogs, but we also need to be put some more clarification.
No, Dr. Gurlal, I'm asking you, what is the association of rabies with the celebration of World Zoonoses Day?
Yes, because on this day, the first vaccination of the rabies is developed by on that day. That's why the rabies day today, the zoonotic day is.
Okay. Thank you so much. So over to evaluator 2. Doctor, please go ahead.
Gurlal, it is really appreciable work at ground level, because you see this KP studies regarding the rabies. This is the need of our Gurlal, what was your sample size? How much population you have covered?
Actually, sir, the sample size we were calculated was 307 samples were the sample size, but we collect the 313 questionnaires from the students, more than sample size.
Okay. And most of the study was you did in the rural areas of the Punjab?
Yes, sir. More because this disease mainly related to the poor community and the rural areas that the already studies we gone through, and so that's why we covered most of the rural area, but also we covered the urban area also.
Okay. What was the aptitude of the urban people regarding the transmission of this rabies, especially among the school children of the urban areas? What was your findings?
Actually, sir, we did not there is not that much difference between the rural and the urban area but the rural area in the rural area, those having the animals at their homes having more knowledge and they are more aware about the rabies.
Okay. So did they have this concept of open wound and all that practices? If dog bites or some other animals bite, did you encounter such type of answers also in your study?
Yes, sir. We put just one question that would you go for wound washing after the bite of a dog? So nearly more than 60% of the students say yes. No, only 57% of the students said no. They does not aware about that thing.
So did you give some recommendations after this study to the Punjab government?
No, sir. Currently, it does not give any recommendation.
You can give your input.
Yeah. After the, sir, it's not published yet, but we are going to publish, and after that we recommend.
Okay. Thank you very much, Gurlal. Thank you.
Thank you, sir.
Okay. Thank you, Dr. Gulal.
Thank you very much.
Nice work. You may stop sharing your screen now. So next presenter we have with us is PV Siddhartha.
Yeah, ma'am. Am I audible now, ma'am?
Yes, you are audible. So you can please go ahead with screen sharing. So PV Siddhartha will present his talk on a proteomic analysis of crude and purified hydrated cyst fluid antigens used in the immunological diagnosis of cystic echinococcus in sheep and buffalo species.
Good afternoon, ma'am. Are my slides visible now?
Yes, they are visible and you may start. Your time starts now.
Yeah, thank you so much, ma'am. Good afternoon, everyone. Myself, I'm Dr. B. Vinayak Siddhartha, PhD scholar from the Department of Public Health and Epidemiology, Veterinary Science College, Veterinary University, Tirupati. So my topic is on the proteomic analysis of the HCF antigens used in the immunodiagnosis of cystic echinococcus across sheep and buffaloes. So the public health significance of CE is it's a mostly neglected tropical disease as per WHO because most of the infections they go unnoticed, and especially in the rural areas where there is a common dog-sheep life cycle. Like the dog feces contaminates the grass, and the grass will be taken up by the sheep, and that life cycle mostly nurtures this cystic echinococcus. And the fraternities that are mostly at risk are the farmers, slaughterhouse workers, veterinarians, dog owners, sheep rearing nomads. So everyone is at a good risk of contracting this infection. Whereas the diagnosis is concerned, the diagnosis is often difficult because there are no prominent clinical signs in the early stages of the disease. So even though a human has a hydrated cyst inside the body, the clinical signs are quite late by the time the cyst would have been developed to a greater size. So the diagnostic techniques normally employed are the imaging techniques like ultrasound, X-ray, and CT. But to our luck, the imaging techniques are not quite efficient because of the misdiagnosis due to the abscess, tumors, or the other cysts. So at this juncture, immunodiagnosis is often quite efficient in the diagnosis. So in this study, hydrated cysts were collected from the liver and lungs of sheep from a local slaughterhouse, and the cyst fluid was aspirated into a small container, and the microscopic examination was done. So wet film examination, H&E staining, and Lugol staining were done. Based on the presence of the protoscoles, these cysts were demarcated into fertile cysts and sterile cysts. Only the fertile cysts were taken for the antigen preparation. Regarding the choice of the antigen, in this study, a crude antigen and a purified antigen were extracted and evaluated. Regarding the crude antigen, the extraction procedure was centrifugation followed by the supernatant collection, which was filtered through 0.4 and 0.2 micron filters, and the filtrate was collected as crude antigen. Whereas the purified antigen is concerned, it was extracted by 3 different techniques. So the first one was the dialysis and ultracentrifugation, followed by Cephedex size exclusion chromatography and protein elution from acrylamide gels. Regarding the 3 techniques, the Cephedex G50 column chromatography was quite efficient because the protein concentration was higher, and even the protein profile revealed a perfect single antigen, which is a purified antigen. Regarding the protein estimation, post-extraction, the proteins were estimated for their concentration by 2 methods, Bradford method and nanodrop spectrophotometry. Bradford method was a bit old and conventional method where the sample size was more and the procedure was tedious when compared to nanodrop. Whereas nanodrop is a simple technique where a single drop of protein was enough to calculate, and a similar pattern regarding the correlation was found. Because in between the 2 techniques, the Bradford and nanodrop techniques, the correlation coefficient was quite high, just the sample concentration was higher in nanodrop. And after the concentration technique, the protein precipitation was done by 2 techniques. This protein precipitation was done just prior to the protein profiling by SDS page, where the concentration was increased by 2 techniques. The first one was TCA precipitation, and next one was acetone precipitation. TCA precipitation did not give any rise in the protein content, whereas acetone precipitation gave a rise to 40% increase in the concentration. So 1.85 mg per ml, it went to 2.52 mg per ml just prior to SDS page. And then the protein profiling was done by 2 pages, SDS page and urea page. Madam, is there any problem now?
No,
Hello?
no, please go ahead.
Yeah. So regarding the protein profiling, it was done by 2 page protocols, reducing page and non-reducing SDS page. The only difference was the beta mercaptoethanol, which was used as a reducing agent, and the both page procedures gave a similar protein profile, where 4 proteins were seen in the crude antigen, whereas a single 72 kilodalton protein was seen in the purified protein. Whereas in the reducing page also the same protein profile was there. Regarding the percentages of the acrylamide gel, 12.5% was standardized to be better for the wide range of proteins. So this is a standardization of SDS page. The sample concentration of 10 microliter was efficient, and the voltage was run by different voltages and different electricity ampere contents. And the 60 volts and 100 volts was efficient in getting clear-cut bands. And the APS concentration was also standardized to 20 microliter of APS for the dissolving gel and 10 microliter for the tracking gel. And the stain was also Comassi G50 was G250 was quite efficient when compared to R250, and 3 hours was standardized as a better staining time. And the protein profiling was also evaluated between SDS page and urea page. And here urea page was a similar protein profile was obtained, but with a clear-cut, more sharp and crisp protein bands when compared to SDS page. And further, the proteins were also tested for their purity by Western blotting. And in Western blotting, the crude antigen revealed 3 clear-cut bands of 64, 72, and 48 kilodalton, whereas a faint 4th band was obtained. Regarding the purified antigen, a clear-cut 72 kilodalton was obtained in the Western blotting. And further, the evaluation of these 2 antigens was done in sheep and buffalo. In sheep, the sensitivity and diagnostic efficacy were more for ELISA, whereas using the crude antigen. Whereas using the purified antigen also, the scenario was almost similar, where ELISA proved to be the better efficient test, whereas indirect hemagglutination test has gone with the lower sensitivity rates and lower efficiency rates. So these are the images that depicts the ELISA, DID, and CIEP, and this was also got IAEC approval for the Animal Ethics Committee. And regarding the buffalo scenario, regarding the purified antigen, DID and CIEP was surprisingly more efficient when compared to ELISA, whereas in crude antigen, diagnostic efficiency were good for ELISA. So finally, the conclusion is the Cephedex chromatography was efficient in extraction. For estimation, nanodrop method was quite good, and whereas protein profiling, SDS page was good for a wider range of proteins, and protein precipitation was good by the acetone precipitation. And in this study, a 72 kilodalton purified antigen was evaluated, and this antigen was used for development of an ELISA kit that was highly sensitive. And sheep species ELISA was quite sensitive, whereas buffalo there is a slight surprise where DID and CIP were efficient. So finally, the scope is there is a better scope to develop a universal antigen and a universal immunoassay for a better diagnosis. Thank you.
So thank you, Dr. Siddhartha. It
Thank
was
you,
really a good talk, and the work is highly appreciable.
Thank you, ma'am.
So my question to you is that did you collect some field samples for testing?
Ma'am, please come again, ma'am.
Did you collect some samples from the field also?
Yeah, madam. The sample collection was here. Sheep, there are 50 sheep sera were positive, and it's like 150 sheep sera. Among the 150 sheep sera, they were demarcated into 6 were positive for hydatidrosis based on the presence of the hydatidrosis during the postmortem examination.
So
Similarly,
go
yeah.
ahead.
So similarly, the total sample content was evaluated in buffalo samples and the what we say the sheep samples. So in buffalo it was 50, and in the sheep it was 150. So 150, the markation of the positive and the negative were based on the postmortem examination, because at the time of collecting of the sera sample, I was present at the slaughterhouse. So I used to check every individual animal for the presence of the cyst. So once the cyst was present, I used to take it as a positive serum sample, and it's considered as a standard when compared to these 4 immunoassays that were evaluated.
Okay, okay. Good. So over to evaluator 2.
It's okay, Siddhartha. It was a very exhaustive study. In which lab you did this work, in Tirupati itself or somewhere else?
Yeah, it's completely done in our public health and epidemiology laboratory, Tirupati, sir, by our own university.
So the areas, the sample which you have collected, that was also in and around Tirupati, or you have covered most of the Andhra?
No, sir. In and around Tirupati, most of the samples were collected, especially the positive samples. But when I tried for other samples a bit distant from Tirupati, they were collected, but they were negative. So they were used as negative sera sample.
Okay, it's a good study. Thank you very much.
Yeah, thank you so much, sir.
Okay, thank you, Dr. Siddhartha. So we have with us one more presenter for this session, Dr. Shreya Dubey. Shreya, can you hear me? So second call for Dr. Shreya Dubey. Okay, so if Shreya hasn't joined us for the session, so we come to...
we can wait for 1 minute if you
Okay,
are
Doctor.
optimistic. If
Okay,
you are
dear.
optimistic, we can. If you are sure, then we can quit. But otherwise, 1
Okay,
minute.
let's wait for another minute.
She is from your campus, ma'am, or outside?
No, Doctor, she is not from our campus. She is from outside.
I think she is not there, ma'am.
Okay, so Doctor, will close this session here. So we come towards the end of this session on viral and parasitic zoonosis. So I take this opportunity to thank my co-evaluator who had spared his time to evaluate the presenters. All were very nice. The work is highly appreciable. It's good to see that we are progressing very nicely in the field of veterinary public health and epidemiology. So, Dr. if you permit, we can close this session and we can move ahead to our next talk.
Definitely, ma'am. Thank you very much. Thank you to the organizers, all the faculty of this Center for One Health. Thank you very much for giving me opportunity to evaluate. Thank you, ma'am.
Thank you, Doctor. Thank you for joining us.
Thank you. Thank you.
We have a next talk in this session, and the talk is by Dr. Pavitra Kaur. Ma'am is Associate Professor in College of Veterinary Science, GADVASU, Ludhiana, and she is part of she's associated with many projects on zoonoses, and she has been working with us as a co-PI in many of our projects on important zoonotic diseases, especially brucellosis. So I invite Ma'am Pavitra Kaur to deliver her talk on diagnostic tools for brucellosis in animals.
Thank you, Dr. Simran. Is my voice audible? Is it clear?
Yes, ma'am, it's very much clear, very audible. You can now please share your screen with us.
Okay, sure. Just give me a minute.
Wherever we are having like this good system with camera and same problem is there for them. That's clean.
Either what we can do is
Dehumidify or something we can.
we have to always have it.
So Dr. Simran, is my screen visible?
Yes, Dr. Sir, it's visible. You may please go ahead.
Okay. So thank you very much, Dr. Simran. Good afternoon to everyone who is there in this session. First of all, I would like to thank the organizers for giving me the opportunity to share my views on brucellosis. Since I have been working on brucellosis for a very long time, so it was, though I had little time with me to prepare this talk, but it was rather easy because the topic, it was familiar with me. So I would start with the talk. So the topic is diagnostic tools for brucellosis in animals. Just a brief introduction to brucellosis. So as we all know, this is a zoonotic and highly contagious bacterial disease caused by several Brucella species, and each species, it has got a propensity to be associated with a specific host. So the common host for Brucella abortus is cattle, but at times the disease can be caused by Brucella melitensis and Brucella suis. So as we know that Brucella, they are species, they are intracellular organisms, gram-negative, cocobacilli, non-capsulated, non-motile, partially acid-fast. Now the disease has got economic importance, especially for the dairy industry, in terms of that the prime clinical symptom is associated with the reproductive tract. And the most characteristic manifestation in pregnant animals is delayed abortion, delivery of sick calves, reduced fertility, reduced milk yield, and all these things will lead to substantial economic losses. So decreased productivity in ruminants, and of course, in case of human beings, it is associated with acute febrile illness, again leading to economic, which holds economic importance. The disease, it is considered endemic in all Indian states, and though it is considered endemic, but however, it is rarely prioritized by the health systems, and WHO includes it as one of its 7 neglected endemic zoonoses. So very brief about the transmission. I won't go into the details. So major reservoirs, we all know they are animals, and with a spillover from the wild animals. And within the animals, the transmission can be through direct or indirect contact with aborted fetuses, placental material, ingestion via broken skin, mucous membrane, sharing of male breeding stock. Sexual transmission probably plays a greater role in the transmission of Brucella melitensis in sheep and goats, along with Brucella suis in swine and canis in dogs. Cross-species transmission among animals is also there. And in case of human beings, the occupational workers, that is the abattoir workers, farmers, vets, lab workers, consumers, they are all vulnerable to the infection when they come in contact with the aborted fetal material, placenta, vaginal discharge, milk products, meat, etc. And in case of humans, the route of transmission can be through oral skin, mucous membrane, or aerosol. And person-to-person spread of brucellosis, it is extremely rare. So when we say that the disease is caused by several Brucella species, so here is a list of different Brucella species along with their natural hosts. So as I told you, Brucella abortus in cattle, melitensis in goat and sheep, suis in case of pigs, ovis in sheep, neotomy in desert rat, canis in dogs, and other species of relatively less importance. And animals, this cattle, it can be affected, as I told you, it can be affected by abortus melitensis, and it can be affected by suis, but the infections are rare. Whereas in case of buffaloes, it is always Brucella abortus and melitensis. In case of sheep, again, with abortus, it is rare. Suis, it is possible that it could get infected by suis, and melitensis, it is there. Similarly, in case of goats, it is mainly by melitensis, whereas in abortus, it is rare. So now, We say the disease is endemic in India, and there are so many reasons for its endemicity. I won't go into the reasons, and the true burden is not known in the general population because it is assessed in defined groups or risk groups. And so, considering the damage and its profound threat to humans, specific identification of the organism is essential to control the disease in animals and subsequently in case of humans. So whenever we deal with Brucella organisms, two things we have to keep in mind. That is, one thing is the aseptic conditions, they have to be maintained. And second is, we should handle all samples as potentially pathogenic and of zoonotic importance. So the general approach is first collection of samples. This is a very simple layout which I have made. First is sample collection based on history and clinical science, followed by, of course, we can go for direct demonstration. I'll be showing in the coming slides, followed by isolation of Brucella species from the clinical samples, identification by staining and biochemical tests, and of course, then further confirmation by advanced diagnostic tests. So when we say sample collection, so as I said, aseptic conditions, and handle it as if they are all pathogenic. They may be negative, they may be positive, but we should handle them as pathogenic. So samples in case of abortion cases, a full range of specimens, they can be collected and submitted for a differential diagnosis. And a whole fetus, it can be sent if possible, but we can go in for fetal stomach contents, cotyledons, discharges, milk, serum samples, and of course, sections of cotyledons or fetal lesions for histopathology, semen samples, lymph nodes. So all these samples, they can be taken. Now the samples, they are followed by, the staining, the direct staining of these samples can be carried out by, that is partial acid fast organisms, these are. So it can be carried out by modified Zeel-Nielsen stain, followed by culture, biochemical tests, and of course, molecular tests in which we have a large array of tests, that is PCR, real-time PCR, followed by sequencing of the conserved regions, 16S rRNA, REC-A, RPOB, MLVA for strain ID identification, MLSA for subtyping, whole genome sequencing. So all these tests, they can be done under the molecular tests. And this could be, in addition to these, the indirect tests, we can go in for like the serological test, that is STAT, RBPT, CFT, ELISA, FTA, etc. And of course, other tests, which I'll be dealing in the subsequent slides, they can also be carried out. So the diagnostic procedures, we can say that for identification of Brucella, it includes direct examination, that is staining, culture, and molecular methods. And of course, we can have the indirect tests, which just now I said there are large numbers. The list is very long. So as per the OIE, this is the list which I have included in my talk. These are the test methods which are available for the diagnosis of infection with Brucella abortus, melitensis, or COVS. So like if we want to confirm suspect or clinical cases, we can go for staining, culture, PCR. And if it is simply, we want to detect the immune response to vaccination or to infection, if we are interested in serology, then we can go in for RBPT. Like if it is, again, for confirmation of suspect or clinical cases, because the samples we receive, mostly the clinical cases, farmers come to us. Then we can go for RBPT, then indirect ELISA, RBPT, STAT, indirect ELISA. These are the normal tests which we follow routinely. In addition to that, we can go for FPA, CFT. These can also be carried out. Of course, we can have the herd screening test like the milk ring test. So when I say staining, so the presumptive identification of brucellosis is accomplished by staining. And in this, the microscopic inspection of stained smears from aborted fetuses or placenta or vaginal swabs, whatever direct specimen, we can stain it by the modified Zeel-Nielsen staining that can be carried out. And here, the Brucella, in direct staining, the Brucella will appear as small red staining cocoa bacilli in clumps. Because as I told, they are intracellular. So because of their intracellular growth, they will appear as small red staining cocoa bacilli. Now, when we talk of isolation, so once a presumptive identification is made, although in the presumptive identification, it can be a bit confusing because we need to differentiate it. So many other organisms can appear like that. So we need to go in for culture. Culture is the gold standard. Though it is time consuming, it is laborious, it requires, there are so many drawbacks. It requires an expert personnel to carry out the isolation tests. But still, despite that, this is considered as the gold standard. So normally, when we have samples, normally if, say, we want to isolate brucella from fetal stomach contents, which is relatively, there is no contamination in it. So the organism will grow very well on 5 to 10% blood agar. No need to add any supplements. But of course, if we are dealing with uterine discharges or any other sample in which we suspect contamination could be there, then of course the selective media is needed. And there are so many commercial available media which are there, like we have from High Media, there is Brucella agar base. So basically, we can have a base, we can put blood in it, 5 to 10% serum or blood, it can be added along with supplements. Supplements which contain antibiotics like polymyxin bisulfate, bacitracin, cyclohexamide, nalidixic acid, vancomycin. And of course, as per the OIE, there are other media also like Farrell's medium, Thayer's, Martin's medium, trypticase, soy agar, Bactose, Streptose agar, all these also give good results. And then if it is, we have to isolate brucella from blood or other body fluids or milk, then we prefer the biphasic medium, which is the Castaneda's medium. And when we say inoculation onto the media, so liquid specimens, if we have liquid specimens, they can be inoculated straight onto the plates. In case of cotyledons, we go in for scrapings. Whereas in case of tissue samples, if we have tissue samples, we homogenize them and then the aliquots are used for culture. If we have milk sample, though it is very, it is difficult to isolate brucella and it depends upon the sample also. For milk sample, it becomes a little bit more tough. So milk sample, first we can centrifuge it, then at 2000 G for 20 minutes, and then loop full of cream and sediment can be used to inoculate the plate. And of course, those samples which are brucella milk ring test positive, they often yield brucella abortus. So the inoculated plates, they are incubated at 37 for up to 15 days and 5 to 10% CO2 incubator. So isolates, once we have grown them on the plates, they are identified on the basis of colony characteristics. So maximum we keep the plates for up to 15 days, but the growth can be evident after 3 to 5 days of incubation in the form of the colonies will be glistening bluish colonies, translucent colonies. And of course, these are when the organisms are in the smooth form. For the first time when we see the colonies when they are in the smooth form. But as they age, the colonies, they can become opaque. And of course, there are exceptions like in case of abortus, COVS, melitensis, and neotomy. The colonies, they are smooth, whereas in case of ovis and canis, they are always in the rough form. And then we can go for differential staining of the organisms. That is, modified Zeal-Nielsen's method can be used, or gram staining can be carried out. And then biochemical tests like oxidase, catalase, nitrate. So many tests are there which can be used for identification of the organisms. And here also, when I say oxidase positive, it is not all. Exceptions are there, like again, ovis and neotomy. They are oxidase negative. Urease positive, again, except ovis and some strains of melitensis. They are oxidase negative. And of course, we can go for rapid slide agglutination. And I'll just not talk about it. This rapid slide agglutination, it also helps in biotyping of the organism. So if we have a positive antiserum for Brucella abortus, we can go for rapid slide agglutination test. So when I say once we have presumptively identified the organism on the basis of biochemical tests, we can go for biotyping of the organism also. So for biotyping, 3 or 4 tests they are required. Like Brucella abortus, it has got 7 biotypes, and of course strain 19 is also there, which is the vaccine strain. So like requirement for carbon dioxide, H2S production, urease activity, thionin and basic fusion. So these are thionin and basic fusion are the dyes which help in, again, biotyping. Like abortus biotype 1 will grow in the presence of basic fusion, but it will not grow in the presence of thionin. So many others like this. And of course, agglutination in case of monospecific sera, this also helps in the characterization of the biotypes. followed by, we can go for another test, that is phages. They can be used, Tbilisi phage, they can be used for biotyping of the organism. Now, if because the strain 19 is also Brucella abortus, so if we want to differentiate Brucella abortus biotype 1 and strain 19, because majority of the tests are similar between them, then we can differentiate by growing the two on media containing, that is thionin and erythritol. So Brucella abortus, that is the field isolate, it will grow in thionin, whereas the vaccine strain will not grow in thionin. Whereas in case of erythritol, again, the vaccine strain does not require erythritol for its growth, whereas Brucella abortus requires. And of course, resistance to the antibiotic, that is penicillin, the field isolated is resistant, whereas strain 19 is sensitive. So this was a little bit about the conventional tests which can be used. That is, we can go for isolation and biochemical identification of the organism. Though this classical method of identification and differentiation of Brucella into different phenotypes, it is into different biotypes, it is based on a number of phenotypic traits. But as I told you, this is time consuming. This is associated with a high risk of lab-acquired infections. It requires, of course, we require a biosafety level 3 highly skilled technicians. And also, you know, these tests, these conventional tests, they are not able to differentiate the various Brucella species. So if we want to differentiate the various Brucella species, as I told you in the beginning, there are so many species of Brucella, about 10 species of Brucella. So their PCR, the polymerase chain reaction, it becomes a very promising method of diagnosis. And this PCR, it is rapidly replacing the conventional assays. And a number of conventional and multiplex PCR assays have been described by various researchers. So multiplex, as you all know, this is just in one reaction, we are putting all the primers together for all the different species or strains, and then we put it into one reaction tube, and we run it, and you know, all the different species, they can be differentiated. So a number of genus and species specific conventional PCR assays, they have been described and they are reviewed in the literature. So I won't go into the details because so many are there, and it is not possible for me to list each and every one. Now, if we talk about the species specific, so the first was the MOS PCR, which was for differentiation of Brucella abortus biovar 1, 2, and melitensis, Brucella ovis and suis. Brucella ovis biovar 1 and Brucella suis biovar 1. And this was the first species-specific PCR using 5 primers developed by Bricker and Holling. So then, of course, this PCR was again modified. The modified MOS PCR was there, which was able to differentiate the Brucella field strain isolates and the vaccine strains. S19, that is RB51. However, you know, this MOS PCR, it was unable to detect Canis, neotomi, Ceti, some biovars of Brucella abortus and suis. So, of course, in between this MOS PCR and Bruce ladder multiplex PCR assays, so many other PCRs also came, like there was a multiplex PCR, which is there in the literature, which could detect Brucella microti also. And scientist Sung et al. In 2011, he introduced some new primers in this, Canis and microti. So it could differentiate all the 10 Brucella species. However, this Bruce ladder multiplex PCR, this is the most widely used when we talk of the species identification and differentiation. And this Bruce ladder multiplex PCR assay is capable of detecting almost all the species and the vaccine strains of Brucella, that is S19 and RB51. And of course, this has been evaluated in so many labs from different geographical origins. Of course, the real-time PCR, again, so many real-time PCR assays have been described. And these real-time PCR assays, we say they have got higher sensitivity in comparison to the conventional PCRs and the multiplex PCRs. And in addition, we have, you know, nested PCRs, semi-nested PCRs. So all these, they have been developed for the Brucella organism. So as this picture shows, The first picture, that is, this is a multiplex PCR, wherein, depending upon how many bands and different band sizes, what we get. You can accordingly, you can identify which species the organism it belongs to. So for each different set of, the pattern, it will be created. For abortus, it will be different. For melitensis, it will be different. For COVS, it will be different. And of course, this second picture, it shows the conventional PCR, which can be done on the cultures also. It can be done directly on the samples. Now, however, in case of multiplex PCR, sometimes, whenever we tried, it was difficult. The multiplex PCR, it is good when we talk on cultures, but when we talk on directly on the clinical samples, so there, these conventional PCRs, they hold importance. Not very reliable, or not reliable, not very good results they are obtained in the multiplex PCR. And of course, the real-time PCRs, we have seen in our own study, and of course, it is there in the literature that the real-time PCRs, they are more sensitive in comparison to the conventional or the multiplex PCRs. Now, this was about the molecular tests. Now, coming to the serological tests. So suppose we have serum samples, and we are suspecting it, we want to test it for the presence or absence of antibodies to brucella. So we have the RBPT and the STAT. I won't go into the details because this is a very common test. This is a buffered, RBPT is a buffered antigen test. So at a pH of 3.65, RBPT, the killed brucella abortus smooth cells stained with the rose Bengal dye, they are buffered. And within 4 minutes, it is a very rapid test. Within 4 minutes, if you are able to see the agglutination, we say the sample is positive. So we can say this is a very fast screening test, and the sensitivity is very high, 99 to 100%. But the only problem is, in case of RBPT, sometimes the antibodies, the vaccinated antibodies, they can also appear in the giving false positive reaction. Now, in case of, the drawback of RBPT, one more drawback of RBPT is that though it is useful for abortus, melitensis, and COVS, but this is not useful in case of Canis because it detects antibodies against the O lipopolysaccharides of brucella, O polysaccharide, that is the LPS of brucella, right? So that is why their RBPT is not useful. Second test is STAT, which is also known as the standard tube or the slow tube agglutination test. Now, again, this is based on the principle of detecting agglutinating antibodies predominantly of the IgM isotype. So large antigen antibody complexes are formed. They get agglutinated at the bottom of the test tube. The only problem is this is a comparatively slow process. It requires an overnight incubation, sometimes more than overnight incubation also at 37. Despite being a standardized and exceptionally robust test for brucellosis, but it has got some drawbacks. That specificity, sometimes specificity is less, which can be enhanced by treating the serum with EDTA. And this minimizes the cross reactions because of IgM. But this test is no longer promoted by OIE, the World Health Organization for Animal Health for bovine brucellosis detection. But still, it is extensively utilized for diagnosing human brucellosis. So this is just in case of, we all know in case of animals, a titer of 1:40 or above, we take it as positive. So next test, next serological test, which is of course the CFT, it is, it is recommended by the OIE. Though it is rarely carried out, as far as I know, in the laboratories across India, it is very less carried out. Probably not because there are so many drawbacks to this test, but still it is a prescribed test for international trade by the OIE. So despite it detects specific antibodies belonging to IgM and IG1 types capable of activating complement. So in this case, the Brucella abortus antigen with the dilutions of heat inactivated serum and titrated source of complement. After an apt period, we add sensitized sheep RBCs. The formation, so what happens when the primary immune complex is formed? It will activate the complement. So it is not accessible to the secondary immune complex of the sheep erythrocytes, that is the sensitized sheep RBCs and rabbit antibody. Hence, it results in slight disintegration of the erythrocytes. So, because as I told you, despite the high sensitivity and high specificity of this test, it is, however, a laborious method. It requires highly trained technicians, appropriate lab facilities. So it is becoming less acceptable in our labs. So as the test, the standardization itself is very laborious. So since the standardization is also laborious, plus this test is also laborious, so it is gradually being replaced by ELISA. So when we talk of ELISA, that is the enzyme-linked immunosorbent assays, the ELISA test is highly sensitive and specific. This is simple to execute in comparison to the complement fixation test. It requires minimum equipment, so which are readily available. Nowadays, they are readily available from numerous commercial sources in the kit form. So we have the ready-made kits. You know, it was way back when we used to standardize the ELISAs and do everything one step. So it was a bit time consuming at that time, but nowadays we have so many commercial sources of these kits that it has become so easy. In half a day we can get the results. So normally two types they are categorized when we talk of brucellosis. So they are generally categorized in two classes, that is the indirect ELISA and the competitive ELISA. And most of the indirect ELISAs, they again use the purified LPS as antigen and mainly detects IgG or IgG subclasses. ELISA, it is highly sensitive. I told it is highly sensitive. But you know, the ELISAs, they are mostly subjected to some non-specific reactions. Just as in case of RBPT, we get false positives. So here also we get some. There are chances we could get some non-specific reactions, mainly ascribed due to some cross-reacting organisms like Yersinia infections. They could be there. So all these things, because of all these things, there is the more specific competitive ELISA. And this competitive ELISA utilizes monoclonal antibodies which are aimed against the specific epitopes that is not shared, the cross reactions, that is not shared with the LPS of Yersinia. So the specific epitopes present in the O polysaccharide chain of smooth LPS of Brucella. And so as I told you, ready, you know, so many commercial kits, they are available, like one company, IDEX. It is very well recommended. IDEX check it brucellosis. The kits are available for serum samples also, for testing the serum samples. The kits are there for testing the milk samples, and the kits are there for different species also. We have different IDEX kits for cattle, buffalo. We have different for ruminants. It is there for milk ELISA. It is a different one. Then we have the rapid antibody test kits from genomics. Then IgG avidity ELISA, dot ELISA kits, then applied biosystems. They also, so many other commercial companies are there which supply these kits, and they have made this thing a very easy job. So now coming to, now here in ELISA, I would also like to say that we have the dot ELISA also. So in dot ELISA, you know, instead of a plate, we are using a nitrocellulose membrane. So then we move on to the milk ring test. So milk ring test, now before I move on to the milk ring test, let me tell you a little bit about the sensitivities. So if we compare, because these are serological tests, RBPT, STAT, ELISAs, indirect ELISAs, of course, the indirect ELISAs, they've got a higher sensitivity. We have seen in our studies also, and it has been reported in the literature also, that is the indirect ELISAs, they've got a higher sensitivity in comparison to the RBPT or the STAT. And in case of C-ELISA, if we talk about the IDEX kits, there they say that it is capable of differentiating between the vaccinated and the naturally infected animals also. So coming to the milk ring test, this is a herd screening test. So this is a rapid agglutination test. Of course, it is carried out on the whole milk or the cream. And in this, the hematoxylin stained brucella cells are added to whole milk and incubated for the reaction to take place. So formation of a purple colored ring is considered as positive. So the presence of this anti-brucella antibodies, it is confirmed by this antigen antibody complexes in the form of a purple ring on the surface of the cream. And if positive, On the contrary, if the anti-brucella antibodies they are absent, the cream it will remain uncolored. How this test is, however, not considered to be a sensitive one, but it can be repeated owing to its low cost. And if positive, the samples they are tested by other serological tests in order to identify the infected animals. Of course, we have the Rivenol precipitation test, because I haven't done it in my lab, so I'm not very sure of what are the things which go on, but just to, in brief, this test is capable of distinguishing between vaccinated and infected animals and also chronic carriers. And it is aimed at eliminating some non-specific reactions, which is based on the precipitation of high molecular weight serum glycoproteins, in this case IgM. And acridine dye such as Rivenol is used for the precipitation, after which it is removed, and the lysate, the supernatant, is tested using rapid plate agglutination test, either in the form of plate test or it could be in the form of tube test using serum dilution. 2-mercaptoethanol test is also confirmatory test for selective quantification of IgG. As disulfide bridges, they're converted to monomeric units, and it is quantified by using 2-mercaptoethanol or erythritol. The latter is preferred due to high toxic levels of 2-mercaptoethanol. And then we have the COVS antiglobulin test. This is one of the most sensitive serological tests. Especially it is used to validate serum agglutination test results from those animals that have been declared negative or which have given ambiguous results in the standard tube agglutination tests. And it could be because of the presence of incomplete antibodies of IgG type that combines with the cellular antigens but fails to produce an agglutination reaction. Again, the test is not recommended to test the vaccinated animals. And of course, we have other tests also, like the Brucella COVS gel test, that is the odor test, that is also there. Then we have the rapid immunochromatographic assays, that is the lateral flow immunoassays, which are labeled with a colloidal protein G conjugate, and different, these strips, they are available. Like we have a methodology developed by Diaz et al. Then we have the Brucella rapid antigen test kits, which rapidly detect bovine Brucella antigen from cow's whole blood, serum or plasma specimen, and it has got a relative sensitivity of 97% and specificity of 97%. Coming to another test, that is the fluorescence polarization assay. Though it is not routinely used in the labs, because extraction of the antigen, in case of this, the extraction is a little bit tedious process. But once we have extracted the antigen, then the test is relatively simple. Just like ELISA, it is very simple and it can be carried out very quickly. And large number of samples can be screened at a time. So this is based on the principle that how quickly a molecule spins in a liquid medium. And this is correlated with its mass. That is, molecules of small size will spin faster and depolarize a polarized light beam more, while bigger molecules, they spin more slowly and consequently they depolarize light less. So basically, the degree of depolarization in millipolarization or the MP values, so that is measured in this test. So what we do basically, as I told you, we have to extract the antigen, and once we have extracted the antigen, that antigen is labeled with the fluorescein isothiocyanate, that is the FITC. So we extract the antigen, we label it with the FITC, and then the serum samples which we have collected from the field, those serum samples, they are incubated with the labeled antigen, right? They are incubated with the specific labeled antigen that is labeled with the FITC. So if antibodies are there in the serum sample against the Brucella species, large fluorescent complexes will be formed. Obviously, the antigen will go and bind to the antibody, and the FITC also gets bound. So large fluorescent complexes will be formed. Whereas in case of negative samples, the antigen remains uncomplexed. And these smaller molecules, they will spin more quickly. Therefore, it will cause greater depolarization of the light, giving low MP values than do the samples which are positive, in which higher millipolarization values they are obtained. So this is a summary of the serological tests which are used for bovine brucellosis. And this I have taken, this table I have taken from Quinn & Carter, 2004 book, Clinical Veterinary Microbiology. So if we talk of the herd test, we have the Brucella milk ring test. Then if we want to test individual animals, we have RBPT, we have the SAT, we have the CFT, and we have the ELISA. And the normal which we do is RBPT, STAT, and ELISA, because CFT, as I said, this is a laborious test. And of course, the supplementary tests like heat inactivation test, Rivenol precipitation, mercaptoethanol treatment, and Coombs antiglobulin test, they can also be carried out. Then we have lab animal inoculations also. So in case of lab animal inoculation, though guinea pig inoculation, mice can also be used, but we go in for guinea pig inoculation, though it is not commonly used. But guinea pigs, they can be inoculated with the suspected sample which we have. Because in this case, we will get the organism in the pure form. So if we have a highly contaminated specimen with us, we can go in for this guinea pig inoculation method. This is the most sensitive test for isolating pathogenic brucellosis. So what we do is, we take 0.5 or 1 ml of the suspect tissue homogenate and intramuscularly it is inoculated into the guinea pigs. Normally, we take two animals, they are inoculated. Then those animals they are euthanized 6 weeks after, and we take serum and spleen for bacteriological examination and isolation and identification. Then another test is a skin test. That is, this is an allergic test which utilizes a protein to detect specific cellular immune responses, and the administration is followed by local inflammation in sensitized animals. It has got a very high specificity. It is useful in eliminating the false positive serological reactions, especially in case of animals which are latently infected and those which lack any quantifiable antibodies. Another test, this is an advanced test again, which this is peptide nucleic acid fluorescent in situ hybridization, or in short known as PNF-FISH. So what is this? This is a technique which can be used for to provide rapid and accurate diagnosis of brucellosis. So in this, we use a fluorescein labeled PNA probe. So basically, it is a combination of using a fluorescein labeled PNA probe in combination with traditional staining procedures. And these PNA probes, they are DNA mimics. Basically, these are DNA mimics with neutral polyamide backbone, right? Which is formed by repetitive N-glycine units, that is 2 aminoethyl glycine units. And this neutral characteristic, it is responsible for high thermal stability between PNA, DNA or PNA RNA bonding compared with those DNA probes. And so specific hybridization will take place between the PNA sequences and the nucleic acid complementary sequences as per the Watson and Crick. So PNA probes, another advantage of using the probes in comparison to the DNA probes is they have got a relatively smaller sequence, and due to the uncharged backbone, as I said, they have got an uncharged backbone. They have got superior hybridization characteristics. That is high degree of specificity, strong affinity to hybridize to the ribosomal RNA. And in this case, in case of Brucella, we used 16S ribosomal RNA as a target probe. So we took this sequence, we labeled it with the PNA, the original sequence. And then these probes, they were attached to the fluorophore dye. Of course, we got them commercially made. And then they were detected by fluorescent microscopy using the FISH method. So this was tested on the isolates also, where we got brightly fluorescent, cocoa bacilli, brightly fluorescing cocoa bacilli were seen, and they were tested on the clinical samples also, where also we got very good results. That is, we were able to see the cocoa bacilli, fluorescing cocoa bacilli. Then, of course, we can go in for immunocytochemistry and immunohistochemistry. Immunocytochemistry we did on the Brucella isolates and on clinical samples. And when we compared it with the isolation of the organism. We compared it to the isolation from those very clinical samples. We processed the samples for isolation, and the results, they suggested that both immunocytochemistry and immunohistochemistry, of course, the immunohistochemistry was carried out on the differential samples. It suggested that they both are sensitive diagnostic techniques in comparison to isolation. So coming to the conclusions of today's talk, the presence of brucellosis, it can be diagnosed by several direct and indirect tests, although each of the method has got its own limitations, advantages and disadvantages. The species, different species of Brucella or biovars, they can be identified by phage lysis. As I said, it was WC phage, they can be used or by cultural, biochemical, and serological tests. PCR, it can provide a complementary identification and typing method based upon specific genome sequences. We have to keep this in mind that no single serological test is appropriate in each animal species and in case of all epidemiological situations. So the reactivity of samples that are positive in screening tests that should be assessed using an established confirmatory or complementary strategy. And of course, it is always advisable to use a combination of tests for accurate diagnosis of the disease. So these are the references from where I have prepared my talk. And so thank you very much.
Thank you very much, Dr. Pavitra. So we got a question. One of the question is, what test do you think of serological test is considered as most sensitive and specific for testing in animals?
Okay. So if we talk of the sensitivity and specificity, we can go for indirect ELISA, we can go for fluorescence polarization assay also. But normally what we do is we go for indirect ELISA because indirect ELISA is more sensitive than RBPT and STAT. Whereas FPA, as I said, it is a little bit tough to carry out. The antigen extraction is difficult. Of course, the test with the highest sensitivity and specificity is complement fixation test. But because it is it is difficult to carry out, it is difficult to standardize, we normally don't do it. So I would suggest indirect ELISA.
Thank you very much, Dr. Pavitra. So it was a wonderful talk, and I hope almost all of the participants have enjoyed it. And on behalf of organizing team, once again, I would like to thank Dr. Pavitra Kaur, who has spared her valuable time and talked about this disease, which is very endemic in our country. Thank you very much, Dr. Pavitra, once again.
Thank you, Dr. Rajneesh. Thank you so much.
Thank you. So could you please stop sharing your screen so that?
Yes, sure.
Thank you. So now we are having with us Dr. Peter Thompson, who has sent us his pre-recorded video, which we will play, and then he will be happy to take any questions if any. So please wait for another 1 minute so that we can start that. So I hope it is visible. Could you please say yes? And my voice is?
Yes, it is visible.
Are you able to hear the video also?
Yes, we can hear. Dr. Rajneesh, we can hear.
Thank you, Dr. Pavitra. Thank you very much. So we do have.
I would like to thank the organizers for giving.
So we do have our next speaker who has sent us our his pre-recorded video, and we are going to play that. So before that, I would like to briefly introduce Dr. Peter Thompson. Dr. Thompson, he is currently working as a scientist. Sorry about that. Give me a second.
I will complete.
Sorry about that. Okay. So thank you very much, first of all,
Dr. Peter Thompson for joining us and sending us this pre-recorded video. Dr. Thompson, he has earned Bachelor's of Science in Biology at Virginia Tech, and then he did his PhD in Behavior Ecology, Evolution, and Systematics at the University of Maryland. He has fellowship at the National Academy of Sciences, Engineering, and Medicine, and then he worked as a postdoc fellow at the USDA Animal Parasitic Disease Lab. In 2019, he accepted a position as principal investigator in USDA APD lab, where he now focuses on evolution of nematode parasites in ruminants. His research program uses genomic techniques to reveal the genetic diversity of parasites within and between species in order to better understand population of GI helminths. And he is also interested in determining the basis and maintenance of anthelminthic resistance in worm population, finding genetic subdivisions among populations that can act as management unit and understanding the transmission of parasites to livestock and wild animals. So we are very fortunate that Dr. Peter Thompson spared his time to record this video, and then we'll be happy to answer our questions, if any, after this talk. We welcome you, Dr. Peter, and we thank you for sparing your valuable time. And now I will go to play this video.
To address the group. Today I'm going to be talking to you about genomics and One Health, how advanced diagnostics can monitor and understand the spread of zoonotic pathogens. The world is interconnected like never before. What you're looking at are the air traffic patterns across the globe. And what you should take away from this is that humans can move anywhere on Earth in about 24 hours, taking with them whatever it is that they picked up along the way. We also ship a tremendous number of things around the globe on giant container ships. Along with those products, we can ship bacteria, insects, any number of things that can cause disease when they arrive to their new location. Add to the fact that humans convert more and more wild places to developed land due to their constant need for new resources, and we end up in a situation where zoonotic path spillover is likely. As new species come into contact, such as bats and pangolins, we end up with unintended consequences. As you all are aware, coronavirus SARS-CoV-2 rose up 18 months ago and the world changed. It was a zoonotic virus that emerged in China in an open-air market where wild animals were kept in close proximity to one another, alive and dead. The virus made a jump from bats, perhaps to a pangolin, and then into humans, and the results have been devastating the world over. The costs have been extraordinary. We are currently almost at 200 million cases worldwide, with 4 million deaths. There are syndromes associated with the disease that may last for years to come and will have significant economic impacts on the people who suffer from such syndromes. In total, thus far, it's been estimated that between 8 and 16 trillion US dollars have been lost in global productivity. So what if we could get ahead of the next pandemic? In 2019, Brooks, Oberg, and Boger published a book called The Stockholm Paradigm. In it, they stated that environmental perturbation leads to shifting distributions of animals. They focus primarily on climate change, but this also applies to the way humans move animals around. This brings animals together that did not previously interact, and in these interactions, pathogens may pass from a historic host to a new host, leading to a host shift. This sometimes results in zoonotic potential and human problems down the road. As a solution, they gave the idea that we should follow a DAMA protocol, that is document, assess, monitor, and act. We need to document the biodiversity that is in all ecosystems so that we can then assess the potential for zoonotic spillover, taking a look at which species are related to known pathogens, whether known pathogens are in new places that we did not observe them before. Then we'll monitor for changes in distribution and abundance in order to understand whether a new pathogen is emerging. Once we see a pathogen emerging, we can act before that pathogen gets out of control. And so our system becomes more proactive rather than reactionary, such as it has been in the COVID-19 pandemic. So how do we document biodiversity? Traditionally, we have used visual identification, morphology. However, there are only a few experts that remain that still can reliably identify pathogens based on sight and differentiate among parasites by morphological features. There is way too much work for them to address everything that we need to do in today's expanding world. That leads us to DNA sequencing. DNA sequencing is a well-known technology. It's reliable, it's universal. Basically everything we know, every living thing we know uses DNA as its genetic code. We have developed protocols where simple databases can be formed and search tools such as BLAST to identify a given sequence as known or unknown. The cost continues to drop over time, making it economical to sequence pretty much as we want. It's just a matter of having the capacity. Today, I'm going to talk most about metagenomics. This is a technique where material is collected from a group of organisms. We then sequence that collective DNA and use bioinformatics to separate out the unique DNA sequences diagnostic for different species. This is what it might look like. You might take a sample of soil down through 3 or 4 different levels. Maybe you would go into an estuary and take a water sample to understand what's in the water column. Or you could go to a farm and sample waste management areas in order to see what pathogens are being passed around the farm. You then extract the DNA using a number of methods and ending up with pooled DNA from all organisms in that sample. You then amplify a locus of choice using PCR. For most analyses done thus far, they've used the 16S ribosomal DNA subunit from bacteria. This is also present in many of the mitochondrial genomes of eukaryotes, making it a nearly universal locus. We then end up with a lot of short DNA sequences put on a next generation sequencer, such as the Illumina MiSeq, generating sequences which can be identified as belonging to one species or another, as represented by the colors here. This is quantitative. If you just stack all of those different fragments up, you can see that most of them were red, while some were purple, green, brown, or yellow. They are specific, so if we compare the sequences to a database, we can give them names. Here I've just used taxon A, B, C, D, and E as placeholders, but it's indicating that these are known sequences. And therefore, we can put names to them. Occasionally, we will come across a sequence that is unknown to us. In that case, we need to add the sequence to the database and then put it on a list for follow-up. Assess whether it is a potential pathogen, whether that's because it's closely related to a known pathogen or has some characteristic that is similar to other pathogens. We can then sample locations or particular areas of interest over time and look for trends. Here you can see that the brown is steadily increasing over time. This is the type of data that would be recognizable for artificial intelligence. And I can see a future where we would put machine learning to work to try and identify these trends as they are becoming apparent and warn us early that we need to address some situation that is emerging. We might also be interested in local variation. Say on a farm, we need to sample the surrounding forest, a field, the soil in the field, where the livestock are kept on the farm, a nearby stream, or even the house, and we might notice that there are interesting trends, and maybe where the livestock are kept, there's a very different distribution of local specific variation. Some of the questions that can be answered by metagenomics include: what species are present in a sample? What are the quantitative differences in species abundance between samples? Are there functional differences between species collections? Are there known pathogens in an environment based on either known pathogenicity or perhaps even a locus a gene that we know to be pathogenic that is being carried by an organism? Are there unknown species that are closely related to known pathogen? Or maybe are there antibiotic resistance alleles present in a location, which is sort of the poster child of One Health? One influential study was published in Nature a couple of years ago, where they monitored urban sewage and looked for antimicrobial resistance alleles based on metagenomics analyses. And what they found was what was present in Europe and North America was significantly different from the other places on the Earth, leading to insights about antimicrobial use and overuse and how perhaps we can get a handle on what's going on. Some of the advantages of metagenomics are that it's simple to run. If you can do PCR, you can run samples on the Illumina platform. There is no advanced training required. You simply need to be able to hold a pipette. It's highly specific. DNA sequences can be compared to global databases in order to understand all of the organisms involved in a particular location. Each added sequence simply expands the database, giving us more knowledge of what is out there. This will eventually be excellent content for future artificial intelligence applications. The technique is flexible. You can change the locus of interest as needed. If you're interested in functional studies, you can focus on a gene of a particular function. If you're interested in antimicrobial resistance, you can focus on antimicrobial resistance genes. What you do is up to you. The technique is flexible and the data is lasting. The alphabetic nature is future-proof, whereas morphological techniques of the past had required some interpretation. So what do we need in order to move forward? Number one, increased sequencing capacity that is distributed around the globe. Particular attention should be placed in developing countries where resource needs are driving people into closer contact with wild places where zoonotic pathogens often emerge. We need to provide standardized sampling guidelines, including the best methods for extracting DNA from a variety of phyla. We also need to think about what loci are best to target based on the questions we are trying to ask. Finally, we need to prioritize where we will do our sampling. As indicated earlier by the example of coronavirus, open-air markets would be an obvious place to start. We also need to do regular sampling of city sewage treatment, showing the utility of that in the previous example. Farms are an obvious place where human management are often changing the way pathogens react to treatment. And so in order to keep an eye on anthelmintic resistance, farms would be an obvious place to sample. The big question is, what do we do about wildlife? We know that 60 to 70% of new diseases come out of wildlife. How do we monitor those for new and existing diseases? We will have to have some priorities and maybe focus in on species closely related to us, such as non-human primates, or our domestic animals like swine or ungulates, or even wolves and felines. Decisions are going to have to be made because we can't sample everything that is out there, but that should be our ultimate goal. As we become more sophisticated, we will not just want to identify what is going on in the environment, the biodiversity in the environment, but also to characterize functionally what is going on in those communities, looking at different soil types, maybe waste reclamation sites. what is going on with the human microbiome and health and how it interacts with zoonotic disease? Can we begin to classify ecosystems as healthy or degraded based on their metagenomic profile? Things are going to evolve. Sequencing technologies are getting smaller and smaller, and here you see the MinION device from Oxford Nanopore, which has been used in remote locations. Their idea is to sequence anything, anywhere, anytime, and they've even found their way onto the space station. In conclusion, we have some simple metagenomic techniques that can be used to document the biodiversity across the globe. The combination of survey and inventory with a widely distributed metagenomic capability can help identify potential zoonotic pathogens. This will guide an assessment of the risks to our One Health. Continued monitoring will allow us to act proactively rather than reactively. If we do these things, we could save millions of lives and trillions in lost productivity. And so my recommendation to you is to get out there and start sampling. Even if you don't have the capacity to do the sequencing in your lab now, if you collect those samples now, we can go back and look at them retrospectively and compare them to what is going on in the future and maybe help make predictions as to how our world will look long into the future. With that, I say thank you, and I will take questions.
So this was the pre-recorded talk by Dr. Peter Thompson. So, we are very thankful to Dr. Thompson for this talk, and so we are having a few questions for you. Dr. Thompson, can you unmute yourself? Is it possible?
Can you hear me?
Hello, am I audible, Dr. Peter?
Can you hear me now?
Yes, perfect. Thank you. So one of the question means the talk was wonderful. And the question is about the developing nations where sometimes we are not able to spend much on such kind of devices which will generate such robust data. So what's your take on that? Means how would we go and try to find out the answers without using this kind of techniques?
So this is an area where I think that first world countries need to take responsibility. We in the US have suffered some of the worst economic outcomes from COVID-19, and therefore we should look at providing such devices to the developing world as an investment in our own economic stability. I see a world where spending $100 billion will ultimately save us $5 to $10 trillion, and that seems like a worthwhile investment to me. And these are discussions which are happening in the first world. For those of you who are currently do not have access to these technologies, you should look for partnerships, especially people like me who are interested in biodiversity, and reach out to see how you can partner in order to understand what are in your samples. The idea is that we need to know what is out there in the world if we are going to be proactive rather than reactive to emerging infectious disease.
Thank you. One more question is about maybe a bit weird question. So do you know any kind of courses where students can find and go through the metagenomic analysis kind of stuff?
There are many free courses out there on the web. Basically, there is not much training which is involved in terms of doing the lab work. If you have done a PCR, you can handle the lab work. The bioinformatic work is fairly straightforward these days, and there are quite a few people who are working to what we call containerize these bioinformatic applications. So they will basically package a group of softwares into a single download, which you can then basically activate that container of software, and you can follow instructions from step number 1 to the finish with detailed guidelines. Basically, my recommendations, if you're interested in metagenomic analysis, is I would look into the CHIME or Mother/Dada pipelines. These are freely available on GitHub, and there are a number of places where you can run them. A standard computer from the last 10 years should be able to run any of these analyses. Sometimes it may take a little bit of time, but if you have patience, you can definitely get through the analyses. They are not incredibly computationally intensive.
Okay, thank you very much, Dr. Peter, for answering these queries. And once again, on behalf of organizing committee, I would like to thank you. And thanks for sending that video, and it was a fantastic talk. We all enjoyed it. And hopefully we will keep you engaging in these events in the coming future also. Thank you very much once again. And thanks for, it's about 5 a.m. There where Dr. Peter belongs to the place. So thank you very much. Sorry for bothering you so much.
Thank you so much for the invitation. It's been my pleasure.
Thank you. Thank you, Dr. Peter. So with this, we are about to conclude this event. So now I invite Dr. Ranveer Singh to please give a vote of thanks to
All right, thank you, Dr. Rajneesh, for a nice one-day session on Zoonosis Day. As you know, we all are celebrating World Zoonosis Day today, and on that, in this behalf, we have organized this one-day session to commemorate this day. And today in the morning, this session was started by our chief guest, Honorable Vice Chancellor Dr. Inderjit Singh, and he shared his view and the importance of zoonotic diseases and why they are important for us and why we need to pay much more attention to them. We were also joined by our 2 guests of honors, Dr. R.K. Singh, who is ex-director IBRI, and Dr. S.P.S. Kuman, Dean College of Veterinary Science. They are well-known personality and in their field, and they should also share their views on zoonosis and zoonotic diseases which are affecting us. Then we had more than 200 participants who registered for this one-day event, and during this entire day, we had about 8 keynote speakers, and out of those 6 speakers were from abroad and representing different countries, different regions, and they all shared their views on various aspects of different zoonotic diseases, such as brucellosis, parasitic zoonosis, then modeling of zoonotic diseases, toxoplasmosis, and food safety issues. Then we also had 2 national speakers from our country, where they discussed about viral zoonotic diseases. And then we also had one speaker who talked about brucellosis. During the day, we also had presentations from students and researchers, and then these two presentations were divided into two sessions, one on bacterial zoonosis and the other one was on viral and parasitic zoonosis. And these two sessions, we had around 6 to 7 presentations in each session, and this sort of presentation was even competitive in nature, where we judge the presentation, and we have competition was very tough, and we are going to announce the best presentations also for these sessions. So we are thankful to all the participants who showed interest and enthusiasm in making this day great and celebrating this World Zoonosis Day, for which we are all known, and it shows the importance of veterinarians in promoting human health and veterinary health. So with that, we come to the end of today's proceedings, and before we leave, let me announce the presentations, best presentations for the two sessions that we had. Now for the first session, that was on bacterial zoonosis. In this session, we had 6 presentations covering different aspects of on different types of zoonotic infections, some related to food and some related to other sources. And competition was very, very tough in this in both the sessions and both the teams. So for first session, we have two best presentations. It was a tie, and the one presentation which was judged as best presentation was by K. Senthil Kumar, rapid and visual diagnosis of canine leptospirosis by recombinase polymerase for amplification assay. And then the second one is by Vijay Sharma on prevalence of brucellosis among goats and associated humans in and on border area of Jammu region, Jammu and Kashmir, India. As far as the presentations in second theme, which was on viral and parasitic zoonosis, in this also there were two presentations which were judged as best presentations. One is by Gurlal Singh Gill, knowledge, attitude, and practice of school students related to rabies in Punjab, India, and then PV Siddhartha, a proteomic analysis of crude and purified hydrated cyst fluid antigen used in immunological diagnosis of cystic echinocosis in sheep and buffalo sheep. So congratulations all the winners, and best wishes to all the participants. Keep showing your enthusiasm and your motivation for participating in many such events. We are also going to have one session on tomorrow. We are having one more colloquium on brucellosis. So I hope all would join us tomorrow also for the colloquium that we are going to hold tomorrow, and we are shortly going to share the link for that also. So now over to Dr. Rajneesh.
So thank you very much, Dr. Randeep Singh, for summing up all that what we had today in this event. And with this, we are concluding this event. And once again, I would like to thank all the speakers, chief guests, Worthy Vice Chancellor Dr. Inderjit Singh, and our guest of honors, and as well as officers of the university, and all the students as well as participants. So thank you very much. Have a great evening and bye for now. Thanks.